that is here hosted. You're in the Jomo Kenyatta University. And he will tell us a bit more about Sino-Africa Joint Research Center. So thank you so much, Professor Gituru, and welcome.
Thank you. Thank you very much. Good morning, ladies and gentlemen. Yes, like you have been told, my name is Robert Gituru, and I'm in charge of this facility where we are this morning. I will welcome you to the Sino-Africa Joint Research Center, but I need to acknowledge that in our presence, we have my senior and the representative, the person standing in for our Vice Chancellor, Professor Jackson Kwanza, and he will welcome you to Jomo Kenyatta University of Agriculture and Technology on behalf of the Vice Chancellor. For me, I just asked for this opportunity to introduce this center where we are hosting this conference today. So like I said, I'm Robert Gituru. I'm director of this center. Let me see whether I'm tech survey, whether this, right, try it down. Let's see. I think I might ask for someone a little more tech savvy. Great. Thank you. This bottom, sour. So this is where we are. This is the Sino-Africa Joint Research Center. This is an aerial view of our center. We are right here, right there. That is where we are. This is our conference hall. This was a picture that was taken using drone technology because one of our laboratories specializes on what we call GRS. So they took that one for us to show where we are. Oh, no. I think I'll have to ask for Paul to, so yes, thank you. Next one. All right. Technology sometimes does that. Thank you. Thank you very much. We start by asking ourselves, what is the Sino-Africa Joint Research Center? The Sino-Africa Joint Research Center is actually a platform of science and education collaboration between China and Africa. And what do we do here? Or what is done at this center? We have two main objectives that we follow or we focus on. Number one is promoting talent cultivation and scientific collaboration between China and Africa. Secondly, we focus on biodiversity conservation, ecological environment protection, food security, and disaster early warning systems. In future, we are likely to add more fields as science continues to evolve and as the needs of society also continue to evolve. But generally, those are the areas that we focus on in the Sino-Africa Joint Research Center. Let's go to the next one. Now, the structure of the Sino-Africa Joint Research Center, like I said, let's go a bit. Like I said, this is a collaboration that fosters scientific and education cooperation between China and Africa. So on the Chinese side, the host institute is our university, Jomo Kenyatta University of Agriculture and Technology. And on the Chinese side, the host institute is Wuhan Botanical Garden Chinese Academy of Sciences. So the two of us, that is JKUAT and WBG-CAS, we together run these headquarters of the sajorec facility here in JKUAT. The research fields that sajorec focuses on are listed there: biodiversity conservation, modern agriculture demonstration, natural products development, energy and water management, microbiology and microbial control, and geography and remote sensing as a field of study. Who are the collaborative partners in the Sino-Africa Joint Research Center initiative? On the Chinese side, we have quite a number of institutes. Of course, the one in bold is Wuhan Botanical Garden, Chinese Academy of Sciences, which is our counterpart in the running of this center. But there are plenty of others. We have listed them there by their acronyms. Then on the African side, of course, the bolded one is our university, JKUAT, which is actually the core host together with WBG. Of course, there are many others there from Kenya, National Museums of Kenya, Kenya Wildlife Service. We also have organizations from Tanzania, Tafiri, Tanapa, from Ethiopia, from Madagascar, from Senegal, from Mauritania. So we have partner institutes also in the sajorec initiative. In addition, we have international organizations that also participate with us in various research and other programs. These include UNEP, ANSO. ANSO stands for Association of National Scientific Organizations. They have been very helpful. Then we have TWAS, Third World Academy of Sciences. We also have Africa Academy of Sciences, ILRI, ICRAF, and several others. So we collaborate with them to achieve our mandate, that is education and research. For example, TWAS usually gives scholarships to students, therefore supporting our talent cultivation. ANSO every year also awards scholarships to many students, therefore supporting the objectives of sajorec. Let's go to the next one. This is just an overview of our botanical garden. For those that may not be aware, right next to where we are, on my left, that should be on your right, we have the JKUAT sajorec Botanical Garden. It's 37 acres just for research, for training and learning. So we have the garden, it has various theme gardens, and I may add that you as participants in this conference are free to walk into the garden as a complimentary from this center. We won't charge a dime. You can walk in there, enjoy yourself, breathe the fresh air, and think about genomics and the future of humanity. Yes, those who are inclined to the religious side, there are some quiet corners that you can talk to the creator. All right. So it's a beautiful venue. Of course, it is a work under progress. A botanical garden takes a long time to achieve its full functionality. So if you see some shortcomings here and there, remember it's work under progress. And if you have an idea on how we can make it better, it's very welcome. So that's just an area view of our botanical garden. And inside the botanical garden, right there, we have what we call the African Pavilion. People like to call it the Makuti. Yes, that is another place where you have the opportunity to sit there and reminisce about the African culture. That's the African Pavilion. And opposite it, there is what we call the Friendship Pavilion. Some people call it the Chinese Pavilion, but it is actually the Friendship Pavilion. It showcases, traditional Chinese architecture. So for those of us who have been or who have lived or studied in China, it will remind you of your good old days. Other theme gardens there are the Aquatic Garden and several others that you get a chance to see when you walk there. Next. Of course, like I said, this is a research and education center. So laboratories are central to research. So we have a number of labs that are equipped for us by the Chinese Academy of Sciences. I would say that they are quite well equipped, but like I said, it is still work in progress. So we are progressively increasing the equipment and machinery in the labs. I know in the course of this conference, you will get an opportunity to go to our labs and even to probably do some work within our laboratories. So we have a lab that deals in molecular biology, another one that deals in water resource management, a lab of natural products development, laboratory of biodiversity conservation. By the way, this is my my pet, because that's where I do my own work. Then we have the laboratory of microbiology. And like I mentioned earlier, we have the laboratory for remote sensing and geographical science. The next one, kindly. Now, I happen to be a biodiversity scientist. That's my area of training. I'm a botanist specializing in biodiversity conservation. So I could not help but put this slide because I happened to be there when we took the picture, and I'm proud of it. So biodiversity research is one of the major things that we do in this center. But I may not go into all the details. I'll just mention something that I know is relevant to this particular conference. Let's go to the next one. I know you are people interested in genomics and genomic research. In Sino-Africa Joint Research Center, we are equally interested in genomics because we know that is the future of science. It's the cutting edge. So we also do, among many other things, we do some genomics research. And we have achieved quite considerable progress and quite considerable expanding of the boundaries of knowledge. For example, in 2021, we had a sajorec team working on the genome of this plant, welwitschia. Welwitschia is a plant that grows in the desert. You find it in Namibia, and people like to call it the everlasting plant because it has an incredibly long period of life. The longevity is just amazing. So we thought that it would be useful to study the genome of that plant, which we did as sajorec, and our work, we were very happy to find that the work was published in Nature Communications in the year 2021. So that is genomics. More recently, we have taken interest in the genus Adansonia, the baobab tree, mabuyu, as we would know them in our local language. And we were interested in the evolutionary history of the baobab because of its unique distribution in Africa and also in the island of Madagascar. So we conducted a study as a sajorec team, of which I was a member, and we were glad to have our work published in Nature just a few months ago. This was in the Nature issue of May. So this is to show that just like you people, we in sajorec are interested in genomics because it is the future of biology. Next one. Like I said, training the next generation of scientists is key to the sajorec initiative. Because in my local language, we say that if you have a herd of cattle and you have just big bulls and big cows, then that herd is set to disappear because you don't have young calves. So using that thing or that saying or that thinking, from the onset, we decided that training of young scientists, young researchers will be a fundamental pillar of the Sino-Africa Joint Research Center initiative. And so far, we have, or rather, Sino-Africa Joint Research Center has enrolled a total of 294 international students from 14 African countries. And these students ordinarily get scholarships to institutes within China, mostly under the Chinese Academy of Sciences, and they go and study their master's and their PhD programs and postdoctoral programs. And one thing that we are very proud about with regards to this project is that 100, if not 101% of the beneficiaries of these trainings all come back to participate in science here in Africa. Unlike other programs that I wouldn't want to mention where we have a serious challenge with brain drain, for this program, we've been very lucky that we get all the trainees to come back here and participate in the building of our country and of our continent. Here with us today, there are few. They're not few, quite a number. I can see them. I don't want to point them out by name, but they were beneficiaries and they are doing a wonderful job. In fact, The gentleman who is operating this equipment is one of the beneficiaries, and he has done very well. We are very proud of him, and we wish him the best so that he will be a good example of the products of sajorec. Let's go to the next one. Yes, like I said about a herd of cows having only bulls and grown cows. Yes, you don't ignore the grown ones. You need also to keep updating and elevating their levels of knowledge. Therefore, the Sino-Africa Joint Research Center also conducts trainings for technical staff and managerial staff in the fields of science. And the picture that we have here are some trainings. In fact, some of them have been held right here in our center. Here you can see where some farmers being trained in the growing of millet. So we do a lot of those. Some are trained outside in China, and especially after the the COVID pandemic, we have increasingly sometimes been using online trainings just to update the skills and knowledge of researchers, scientists, and even local farmers. So we always look into the future. So the question we have to answer is, sajorec is what? Where do we see ourselves? Which direction do we want to go as our center? And we say we intend to extend sajorec's scientific network inside and outside of Africa. We want to continue to build robust research teams in various disciplines and develop young talents. Our areas of interest, like I said, biodiversity conservation, ecology, modern agriculture demonstration, food safety, environmental protection. So that's just a brief outline of who we are as the Sino-Africa Joint Research Center. We may not have had enough time to do justice to that topic, but I am here. I'll be here throughout this conference. And if you need some more information, some more clarification, if you probably would want to, apply for a training or a scholarship, I'm available, and the rest of the team are available, and you are most welcome to come and see us. Like I said, you are also free to explore our center, the Botanical Garden, and even across our gate, we have 10-acre modern agriculture demonstration area where right now you can see greenhouses under renovation. As a guest of this conference, you are also free to walk in. I know some of us here are not only genomic researchers, but they are also farmers in their own right. So don't be shy. Walk in there and ask, hii mahindi inapandwa namna gani? Yes, because we wear many hats. Thank you very much. And I'm glad that you gave me your ear. At this point in time, I have the pleasure to invite my senior. Actually, he's my immediate superior in the Division of Research, Production, and Extension. He's the Deputy Vice Chancellor, Research, Production, and Extension, Jomo Kenyatta University of Agriculture and Technology, and also a scientist in his own right. He's in the mathematical sciences, an accomplished scientist as well. This morning, he's here to represent our Vice Chancellor, Professor Victoria Wambui Ngumi, who had to be away for another engagement. We have her blessings, and that's why she has sent Professor Kwanza to come and represent her. Kindly a clap as we welcome Professor Kwanza to address us.
Thank you, Professor Ngituru. As you have heard, my name is Professor Jackson Kwanza. I am in charge of the Division of Research, Production, and Extension, where sajorec falls. We have 3 centers of excellence in the division. We have Sino-Africa Joint Research Center, and we also have Institute of Energy and Environmental Technology, and we also have an Institute of Biotechnology Research. As has been said, I'm here to represent our Vice Chancellor, who would have liked to be here, but because of other engagements, she could not make it. And she has she's supporting this conference. That's why I'm here to represent her. And before I read her speech, as Professor Ngituru has said, I'm also a researcher in my own right. I'm actually a professor of applied mathematics. And as in mathematics, we say, there is no science without mathematics. so I feel I'm at home whenever we are talking about science, whether it's biological sciences, physical sciences, even applied sciences, as people, especially in applied mathematics, because we apply mathematics in biological sciences, in physical sciences, even in engineering and so on. It's like it's applicable everywhere. And my area of specialization is fluid dynamics. And I think also that is a very, very wide and important area, because starting with what is free in the world? I think it is air. I don't know of a place where air is a sport or there is trade about air, and air is a fluid. And I'm actually a specialist in fluid dynamics, starting with air, of course, name all the fluids. In front of me are serious scientists who know what fluids are. Now, having said that, let me read the words of our Vice Chancellor, what he would have told you officially if she came. Our Vice Chancellor by name is Professor Victoria Wambui Ngumi. She's actually a botanist. I'm sure she would really have enjoyed being with you here, because what you are going to do in the next 5 days is what she likes best. Now, this is her speech. Professor Anne Muigai, chairperson of the East and Central Africa BP, that is BioGenomic Project, local organizing committee, members of the organizing committee present, Professor Robert Kituru, Director Sajorec, presenters and workshop participants, ladies and gentlemen. Good morning. It is a great honor and privilege to welcome you to the Jomo Kenyatta University of Agriculture and Technology during the second East and Central Africa BP Open Institute for Genomics and Bioinformatics workshop organized under the African BioGenome Project. This workshop, running under the theme Sustainable Pathways for Biodiversity Conservation and Bioeconomy, Integration for Growth in Eastern and Central Africa, underscores a tacit commitment to foster capacity building among early and mid-career scientists in genomics. And allow me to interject something which Professor Ngituru talked about here. He talked about in their community, they talk about, I mean, if you have bulls and grown-up cows, and then you don't have young ones, I think you are threatened with extinction. I'm happy there is focus of building capacity among early and mid-career scientists in genomics, because it's good as we age, we also mentor young people to take over. I applaud the noble efforts being championed by the African BioGenome Project, geared towards driving scientific collaborations and partnerships, enhance human and institutional capacities, and meticulously sequence the genomes of Africa's rich and diverse species. It is gratifying to note that the objective of this workshop is to foster capacity building of early and mid-career scientists in current and emerging and genomic technologies. The Africa's BP's mission is clear and profound, built on a strong commitment to develop digital innovations that foster a sustainable environmental and conservation network, uniting interdisciplinary participants across the continent. One of your objectives is to deploy relevant skills and technology transfer mechanisms throughout Africa, ensuring the scientific community is equipped with cutting-edge tools and knowledge, as well as establishing a comprehensive functional database that will ultimately provide critical information to advance research and conservation efforts. I wish to encourage the project leadership under Professor Anne Muigai for remaining committed to explore and exploit the applicability of machine learning algorithms to enhance and advance the genomic studies. It is worth noting that you have taken a progressive step towards developing optimal DNA sampling protocols tailored specifically for agricultural and biodiversity systems, ensuring the highest standards in research methodologies. These efforts should be supported, and we are happy to work together under this initiative. I am delighted that in our midst, we are honored to have young academic staff and recent graduates from JKUAT, our sister universities, and the local research institutes. Your presence here today signifies a collective dedication to advancing the frontiers of science and research in our region. I am confident the knowledge and skills the participants will gain from this workshop will significantly enhance research in your respective institutions, driving forward our shared vision of a prosperous, biodiverse Africa. The insights you gather here will not only benefit your personal and professional growth, but also contribute to the broader scientific community and our collective efforts towards sustainability and conservation. As you interact in the next 5 days, I encourage you to network actively. Please don't walk out of this room without knowing the person seated next to you. You should interact and network so that you have friends across Africa. Your collective insights are vital to the success of African BP and the broader goals of biodiversity conservation and bioeconomic growth in our region. As I conclude, I extend my heartfelt gratitude to the JKUAT alumni and indeed the three committees of the project for their invaluable contribution and dedication to this initiative. Your continued engagement with the university community and other stakeholders, such as the one presented by this workshop, is highly appreciated and instrumental in fostering a robust academic and research environment. Once again, welcome to JKUAT. I wish you successful workshop, deliberations, and rewarding experience. Thank you. So I am not going to add any other thing. After our VC has spoken, we stop there.
Thank you. Thank you very much. All right. Thank you very much, Professor Gituru and Professor Kwanza. We feel welcome to the institution and to sajorec. And I may not have mentioned that Professor Gituru is not just the director of Sino-Africa Joint Research Center. He is also among the founding members of the center. And through his work, he has mentored very many students. He has published over 100 research papers and contributed to 3 books within his areas of interest, which is plants conservation as well as use of plants in medicinal research. So I am very grateful that you have invited us to feel free and to visit the Botanical Garden when we feel a chance. And this institution, as we have seen, has beautiful labs that are fully equipped that we also hope to get a chance to visit. Now I would like to invite Professor Anne Muigai, the chair of the Africa BioGenome Project, to give us a welcoming speech to the members here today, as well as those online. Those online, thank you for joining us. We hope you're keeping up with us. We had a chilly morning here in Kiambu. The weather has just turned and we are glad it's sunny. So Professor Muigai is a professor of genetics. She's the Deputy Vice Chancellor, academic and student at the National Defense University in Nakuru. She's passionate about research and has had several teaching and administrative positions in the Kenyan public university sector. She currently sits on the Bayer Ethics Council and was a member of the WHO Advisory Committee on Governance and Oversight on Human Genomic Editing between 2019 and 2021. She holds an MSc in plant science and a PhD in population genetics and molecular biology from the Jomo Kenyatta University of Agriculture and Technology. Professor Anne, you're welcome and thank you.
Thank you very much and good morning from Nairobi, Kenya, and to all of you who are online, karibuni sana, as we would say. My name is Professor Anne Muigai. I'm the chair of the African BioGenome Project, and I'm extremely delighted to have been in the presence of our Deputy Vice Chancellor, Research, Production and Extension, Professor Kwanza, and we're extremely very grateful to sajorec and the directorship with Professor Gituru for having hosted us here. So Bwana DVC and director, we have 200 participants joining us online. And here in the room, I can see we have about 100, give or take. So, and we anticipate as the day goes on, we will have many join us. So I want to take a few minutes just to elaborate on this great initiative known as African BioGenome Project. And as you can see, it's collaborative effort of many scientists across Africa. We are over 300 active in this particular project, and we are performing the project and our roles and responsibilities from wherever we are on the African continent. This is what I will tell you about what our goals are, and we can move on to the next slide. So we aim to coordinate all non-human genome sequencing efforts that are available going on in the continent. And that is because the sequencing of the African Human Genome Initiative is going on very, very successfully. So we don't want to duplicate that. What we want to do is everything else that they are not doing. And we also want to act as a forum for deploying data, genomic and bioinformatic data that is made available through these sequencing efforts. We also want to initiate, develop, and progress any sequencing efforts and coordinate them from a central point so that whatever is being sequenced only needs to be done once. We don't need to duplicate it. And we also very, very much want to start our own African databases, start the conversation about where should this sequence data be stored, who should host it, where should it be hosted, what sort of infrastructure do we require, and how can that data be accessible to all the Africans who want to use it to further knowledge, to use it to make vaccines or develop whatever it is that knowledge is available for. Next slide. Our project hopes to cover all eukaryotic species. So all of you who are here, who are working on a species, eukaryotic, but that means it can be bacteria, it can be microscopic, it can be, macroscopic, animals, anything, insects. You come with your species and let us sequence it and let us do it properly so it never has to be repeated ever again. So we will cover all eukaryotic, but we want to do it systematically. So as we apply for funding, we will be able to lock in species. So right now, we have started with the vertebrates of Africa in collaboration with the Vertebrate Genome Project, coordinated from the US, University of Rockefeller. We have also initiated 10KP, so they're plants that are being sequenced, coordinated from West Africa, University of Port Harcourt with collaborators in the US. And we will move on, depending on the mini research groups that we will form. Next slide. So we find that they are very, very strong Africa to developed world collaboration. So people here in Kenya, in this university, are collaborating with people in the US, in people in China, in people wherever it is. But within Africa, our networks are not very strong. So one of the goals of this project is to increase and foster collaborations within ourselves. Because when we harness what we have, our capabilities, not only will we be stronger in terms of pitching for grants, but we will also be able to bring our resources together, our knowledge, and build on the capacities for that. We also want to ensure that any policy initiatives that are being put forward have our input, because we are the ones who are on the ground. Next slide. So I want to give you a little timeline. So this project started before 2021. We started way back in 2019. But that was just the conceptualization and the conversations that we're beginning to have with partners around Africa and outside. But we launched it in June of 2021 as the African BioGenome Project. And immediately after that, we launched the pilot phase. So the pilot phase just ended in March 2024. It was a 3-year pilot phase to be able to write up our policies, to develop our infrastructure, to develop any of the sequencing infrastructure that we needed. And once that has ended, and I will show you some of the genomes that we've been able to sequence, we are now going to replicate that, and we are ready to replicate it and duplicate it across the continent. So now we are going full on to sequence the 100,000 genomes that we promised to sequence in the next 10 years. And I think we're making very good progress towards this. Next slide. So we have 4 programs within the Africa BioGenome Project. We have the Nelson Mandela African Genomes for Conservation of Nature. Within this particular project, our focus is to sequence all vertebrate animal species within Africa. The next one is the DAISY Africa BioGenome Project for grassroots conservation. And within this, we want to sequence all African plant species, so that are use to us. So it could be a plant that is endangered, it could be a food crop, it could be a crop that is has a cultural significance. So within that, we hope to sequence those within that project. Then we have the AfricaBP community roundtable of practice. Within this, we want to be able to have a community through which we can be able to discuss issues around the Nagoya Protocol. What does it mean for us? How are we able to make sure that it's applicable to me in Kenya or you in Nigeria, wherever you are? So by learning what your law is in your country and what the law is in my country, then we're able to actually talk about access and benefit sharing. What does it mean? How are we able to share the proceeds of what we are doing with the people who are on the ground? And then the AfricaBP Open Institute for Genomics and Bioinformatics. This is the, if I can use the word, the academy, the training wing of the Africa BioGenome Project. And this is what has brought you and me together here today. Next slide. So let's talk about our highlights. We published, next slide, we published our position paper in Nature in 2019. And here is where we put out our dreams and our goals and our objectives of sequencing 100,000 species. The goal is to safeguard biodiversity. The goal is to be able to monitor what is happening. And the goal is also to protect what we are doing. So this was our position paper. We committed to do this in 10 years, and we costed it at $100 billion. Now, many people ask, why would Africans who have so many competing interests, food, we have competing interests of provision of Medicare, sanitation, we have climate change hitting on us, why would we invest this kind of money in something that is, can I use the word, not so useful? Actually, it's the other way around. Now that we know that we have this challenge of climate change, that our species are beginning to be affected by the environment, we need to be able to deploy technology, to deploy genomic data, to be able to breed better plants, to be able to breed faster growing animals, so as to be able to provide better, quality food and even more. So this is an investment into our future. So it's not money that goes to waste. Next slide. The African BioGenome Project, I must say, has been very, very well received around the world. We've been invited to many fora, including the United Nations, the World Science Day, the US Summit on Africa, to be able to talk about our project. So it was received with a lot of respect, and also acknowledging that it's a project by Africa and by Africans for Africa. So we are very, very delighted with this. So we have recognized that for us to be able to sequence those 100,000 genomes, we will generate over 200,000 gigabases of DNA sequence. And I'm very, very happy that we have the chair of our sequence and annotation committee with us, Yves Hernandez, if you can just wave. He'll be the one who is responsible for ensuring that all this data is captured. But the question is, how will we be able to do it? Do we have the Africans? It's very easy for us to export these sequences, export the DNA outside, and it will be done elsewhere, and for maybe reasonably less than what we would do it locally. But the downside is, in the long term, we lose as Africa because we have fewer people who are able to do this hands-on on the continent. We have no databases where this information can be stored. And we have serious infrastructural gaps that we'll be able to bridge. So we thought as we advance and begin to sequence these 100,000 genomes, it's important for us to start looking at where are the gaps. The gaps are institutional infrastructure. So I'm happy to see that already we have infrastructure in an institution like Jomo Kenyatta, like Sajorec. We have the genomic labs, we have the bioinformatic labs, but maybe in other institutions, we will need to be able to transfer some of this technology. Then where are the people? The people are not there. And that is why we need to bring you on board, to be able to train young early career scientists like yourself, like myself, to be able to provide information for genomics and bioinformatics capability. Next slide. And that is what led to the institution that is, we are now calling the AfricaBP Open Institute for Genomics and Bioinformatics. We've published our first paper in Nature Biotechnology in 2023, and this is where we put out the roadmap of how we're going to bridge the information and capacity gap. And I'm very, very happy to note that just in last month in July, we published our third paper in Nature Genetics. Now, reporting what we have done between 2023 and 2024, especially in the training. In these particular trainings, and I'll show you, we were able to reach more than 4,500 African scientists around the regional continent through harnessing the power of technology. And then in hands-on practicals, we're able to train more than 400 African scientists just on genomics and on bioinformatics. So it's, when we think about where we're going, we still have a big gap, but to be able to do that in 1 year has been really, really profound. Next slide. So this is what we did in 2023. We held 4 regional workshops, exact duplicates and replicas of this. West Africa held its first workshop in May this year also, which is now the series for 2024. And we registered more than 3,000 participants, so we can actually see the drive, the number of people who want to come. For our practical hands-on training, we registered over 3,000 applicants, but we only had 300 slots. We pushed that by another 100. Can you think about 100 people in a lab? Those are many people. We pushed it up by 100, but it's still short of the 3,000 people who applied. So that shows you what the demand is. And that's what our institute is going to do. We're going to successively, through regional workshops, be able to run these workshops around the continent and follow through with longer fellowships to be able to drive this. So in North Africa, South Africa, East Africa, so we hold these workshops, and the workshops are all the same. We have 2-day style conferences held on a Monday and a Tuesday. And then the following day, next slide, we have hands-on practicals. And the practicals are free, completely free for our participants. And we really, really rely on our technology companies. They partner with us to provide the reagents, to provide the resource people, and to provide the hands-on capability in their labs. And I hope you'll have time to go around and meet them. We have them exhibiting here, and they run this for free. And for us, that is a great benefit, because that is the only way we will be able to offer this to you. So these are examples of these practicals that happened around the world. Next slide. And for us in East Africa, we have teamed up with West Africa to make one region. And therefore, we will have a hands-on practical in University of Kinshasa, and also in Cameroon, in our facilities there. So as our Vice Chancellor has said, the impact of these workshops is immense, but the training needs are very, very high. And here we have examples of what you walk away with. The participants will walk away with a certificate after the 2-day conference hearing. And after the 3-day hands-on practical, you also get a certificate. I think it's the next slide. So what is unique about our workshops? They are openly accessible. We provide them for free. We leverage on the partnerships of technology companies and well-wishers to provide the resources, the infrastructure. And I'm very, very happy that Jomo Kenyatta and Sajorec have been able to provide this facility for free for us to be able to offer it at no cost to yourself. Of course, the laboratories will have to be financed through other models, and that's where our technology partners come in. So let me tell you about our sequenced genomes. So far, we've been able to sequence, right now, I think we're on 20. Most of these have been sequenced in South Africa, and we're waiting for an instrument to be placed in Nairobi, Kenya, in Sajorec, and other public universities. So we have indigenous African livestock being sequenced. You can see the Nguni goat, Bosmara, the sheep, the cattle. We also have indigenous chicken. We have some tilapia. We have the two herbal species, and we also have the bird species. So as we continue with this, we hope to be able to sequence your species. You apply for a grant. put that you want to do whole genome sequencing, cost it. We will provide the technical support for you to be able to do this. We will provide the labs, and that way we'll be able to sequence your species for you. As this is to show you that the genome quality is really, really high. It's high, state of the art, is acceptable all over the world. Yes. So what are our plans? Again, we will have 4 workshops this year. West Africa have already held theirs. We are holding ours now. South Africa will hold theirs in September of 2024 towards the end of the month, and North Africa will hold theirs towards the end of the year. And through these platforms, we hope that you will be able to move with us, because you're the future of genomics and bioinformatics on the African continent. We also want to say that with the partnership of your institution, those of you who are not a part of Jomo Kenyatta or part of the other institutions, make sure that your institution signs an MOU with us. Then if the institution signs a memorandum of understanding with us, you as a student will be eligible for fellowships that are free to the student, that are with partnerships. So these are a number of fellowships we've been able to offer. These ones were held in Mohammed VI Polytechnic in Morocco. We had students from around Africa apply. We shortlisted them, flew them to Morocco. They spent 7 days learning hands-on genomics for free. Yeah, if you have insurance, get your university to pay your ticket. We will cover the rest of the cost. Next slide. So I want to leave you with some food for thought. It is evident that we can sequence high quality genomes on the continent. We've been able to show this, right? But it is also evident that if we want to move very, very fast, maybe the easiest way for us to do it is to sequence off-site, off-continent even. But this, like I have said, will only address a short-term goal. That we will get our sequences, we'll be able to publish in Nature and wherever it is, but we'll have had very, very little input on the ground. So we want to go slowly to ensure that as we build in capacity, the capacity that we're building, you're the one who's carrying on this sequencing and genomics training. And when you go on, you bring on other younger scientists with you, yeah, because by then you'll have had your PhD. Then with your PhD, you can have a master student, and you transfer the technology. What this will do is to ensure that this infrastructure is fully utilized. So if Professor Gituru here, for instance, buys that very, very high-end equipment, and it sits here for 5 years without being used, what will happen to it? Yeah, it will get broken down. It will also never work. So only if we are able to utilize these resources after they've been placed. So the investment is really huge. And therefore, we have to make sure that it is utilized. And that by doing this, we are able to ensure that the sustainability of this genomics and bioinformatics infrastructure on the continent. So this is Dr. Thangod, who is my co-chair, the co-chair of the African BioGenome Project. And he said how many scientists sent their samples off-site, but they're not proud of it. Many of us have perhaps been guilty of sending samples to Europe for sequencing very quickly, but you're not happy about it because you want it to be done locally. Again, the idea that it gives only a short-term gain and not a long-term gain. So how many scientists complain about limited capacity in Africa, yet fail to lay the foundation? And I'm happy to say that through his idea, the African BioGenome Project was the brainchild of Dr. Thangod. So through that, he's laid the foundation to actually establish this great project that is now changing the face of genomics around the world. So we need to be able to train, but even as we train bioinformatics and genomic scientists, wherever it is, we have to be able to retain them. Professor Gituru talked about brain drain, that we train people, or the Chinese train people, and then they disappear somewhere in Europe or in America or wherever it is. But if we have genomic infrastructure on the continent, we have a lot of work. There's a lot of work. We have many genomes to sequence. There's a lot of science that we can actually do on the continent. And it is only through establishing projects like this, using your support and your interest in this particular project, that we'll be able to retain and pay people on the continent for the work that they're doing. And I thank you very much for joining us on this initiative. And I really, really hope that you will join us on this effort and bring your colleagues. In fact, maybe we should next time move to the KICC because we can't fit into Jomo Kenyatta because of the numbers of people who want to hear about it. And thank you very much. I look forward to a very, very fantastic week of presentations. Thank you.
Thank you. I hope we are excited. Are we? Are we? When we come together for to work towards a goal, we can achieve tremendous things, right? So now we're building infrastructure, we're building capacity, and we've identified what we want to sequence, right? For the future of our continent. I love that we sequenced our indigenous chicken. Now we can figure out why it's sweeter than the other one. So yeah, we are running slightly late. So our next session, we will be taking our group photo. So we will go out to the reception where we registered when coming in. And then after that, we will have our tea break. Tea break will be served on my left, your right. And we will keep it brief because we are running a bit late on our schedule. So we will start with the photo session so we can make our way there. And then I will call you back in when I'm ready for you from the tea. So for the online participants, so it is now 10:18. Our group photo, let us hope we can take about 10 minutes. So that will be 10:30 and we can have tea for 15 minutes. What time will that make it? 10:45. So we will back here at 10:45. That is when we will begin our next session. So thank you very much. Let us make our way to the group photo at the reception.
Welcome to Africa. This new website features source, simple and intuitive, with relevant content. In today's video, I'm going to take you through the whole process of ordering a product, the same website. So to start off, Africa Biosystems as a company focuses on key sectors. Right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of our products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click on either of them, we get to find even more categories, subcategories inside. Okay. So in today's demonstration, let's go straight to life sciences solutions. Okay. And we are actually here on the life sciences group category page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division. Okay. So I'll just go straight to biosciences division and just click that. From here, I'm able to see some of the products under biosciences division. Come to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updating. In today's video, I'm going to take you through the whole process of ordering a product in the same website. So to start off, Africa Biosystems as a company focuses on key sectors, right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of our products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service and support, and lastly, we have general lab products. If we click on either of them, we get to find even more categories, subcategories inside. Okay. So in today's demonstration, let's go straight to life sciences solutions. Okay. And we are actually here on the life. Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated. In today's video, I'm gonna take you through the whole process of ordering a product on the same website. So to start off, Africa Biosystems as a company focuses on key sectors, right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of our products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click on either of them, we get to find even more categories, subcategories inside, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And we are actually here on the life sciences group category page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just go straight to biosciences division and just click that. From here, I'm able to see some of the products under biosciences divisions, and quickly, I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I can just come here and click this fetal bovine product. It takes me now to the product page for fetal bovine serum, FPS. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and availability. And that's how you actually make an order on this website, okay? So to make an order or even request for price, you just click on this button here, which takes you straight to the checkout section. And from here, this is where you've been asked a few questions before the order is actually placed. Yeah, so when you're here as a client, just get to fill in a few information. So I'll just do it quickly. Probably I'll just say I'm from ABC company. Then just put in an email. Yeah, ABC company. Let's say the email. Then I'll just put in my name. So maybe John. Maybe it's John. And my last name is what? John Awad. Here I can either put in the phone number. I think the phone number is actually optional, but actually good, so that the sales team are able to get to the client in the quickest manner possible. Okay, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. The next thing is just to place order as the client. So I'll just click here. The order is being processed, and shortly the process should actually be successful. Yeah, so as you can see, as a client, I'll just get this message. Thank you, your order has been received. And it will go now to the relevant department of the sales department, so that they can now check back with the client, get to advise exactly how much the costings and everything. Yeah, so that's it for our presentation. I hope everything was helpful. Life sciences table page. When you come to this page, you get to see three Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated. In today's video, I'm going to take you through the whole process of ordering a product with the same website. So to start off, Africa Biosystems, the company, focuses on key sectors. Right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of our products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click welcome either of them, we get to find even more categories, subcategories inside, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And we are actually here on the life sciences group category page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just go straight to biosciences division and just click that. From here, I'm able to see some of the products under biosciences divisions, and quickly, I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I can just come here and click this fetal bovine product. Takes me now to the product page for fetal bovine serum, FBS. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and availability. And that's how you actually make an order in this website, okay? So to make an order or even request for price, you just click on this button here, which takes you straight to the checkout section. And from here, this is where you're being asked a few questions before the order is actually placed. Yeah, so when you're here as a client, just get to fill in a few information. So I'll just do it quickly. Probably I'll just say I'm from ABC company. Then just put in an email. Yeah, ABC company. Let's say the email. Then I'll just put in my name. So maybe John. It's John. And my last name is what? John Ward. Here I can either put in the phone number. I think the phone number is actually optional, but actually good, so that the sales team are able to get to the client in the quickest manner possible. Okay, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. The next thing is just to place order as the client. So I'll just click here. The order is being processed, and shortly the process should actually be successful. Yeah, so as you can see, as a client, I'll just get this message. Thank you, your order has been received. And it will go now to the relevant department of the sales department, so that they can now check back with the client, get to advise exactly how much the costings and everything. Yeah, so that's it for our presentation. I hope everything was helpful.
In 2020, AfricaBP introduced a single system, which allowed us to have qualification 8 times more than in the single system.
Professor Kanganezin Mapoli, Deputy Executive Dean at the College of Agriculture and Environmental Sciences at the University of South Africa. So this is very excellent news for us and a good opportunity for us seeing the PacBio platform actually growing in South Africa, especially now the acquisition of PacBio Radio. Thank you.
My name is Matati, an NGS lab technician here at Illumina Biotech. Using the new review system, fewer kits are required in library prep up to sequencing across the various applications we provide. Using the new streamlined workflow, library prep is quick and efficient.
My name is Howard, part of the NGS team at Illumina Biotech. And one of the stuff we had data a couple of this week. And one of the advantages of the system that not only does it produce strong, high quality reads, but when you actually work with these reads, it gives you one of the most comprehensive views of the genome. If your data not only do we have access to the genomic landscape of the genome, we also can zoom down into single nucleotide variations, which is very important for the variety of applications. So very excited based on top of this.
In addition to the radio system, which is our long-read platform, I'm thrilled to introduce to you the new PacBio system, which is a short-read platform. The auto system is based on the sequencing by binding chemistry, which is a major contributor to the type of data that you get. By the type of data, I mean the Q40 plus level of accuracy. The independent closer lanes and the external clustering brings a level of flexibility that is never seen in the short read market before. The accuracy will allow our customers to sequence at much reduced depth and also reduce the cost of sequencing. We are so thrilled to have this system and start offering the services to our customers across the African continent.
So good morning again after that short break. One of the goals of Africa BioGenome Project is to develop capacity of young early career scientists. One of the ways we are fostering that in AfricaBP, especially during the workshops, is to give opportunities to our early career scientists to make presentations. So you realize for the presenters, and I encourage you for those regional workshops that are yet to happen, indicate whether you would like to give an oral presentation or a poster presentation. The posters of our young early career scientists will be on display on this side of the hall. So as you're walking around, please pay a visit. I also hope that you have noticed, that we have some recycling bins at the end of the room with partnership with Taka Earth and Coca-Cola. We want to make this workshop a sustainable, environmental, green workshop. And therefore, we urge you to drop your waste inside that bin. And at the end of the workshop, we will be able to give you a sustainability report to tell you how much waste you generated in 2 days and what the composition of that waste is. And that is through the generous partnership that we have with Taka Earth. So even your serviette, drop it there, your soda bottle or your water bottle or any paper, it will also analyze it for you and tell you that this is a plastic waste, this is a paper waste. So we will now be able during this conference to have presentations from our early career scientists. But before that, I would like to welcome Professor Dickson Andala, who is a keynote presenter, and he'll be giving talk on sustainable research funding ecosystem. So you have noted from our theme, we are actually looking at how we can make not only our research sustainable, but also our activities as we go on. So Professor Dickson Andala is a professor of material chemistry at Multimedia University of Kenya. And currently, he's the Chief Executive Officer of the National Research Fund. And you know the National Research Fund is the government agency that is given the custody of managing funding for research. And we have all, I hope, applied for funding from them. The National Research Fund's mission includes supporting projects that address national priorities and enhancing STEM fields through funding, resource mobilization, and capacity building. So Professor Andala has been instrumental in shaping science, technology, innovation policies in Kenya and across Africa. As the CEO of National Research Fund, he is responsible for the strategic and operational leadership of the fund, which is a vital resource for Kenya to promote research and innovation. Under his stewardship, National Research Fund has grown from strength to strength, has strengthened the research infrastructure and the technology infrastructure within the country for sustainable development. That is so important because whatever development we do has to be sustainable. He is also instrumental in research on commercialization and innovation and has instituted innovation hubs which are resulting in significant societal impact. Professor Andala is a strong advocate for leveraging on partnerships between academia and industry to create sustainable employment opportunities and also elevate the quality of higher education. He received his PhD in material chemistry from State University of New York in Birmingham and completed a postdoc in Pennsylvania State University. So Professor Andala will be making his presentation virtually, and I welcome you, Professor Andala, to go ahead. Thank you very much. Professor Andala, we can see you. Kindly unmute yourself.
Thank you. The host was not allowing me to unmute. I think I've been given the rights. May I also be allowed to share my screen?
Yes, Prof, we are working on that.
It's still disabled.
Give us a minute, please.
Hello? Okay, email to you. I'll tell him email. Okay.
So as we're waiting for the presentation to be sent to us so we can load it from here, Dr. Cecilia has asked me to give an interesting story. I don't, under pressure, you can't think of an interesting story. But in our audience, we have a distinguished member of the African BioGenome Project, who is the sub-chair of the annotation, sequencing and annotation subcommittee. Please come and introduce yourself. I know you're giving us a presentation later. All the way from Europe, although he's a national of Africa, but he can tell us a little bit more about himself and how you got yourself to become the co-chair of a pilot subcommittee.
Thank you, Prof. Ann. This was a story for tomorrow. I guess this is Cecilia idea. Well, as Professor Ann mentioned, I'm the chair of sequencing and assembly subcommittee. It sounds really big, yeah. Thanks to my two other co-chair at the moment. It appeared to be not that easy. Heavy on me. So I'm Yves Emmanuel Tichwa. I'm, I would say, a product of PAUSTI, JQuat here. I did my PhD here I graduated here in 2021. And next after that, I hope that is good English, sorry. My first language is French. So next after that, I joined AfricaBP, which is the African BioGenome Project, as a member of the Sequencing and Assembly Subcommittee, and I was chair at the time. And with some movement in the subcommittee, I was asked, or I would say I was proposed as the chair of the subcommittee, which I really in the first place, I say this, I'm not sure I can do it because I'm currently a postdoc at Freie University in Berlin with my other, or let's say with what I do every day. I was like this, I can't do it. But with the support of the co-chair who are senior scientists, me as a junior, they say, no, it's okay, you can do it. And this is how they took my hand and I was able to manage day by days with meetings, with organizing meetings, organizing a meeting with partners where we have to, because most of the species that Professor Ann presented, which I will again come on tomorrow during my presentation, are sequenced with the aid of partners. So we partner with sequencing industry like in Kaba, exposing outside, like University of South Africa, VGP, which is the vertebrate genome project. Thank you, a part of the genome project. So we partner with such institution where we sequence our genome, and that's how we, because we do not have, as AfricaBP, a center with a lab equipment, so we have to partner at the moment, or at this, as she said, this is a pilot phase. So it was to show it that we are able to carry out sequencing, and subsequent analysis is being done, like assembly, which is still in my committee, and annotation, which is another committee. So that's how we manage the sequencing and assembly subcommittee. Yeah, thank you very much.
Thank you very much. And we're happy to see that the training initiatives that have been done in Jomo Kenyatta are contributing back to Africa. So Professor Andala, we are ready for you now. We are going to load the presentation from our end, and you just signal when you want us to move the slides. Thank you very much. Professor, are you able to unmute yourself?
Yeah, now it has worked. Probably I'll start off by recognizing the eminent researchers in the Africa BioGenome Project present today, the university leadership also present, and the organizers for inviting me. I'm delighted to join you in this very important research dissemination workshop, of which I'm passionate about. I'll do my presentation. I'll try to recover the lost minutes, and I hope everybody can be able to hear me from the other end. Can you go back to the first slide? Yeah, as mentioned earlier, my name is Professor Dickson Andala, and I'll be talking about unlocking genomic research potential, the funding environment and strategic partnerships for sustainable growth. Next slide, please. I'll give you an overview about National Research Fund. We were established in 2013 under the Science, Technology and Innovation Act, and we became operational in 2015. Our mandate and mission is to facilitate research funding in Kenya by promoting scientific innovation and development. So we've been supporting several research projects across the country and partnering with our international partners to ensure that we build capacity in research. Next slide, please. Most of our functions are 13 in number, but the main ones which I'll speak to include mobilization of resources from government and private sector. We also manage the fund so that it can be able to grow in size. Similarly, we support development of human resources in terms of grants to individuals and institutions. We also allocate research funds for multidisciplinary research among universities and research institutions. And we also support the development of research capacities within the areas of national priority. Next slide, please. We fund research across several thematic areas, and I'm happy under the Africa BioGenome Project, we fund agricultural and natural sciences, which speaks to your workshop today. Others are biological and health sciences, physical and industrial energy sciences, infrastructure and information sciences, humanities and social sciences, and finally, earth and space sciences. Next slide, please. We provide several types of grants. The first one is multidisciplinary research grants. These are aimed at supporting collaborative research. Secondly, we also support multilateral and bilateral research grants, and these are actually these are of strategic international partnerships, which brings together researchers from different countries. For instance, we've been able to support researchers in South Africa and Kenya. Similarly, under the Newton Utafiti Fund, we supported researchers in Kenya and those in the UK. We've got strategic grants, and this one addresses national priority research interventions. A perfect example is COVID research grant and the one on locusts. Next slide, please. We also support participation in scientific events. These include workshops, seminars, and conferences. We've got also innovation grants. This supports innovations, and these are channeled through Kenya. Kenya is our sister agency, which is a Kenya National Innovation Agency. We've got another sister agency called NACOSTI. We work closely. We've got incubation grants, and these ones are geared towards supporting commercialization of research outputs and also funded through Kenya. Next slide, please. As NRF, we've been involved in strengthening research capacity in Kenya, and key among what we do is we've established a national research repository, and in this national research repository is an open access resource whereby researchers are encouraged to upload their research and share the research with their colleagues. And this national research repository was facilitated through the British Council and Foreign Commonwealth Development Organization. We've got several research output deposited within the repository. Currently, we are also planning to have a national research festival, and in this national research festival, we are bringing together researchers, research institutions, and funders. What we are going to achieve in this case here, we want to build a resilient ecosystem which brings together scientists, more specifically in all the thematic areas I've already pointed out. The event is happening between the 19th to the 23rd of August, and I extend a warm welcome to all those who are participating in this Africa BioGenome workshop. Similarly, we shall be able to recognize national outstanding researchers through the National Research Festival. Through our activities, we've been recognized, next slide please, through what we call National Diversity and Inclusion Awards for the good work National Research Fund is doing. This was received in April this year. Next slide please. Now this brings me to the topic of today, which is NRF support for the genomic research. As already mentioned, NRF recognizes the importance of genomic research in addressing health challenge, food security and health challenges in Kenya. For instance, the problem of loss of flora and fauna can be addressed through this biogenome project. A key reminder is in a recent report launched, we noted that about 123 species are critically endangered. And through this biogenome research project, we can be able to recover the loss and repopulate. The other thing that we need to also mention here is the integration of bioeconomy into the GDP. Through the biogenome project, we should be able to essentially bring resources through planting of those species that are actually of economic importance. And as such, NRF is cognizant of the Africa BioGenome Project, which was launched in 2021 by several African scientists and African institutions, whose mandate was a pan-African initiative geared towards building capacity and infrastructure to generate, analyze, deploy genomics data for improvement and sustainable use of biodiversity and agriculture across Africa. As such, NRF has funded several projects, and we can be able to see in the following slide. And most of those projects funded are in line with the bottom-up economic transformation agenda, which is the government's economic blueprint. You can be able to see here we funded about 8 institutions to the tune of 183 million Kenyan shillings, and all these are geared towards genomic research. We've got one in University of Eldoret, where they established a lab in fish genetics and genomics research. We have also at Cambridge, where we also have a center of excellence in stem cell research. We've got Bukura Agricultural College, where we've got a biotech lab for training and research in rapid multiplication of planting materials. So as NRF, we are fully supporting genomic research. We have calls which are already also available online, for which you can be able to visit for those Kenyan researchers in the house. We've got bilateral programs with the UK, as well as South Africa NRF, to be able to support research in biodiversity. Next slide, please. So NRF has several partnerships and collaborations, all geared towards international support and, of course, local research funding. And we've these initiatives, which I've listed here, where I'll encourage our researchers to visit. One is the Darwin's Initiative. They support research in biodiversity. We've got Cambridge Africa Alborada Research Fund. We've got Bill and Melinda Gates Foundation, and this has done a lot in funding of biogenome research, as well as biotechnology and bioinformatics. We've got the Newton Utafiti Fund. This one has been renamed into International Science Partnership Funds. We have an active call that we are encouraging researchers to apply. We also have Science Granting Council's initiative. This brings together research in 17 countries in Africa, where researchers collaborate through the granting councils. Kenya is a member of the SGCI, as well as all the participants have already seen on this platform. We've got Horizon Europe, which has got also active calls that are supporting research in biogenome. Next slide, please. So there are other several opportunities that exist in biogenome research. One is professional opportunities, as well as granting opportunities. Having reviewed literature, I noted that the National Institute of Health in the USA also funds research in biogenome research. European Commission, Medical Research Council in the UK, we've got the Genetic Society, Open Bioinformatics Foundation, as well as Cancer Research Institute. The other foundations supporting genomics and bioinformatics research include the Encephalopathy, which is the SYNGAP Foundation, National Ataxia Foundation in the US, Cystic Fibrosis Foundation, as well as the FRAXA Research Foundation. Next slide, please. While in Kenya, I'll encourage you to form collaboration with these institutions, and these institutions are leading in matters biodiversity, bioeconomy, and biodiversity research. We've got the International Livestock Research Institute, ILRI, which is leading in bioeconomy and biodiversity research in Kenya. We've got the University of Nairobi under the Institute of Tropical and Infectious Diseases. We've got Center for Biotechnology and Bioinformatics. We've got Kenya Agricultural Livestock Research Organization, CALRO. We have got the University of Eldoret, Kenyatta University, and more importantly, the Egerton University, where the International Center for Genetic Engineering and Biotechnology is being established in Kenya. So I'll encourage you to collaborate with researchers in Egerton, as well as these other institutions. Similarly, we've got KEFRI, which is Kenya Forest Research Institute, and the Kenya Marine and Fisheries Research Institute. Next slide, please. On the international front, Kenya, we've got the Consultative Group for International Agricultural Research, the CGIR, and this brings together a consortium of 15 research centers, all focused on bioeconomy and biodiversity research in food crops, as well as World Fish Center in fish. We've got the African Center of Excellence for Genomics and Infectious Diseases. This is also based in Nigeria. We've got the Wellcome Sanger Institute in the United Kingdom, among others. So I'll be able to encourage our scientists who are working on biogenome to reach out to these centers for collaboration and partnership purposes. Next slide. In conclusion, the integration of bioeconomy in biodiversity conservation can incentivize sustainable development since it leverages biological resources, processes, and principles to produce goods and services in a sustainable manner and improving livelihoods. The critical role of research and innovation in driving economic growth, social progress, and sustainable development cannot be overemphasized, more so in assisting the world to meet the SDGs, the United Nations Sustainable Development Goals. With that, I want to thank you once again for inviting me to speak to you, and more specifically on matters to do African biogenome research. I look forward to collaborating with several research institutions that are represented here today, as well as you individual researchers, to map out ways on how we can be able to build capacity and attract more resources to support the African BioGenome Project and research on bioeconomy and biodiversity. I thank you all, and may you enjoy the rest of your day. In case there are any questions, let me know. I'll be more than willing to answer.
So, Professor, I hope you have had that resounding applause that you have been given from the site in Nairobi. Indeed, we are very, very excited to see the funding ecosystem, because the next question is always who is funding you and where are you going to get the money? And with the genomics, as we know, being not very, very cheap, it's not easy to get the monies that you need. And we're very, very excited to see that not only will we be able to get funding to attend conferences, to do projects, but also through your institution, we will be able to form even stronger partnerships. And I look forward to us in the room here forging those partnerships with your assistance and with your collaborations. I would like to invite questions from the floor, and you will also maybe check for us whether there are any questions online, and we can be able to have them. Any question? Do we have a question from the online audience? No question even about travel plans. So the African BioGenome Project, through its fellowships, sponsors on-site hands-on training in one of our 4 regional nodes. So, Professor, our nodes are based in North Africa, where we have a collaborator in Mohammed VI University. We also have collaborators in University of South Africa, University of Port Harcourt in Nigeria, and Jomo Kenyatta in Kenya, which covers East and Central. So the question that I would have for you, Professor, is that African BioGenome, through our partners, is able to cater for the on-site expenses for many of our fellowships, but we don't have money to fly the participant to the particular location. For instance, the one location we had to fly students to was to North Africa in Morocco. Would we be able to, or would even the fellow, after they have won the grant, would they be able to apply for support if they are Kenyan to be able to travel, or if they're East African for that matter? Professor, check if you're muted, please.
Thank you for the question. Yes, we actually support travel. Can you be able to hear me?
Yes, you can.
Right. Okay, thank you. We support individually those who want to participate in conferences, and we also support conference organizers. For instance, if this conference is under JQuat or PAUSTI, PAUSTI can apply to NRF, then we can be able to see what aspects within the budget we can be able to support. We encourage, I know this is international. However, for those who are Kenyans, the support is always there. However, in the case of the international, we can always reach out to our bilateral partners to be able to support the regional, like the SGCI. If you're from the SGCI country, we can have a grant that can be able to cater for those who are outside Africa, outside Kenya. And then I've seen a question on sharing the slides. So probably those who are interested can get in touch with the organizers. They have a copy of the slides. And then there's a question online on how do you apply for the grants or the funds. In this case here, the funds that are open, you can visit our website, www.nrf.go.ke. We've got several multilateral calls, which involve several countries, and those ones which are multi-institutional. And then there was a question from EBSA who wants to ask a question. I have not seen the question. Then there is a question from Tobias Innocent. Maybe they can ask.
Prof, there's a question here about grants. Is it possible also to get grants to organize such conferences as this one, or is just specific on projects? Thank you.
Yes, we support both dissemination and research itself. So what you're doing is dissemination. So there is grants for dissemination purposes, and there's also grants to participate in scientific events.
Thank you.
And perhaps just a final question on an innovation. How are we able to discern what is an innovation? For instance, are projects innovative ideas? Are those applicable, or it has to be like some gadget or some technology implementation device that is eligible for the technology support? Thank you.
Yeah, that's a good question. We have what we call end-to-end support in terms of when you're doing your research, you may end up with an output which we may term as innovation. We flow that to, or that is picked up by Kenya. Kenya analyzes or does a review on the same. Then they liaise with KIPI. KIPI is Kenya Intellectual Property Rights Institute, from whereby now they can now determine how much you are eligible. So on innovation, which is research to commercialization, you have to rope in the innovation agency so that they can be able to determine if it's an innovation or not. And then we can be able to fund you through Kenya.
Professor Dickson Andala, as the CEO of NRF, we recognize how busy your office is. We really appreciate being our guest and having given this keynote address to us to be able to enlighten us on the funding ecosystem within Kenya. I think the impression is that the funding ecosystem is dry, but what you have been able to show us that it is actually very rich, using the government resources, but also resources and partnerships that you have been able to bring forward have enabled this ecosystem to be better than what it would have been. And we thank you very much for gracing our function, and we would like to give you a resounding applause for your participation. Thank you very much.
Thank you, and I look forward to future engagements with the biogenome research. You will all have my support in the near future if you reach out. And then finally, there is a lot of funding through Bill and Melinda Gates, which is not being absorbed by the African countries. So if we can reach out through APHRC and NRF, we can see how we can be able to assist. Bye for now, and enjoy the rest of your days of training.
Thank you. Thank you, Professor. So with us online, we have the next presentation from Dr. Helen Nigusi. Professor Dr. Helen Nigusi is an assistant professor in genetics and bioinformatics in the Department of Microbial, Cellular, and Molecular Biology in the Addis Ababa University, AAU, where she is the chair of department. She also serves as a national node representative of the African BioGenome Project. She's the one who's the node representative for the African BioGenome Project in Ethiopia. She has over 20 years as an active researcher and lecturer, both in undergraduate and postgraduate levels at AMBO and AAU, and has contributed to the mentorship of over 24 MSc and PhD students, 13 of who have successfully completed their studies and are now continuing on with their work. She's very passionate about curriculum review, thesis examination in various university students, and has over 200 graduate students who have passed through her hands. She is currently chairing the Department of Microbial, Cellular, and Molecular Biology, and is a very, very active member of the Society of Ethiopian Women in Science and Technology, and is one of the leaders of the Ethiopian Society of Animal Production. Professor Nigusi is also a very active member of the African BioGenome Project, has been a very, very supportive collaborator with ourselves, having participated in running practical workshops last year in 2023 and also this year. So we welcome Professor to give us a talk on capacity building in genomics and bioinformatics for research and sustainable development. Professor Helen, if you're online, please go ahead.
Okay. Thank you, Professor Anne, for your generous introduction. And I would like to welcome you all, both at Nairobi and online, to my presentation. I'm going to present entitled Capacity Building Genomics Bioinformatics for Research and Sustainable Development, with special focus in Ethiopian case. This next slide. Next slide, please. I'm going to talk on a brief introduction, the application and the capacity building going on in Ethiopia, and the challenges and the way forward. Please, next. As introduction, as we all know, we have 17 Sustainable Development Goals and the UN agenda, which will be achieved in the 2023. Among these, the 17th Sustainable Development Goal, Development Goal 2 and 3, Zero Hunger and Health and Well-being. Next. This agenda is entirely focused on the food security and good health and well-being, the good health and well-being for all nations across all age. And we have also another agenda, Agenda 2063, the Africa We Want. This has 7 aspirations. Aspiration 1 also focus on, again, health and well-nourished citizen. Modern agriculture for increased productivity and production. This is also another focusing on health and food security. But to achieve this sustainable goal in 2030, again in Agenda 63, we have a challenge. The first challenge is a population growth. Population growth is increasing globally, but specifically in Africa, it will be doubled in the next decade or 20 years. So we have to feed our nation in all form. Another challenge is climate change. The climate change also another challenge, not to produce the expected level of food due to different challenges both in livestock and crop, and nowadays everybody of us is facing. So to achieve the sustainable and the sustainable goal and the food security, we have to use the technology. One of the technologies, genomics and bioinformatics, is a very important tools to improve the productivity of the livestock and crop, and also mitigate the health issue. So this bioinformatics and genomics is a very important aspect and one of the tools we need to implement. Please, next slide. So this genomics and bioinformatics is a very important tools, and both have a significant application in every disciplines. It is a multidisciplinary approach and will contribute in research and sustainable development. Please, next slide. Next slide. For example, the genomics and bioinformatics research can contribute a lot in agriculture. For example, in crop, it will contribute in different aspects. For example, to mitigate climate change, it can apply in identifying those of resistant crop variety. It also contributes in genome editing, which creates a very resistant and productive crop. It also contributes in livestock as well. Next slide, please. This genomics research and bioinformatics also play a significant role in livestock research in improving the productivity of different livestock species, identifying a resistant breed, and to create a climate smart livestock breed, and even in the genome editing and maintaining the health of the animal in vaccine development and identify different fatal diseases. Please, next. Genomics and bioinformatics also has a significant role in genomics research to understand the microbes in our environment. What are they doing? Where are they? And to integrate in different public health and to mitigate, to put a strategy for mitigation in climate action. It also contribute in microbiome research. We are in One Health era. We need to understand and do different research from animal, the environment, and human health aspect. So in this regard, both genomics and bioinformatics has a significant role for all the disciplines. It also contribute for this next and biodiversity conservation. It contributes a lot, and one of the agenda of African BioGenome Project, we have so many endemic diversity in Africa and Ethiopia. So this genomics and bioinformatics will help on that aspect. It's also applied in different clinical application in diagnosis and the treatment. It has multidisciplinary approach. It also contributes for forensic application. So bioinformatics and genomics has contribute a lot. So the capacity building in this discipline is really important compared to the human resource we have at Africa level and the developed world. Is there is a big gap, so we need to increase the human resource and the capacity building at Africa level, of course at Ethiopia national level as well. Please, next. It also role in the molecular medicine. Nowadays, we are in the personalized medicine era. So this disciplines has play a significant role in early detection of the genetic disposition, improving diagnosis in pharmacogenomics, which is very important in gene therapy and control system for drug discovery. So it has lots of contribution in different, this is a few, but it has different contribution. So when it's a capacity building is really important, as mentioned earlier. So we have started a capacity building effort at Ethiopia level since late 2018. Please next. So the capacity building efforts in Ethiopia have been supported or in the collaborative with African BioGenome Project, Addis Ababa University, Bio and Emerging Technology, and H3 Bionet. The first training workshop takes place in 2019 in the collaboration of Beka Il Rehab, and it's a part of a fellowship community, bioinformatics community of practice. It was the 2 years fellowship, and part of it is capacity building. So this was the first initiative and training workshop. It also extended, and one of the Ethiopian university, Walkite University, in the same year in the collaboration with Bioemerging Technology Institute. It's also another workshop in the collaboration with ICGB, International Center for Genetic Engineering and Biotechnet, which is also again the collaboration with Beten is leading the capacity building in Ethiopia in the collaboration with different partners. We also host the first East and Central African BioGenome Project last year. We had a very interesting practical session and gave opportunity for the national PhD students and researchers involved across different universities and research institutes. We also, next please. We also host the short-term virtual training, which is 3 months in the collaboration with H3 Bionet based in South Africa. We host the last year, 2023, even the current this year, which is recently completed. And most of the researcher and graduate students from different universities and institutes involved in the research. We also again host in the late March another workshop in the genomic surveillance in the collaboration with Betin and ICGB. This is also the Biotechnet initiative for East Africa. We also, now we will going to host this year African BioGenome Project. This is some of the publication which has been done together within African BioGenome Project since 2019. Next, please. So there are also some initiatives as a graduate program level in few of our universities. Those university, Addis Ababa University, Gondar University, and Jinma University has MSc program, which is also a very interesting initiative because there was no any program before. So we have different effort to bring this genomics and bioinformatics on board. However, we have a challenge, which is not only specifically for Ethiopia, it's common in Africa. We have some limited access for computational facility, for example, HPC, because most of our graduate students and research fellow have an opportunity and generate the data in the collaboration outside, but they don't have such access to use their data and do further analysis. In some institutes, there is no strong internet power to support such facility. We have also budget for capacity building. For example, in every call, there are a number of applicants, but we couldn't afford to provide this hands-on training for the applicants. Sometimes it is more than 2,000 for one call. So this is a challenge we faced so far, but we are trying to strategize, collaborating the existing institute, and to bring hands together to improve such capacity building and to allocate in every national university just to use this open institute approach. So we will continue to collaborate with African BioGenome Project, and I would like to acknowledge those institutes to give this opportunity and to contribute for the young scientists, African BioGenome Project and its partner, Addis Ababa University and Bio and Emerging Technology Institute, because this institute is always gives their helping hands to host this practical session for the Ethiopian side. So thank you so much, and you are welcome if you have some comments and suggestions. Thank you so much. Asante, እናመሰግናለን ገለቶማ, and those all of you were this in Ethiopia language. Thank you so much.
Thank you very much. Yeah, thank you very much. I think we're the ones who are very grateful to you, Dr. Helen, your institution, and also Betty, because of the not only providing the human resource to be able to do the training in Addis Ababa, but also to provide the reagents and the other resources. So we're very, very grateful. It's important to also note that the challenges you have listed there are not unique to Ethiopia. We also have faced them not only in Nairobi, but also parts of Africa. But what is encouraging to see is the solutions that you have also developed, because it's one thing to say, I have this problem, give me money. But I have noted very much that you are addressing these challenges by factoring in budgets for capacity building, by leveraging on digital mechanisms for broadcasting your research. So right now, I'll open the floor to questions. Anyone who would like to ask a question on site for Dr. Helen can go ahead. Yes, there's a question over there. And maybe you can also check for me whether there are questions online. Please give us your name and the institution you have come from and the country.
Okay, good morning. My name is Victor Omondi Njolo. I'm from University of Chinese Academy of China, University of Chinese Academy of Sciences.
Do you mind standing up so we can see you better? Thank you.
All right. My name is Victor Omondi Onjolo from University of Chinese Academy of Science, that is in China. And a question to Professor Ellen. There is a new issue about environmental, social, and governance issue. I would really wish to know, like, have they factored in issues regarding participation in terms of gender, in terms of age, for their capacity building reporting to enable the environmental, social, and governance reporting? Thank you so much.
So, Dr. Helen, the question is, go ahead.
Okay, thank you for the question. Sorry, I didn't present the participant and how to consider in the capacity building. In the selection criteria, we try to accommodate all those standard criteria. For example, most of the time we will try to balance gender balance. We tried to make it 50-50, but sometimes we couldn't match, but 50-50 or 45-55. We try to maintain the gender and the age and the relevance as well. Who is currently going on this genomics and bioinformatics, and who is really use this practical session for the current research. So we try to consider all eligible applicants from public university research institute across all age, as long as his research is relevant for this practical session, we'll consider. But most of the time we should prioritize because of the fund. So the more prominent and appropriate participant will take the lead. But there is no limit for application and participation. Thank you.
I think if I can add on to that, when it comes to capacity building, more and more, especially with emerging women scientists, the more women rise up the ranks and become, professors and principal investigators, the more hope and visibility you give to the younger. And let me just say that when I was an undergraduate student, the ladies were among the minority. But now as we go to class, we have almost not yet 50-50, but we're inching towards 50-50. And when I look around the room, if we were doing a training like this when I was in an undergraduate, I'd have been one of 3 ladies. But as we move along, look at the numbers in the room, the numbers are quite many. So I think we're getting there, and with inclusivity, we should do that. So there's a question for Dr. Helen. What is your future plan to give such type of training, I think, to more Africans? And there's a question that I will answer. Is it possible to become a member of the AfricaBP project? So Dr. Helen, they're asking, okay, what is your future plan with regard to giving capacity building around Africa?
Okay, yeah, just our future plan to keep moving forward as long as the demand of the training, the genomics and bioinformatics is continue, but hosting one session career may not enough compared to the demand we have. So we have a plan to have different sites across different universities, because now we have more African BioGenome Project, and we have a resource compared to the last 3, 4, 5 years. So we have a train of trainers. So we'll use them and duplicate such effort across different universities, as long as there are a volunteer and to host say type of workshop in their local university or research institute. So we are advocating to host this workshop, and we'll provide human resource, and we are volunteer to help them to hold say type of training as their institute. And we are also starting to move out of Addis and host in the local universities in different regions, as long as they are interested to provide some support to give hands-on training at their center. Thank you.
Thank you. Thank you very much. And let me just add that as young early career scientists who are applying for opportunities, bring something to the table. If I'm offering you training in Morocco or training in Ethiopia or training in, Nairobi, and maybe the training is in Pwani, be ready to just put something of your own, because you see, maybe I'm paying for you the flight, I'm paying for you I brought the trainer there, I have done the course selection. There's a lot of resource. So what we find in AfricaBP is that sometimes students or early career scientists will turn down opportunities because they say, why are you not paying flight for me? You should actually, it should be the other way around. You should tell me, I have paid my flight, I have money for this, I have money for this, now give me this opportunity. So it's a partnership. And I think for capacity building, don't be too poor. Be ready to, to give something of your own. Dr. Helen, comment on this, please.
Yeah, you are right. We should coordinate these efforts. If me, for example, I am always ready to contribute my profession, to give training, everything and the volunteer base. In some for example, in Betin, they always contribute and supporting those, sponsoring those students to provide some important, to cover some important cost. Addis Ababa University will contribute human resource. So jointly we can make something. So the same is true for others. Everybody should contribute what they have at hand and what he can. The same is for the institute or a individual participant. And the early career scientist also sometimes willing to contribute something from their own side. For example, in our practical workshop, there are some participants, they cover all the cost, travel cost, accommodation, and to take the training and come into the center in Addis. So there are say type of interest and contributions. The only thing is we need to coordinate such efforts and create a platform like African BP doing.
So, and I think we have the people from Congo here.
Yes, I can see the people from DR's Congo joining us live. As you can see, it's a truly East Africa, Central Africa workshop. And what is very, very unique is that because they speak French, they have gone ahead and gotten a translator. So that's how eager they are to not lose any out on this workshop. And we really, really applaud you all the way from DR Congo.
Yeah,
I think it's University of Kinshasa.
You will ask in French and I will translate in English. Okay.
Excellent. Thank you so much, Dr. Mafiwa. Dr. Thank God, is there a question on board?
Yeah, they have a question. Dr. Antonio will be translating.
Please go ahead, University of Kinshasa, Dr. Antoine.
Okay, come here. Yes. So he says, but sometimes tu peux me la poser la question. Go ahead.
La question est celle-ci. Je disais, c'est une bonne idée d'avoir pensé à nous, Université de Kinshasa, précisément le département des sciences de base dans le service de biologie moléculaire. Nous savons très bien que la bioinformatique est très vaste. Il y a plusieurs domaines. Est-ce que cette initiative, qui est aussi loyale, ne peut pas être un peu élargie au niveau de l'État, au niveau institutionnel, pour que ça profite au plus grand nombre des Africains ou au plus grand nombre d'autres institutions?
Okay, thank you. I will translate. The question is saying thank you for your idea to involve the University of Kinshasa in this workshop. That is a very important workshop in bioinformatics. So, as the bioinformatics is too large and with many applications, it's not possible to go beyond private initiative to involve different government for enlarging this capacity building in order to cover more informatic tools and topics and to cover more different programs. Thank you.
Dr. Helen?
Okay.
The question is bioinformatics is very broad. How do we, are we able to do everything?
Yeah, this is also a very interesting question. Yeah, I presented in the general way, but when we organized in the past 3 years, we focused in 2 major groups. Those of the agricultural bioinformatics part, most of the participants is focused on agricultural, either livestock or crop. And the second one is focused on health aspect with other partners. For example, this ICGB Biotechnet, we hold the bioinformatics for genomic surveillance. The three workshop from their side, their partnership, and together with Armand Hanser Research Institute, will focus on the health, for the health researcher, and the other one is for the agricultural bioinformatics. So we used say type of platform, but in the future we will try to customize according to the demand and the need and the partnership we have. But for the time being, we focused on health and agriculture. And in the African BioGenome Project, most of the time the 2 years is focused in the NGS next generation sequencing data. This type of training will accommodate both the health, those participant has health background or agricultural, either crop or livestock background. But we try to customize according to the partnership and the demand we have, not only focused in the general one, and we will also try to customize it in the near future. Thank you so much.
I think we are very, very grateful. The journey is long. You cannot learn the subject within one day.
Mm-hmm.
And it's very important to tackle it in modular form. So generate data in your own data to analyze it, and you'll be able to add on your skills as you go ahead. So thank you very much, Dr. Helen. Let's give her a round of applause.
Thank you so much. You are welcome.
We value your partnership and thank you.
Yeah, me too. Thank you. Thanks so much.
So as I mentioned earlier, go ahead.
Thank you so much for the opportunity, and we'll continue our partnership. Thank you so much.
Indeed, we value our partnership with you, and without you coordinating Ethiopia, we would be lost because it's a vast country, and with you there, we are able to collaborate in a fast and seamless manner. So I had mentioned earlier that the African BioGenome Project is bent on encouraging and recognizing the input of early career scientists, because the early career scientists are the workhorse of data generation. You're the ones in the lab, and you matter specialists. And therefore, the 9 early career scientists making presentations between today and tomorrow. Today, we will have the first group led by Eunice Mulango. Eunice Mulango is a PhD student here at Jomo Kenyatta University of Agriculture and Technology. She holds a master's in biotechnology from Jomo Kenyatta University of Agriculture and Technology. She will be giving us a talk on exploring insects for biopharmaceuticals and harnessing their potential for medicine. As you can tell from her talk, her research interest focuses on comparative genomic analysis of microbial influences on mortality rates of crickets and how this can be harnessed for better utilization. Eunice, you have the floor. Eunice, come forward. Thank you. Let's give her a round of applause as she comes.
Thank you for the opportunity. I don't know if it's morning or afternoon, but good afternoon all. My name is Eunice Mulango, as stated, and a PhD student here, but I also work for the Kenya Institute of Public Policy and Research Analysis. And so today I'm doing a policy paper because I do policy. So my policy is to propose on exploring insects for biopharmaceuticals and harnessing their potential for medicine. Yeah, next, please. Yeah, so that is my outline for my work. I have the introduction, pharmaceutical landscape, my motivation, policy gaps, the objectives, theoretical framework, empirical studies, methodology, results, and conclusion. Next, please. So, healthcare accessibility and affordability has become a pressing global concern, and it affects millions worldwide. And the conventional pharmaceutical production involves high cost of production, and healthcare has become also costly. And there is another challenge about the development of the antimicrobial diseases, antimicrobial resistance diseases, whereby a lot of diseases are becoming resistant to the normal medicines. So because of that, an alternative needs to be looked for. And as a result, many individuals are in need of life-saving medications and find themselves unable to afford them. Next, please. So in my study, I propose that we can use insects to produce biopharmaceuticals. And there are different kinds of insects like termites, black soldier flies, grasshoppers, and beetles that can bring a beacon of hope in our region. And this is because they are available in our biodiversity. They are sustainable and accessible, and they can be used efficiently in our healthcare. And so by harnessing their unique capabilities, insects have the potential to be used in biopharmaceutical production. Next, please. So in my study, I also needed to bring out the understanding of the pharmaceutical landscape globally and also regionally. And globally, the global demand for pharmaceuticals has been driven by various factors such as advancement in medical science and technology. And the pharmaceutical market has reached $1.27 trillion. And if we project up to 2028, it will go up to $1.5 trillion. And also that is the normal pharmaceuticals. For biopharmaceuticals, right now they stand at $381.4 billion. And it is expected also to go high up to $26.2 billion by 2028. And that is a growth rate of 6.6%. So these markets are fueled by the prevalence of chronic diseases and also the advancements and increased investments. Next, please. So there is the regional perspectives that is in Africa and also in our country, Kenya. And we have the high disease burden of malaria, HIV, TB, and other diseases. And when we have this high burden, we have our country relies heavily on imported pharmaceuticals, and 99% of our medicines are imported. And one promising avenue is the exploration of alternative sources of insect-based pharmaceuticals. And these can be used because they are cost-effective and sustainable. And in our country, we can have initiatives that can leverage the use of biodiversity to produce medicines. So moving forward, robust research initiatives are needed, and policies are needed to be formulated to be able to handle this challenge that we have as a country. Next, please. So the just as I have stated, that table is showing those global outlook and the regional perspectives of the pharmaceutical industry. Next. So the motivation of my study is stems behind the urgent need to address the persistent healthcare challenges facing the society and the emergence of infectious diseases that are on the rise, and there are limited avenues for effective treatments, and the traditional methods of producing medicine are not cost effective. That is, we incur high cost to produce those pharmaceuticals through the normal methods. And there is also the issue of environmental sustainability, and we also need to have other innovative solutions to the issue. Next, please. So insects offer a promising avenue for biopharmaceutical research due to their rich biodiversity, their intricate biochemical properties or simple biochemical properties, and they have novel therapeutic compounds, and exploring the medicinal value of insects can present an opportunity to diversify the pharmaceutical pipeline and overcome the existing therapeutic limitations and develop sustainable treatment options. Next, please. So with this study, there are some policy gaps regionally and globally and nationally. Globally and regionally, one of the foremost policy gap is the absence of unharmonized regulatory standards governing the use of insects as biopharmaceuticals. And nationally, as a country, we also have the absence of policies and regulatory frameworks that prevent the hurdles to get investment and innovation in the insect-based pharmaceutical production. Next, please. So the objectives of this study were to assess the future of insect-based biopharmaceutical production in Kenya and to give policy recommendations on the use of insects for biopharmaceutical production in Kenya. So we have the theoretical framework where we have the previous theories that are talking about use of insects as biopharmaceuticals. We have the ecological and evolutionary theory. We have the bioprocess engineering theory and the immune response mechanism theory. Next, please. Then we have empirical studies, that is studies that have been done before. Worldwide, insects have been found that they have the potential to produce insulin. Insects have been used to treat wounds. We have the green butterfly that is used to treat wounds, and also black soldier fly is used to treat wounds. Then we have antibody. Insects have been found to produce antibodies that treat diseases and also different infectious diseases and also HIV. Next, please. And also insects have been found to have the potential to produce, they can be used to produce vaccines that can be used to also treat various diseases, such as dengue virus, HIV, and hepatitis B. Yeah, so those are some of the examples of the insects that have been explored. We have 12 insect orders that have been explored for drug discovery and have been highlighted in red. That slide. Next, please. So, due to time, let me go. That was my methodology, the futures foresight methodology, where we have the problem framing and the study that involved the SWOT, SWOT is strength, weaknesses, opportunities, and threats. Then we have the environmental examination. Then we have the pestle analysis that involves the political, economic, social, technological, and legal environment. Then we have the trend analysis and the scenario evaluation. Next, please. So we have talked about the SWOT. Next, please. We are out of time. So those are the strengths. The insects are from the methodology. The results say that the strength is that the insects have a rich biodiversity and their biochemical complexity is simple, so they can be used to easily. They can be used to the research can be done on them for drug discovery, and they are cost effective, and also the use of insects for pharmaceutical is sustainable, can be sustainable. Next, please. Then weaknesses is some of the insects can have a complex, some of them can have complex biological complexity. Some of the insects can have challenges with production. For example, if there are mortalities and diseases, insect diseases, and then market acceptance, there is the public perceptions where people might not accept, and also the regulatory hurdles, like getting the permits and all that. Next, please. And so for the opportunities for this study, we can have novel discoveries, the growing demand, and collaborative research between different institutions. Next, please. For threats, there are the competition from the traditional pharmaceutical companies, the regulatory uncertainties, environmental concerns, and also the public perceptions. Next, please. For the environmental examination, we have the different drivers of change, and drivers of change can be influenced by the economic, that is the cost effectiveness of producing the biopharmaceuticals, technological advancement, that is advances in the genetic engineering and biotechnology. We have also the social, the shifting of public attitudes towards sustainable and novel sources of medicines, and environmental, the sustainability of insects as source of pharmaceuticals. Okay, next. Conclusion. So from my study, I can conclude that insects represent a promising frontier in biopharmaceutical production, and through rigorous research and experimentation, their full potential as biopharmaceuticals can be unlocked. And by addressing these challenges and enhancing their advantages, this innovative approach holds a promise for our nation. Next. Recommendations. Yeah, so it is crucial to intensify the efforts to identify the insect species that can be used for biopharmaceutical production, and also foster collaborative frameworks from different institutions such as industries and academia, and also stakeholders, to ensure the development and approval of insect-based pharmaceuticals is achieved. And significant investment is also needed for this approach. Thank you.
Thank you very much. We will have a question and answer session at the end. So just take your seat. And so we will have the next presenter. And the next presenter has a very unique name. That is Manikos Rudolfa Benzbang Kotango, who is a biologist, holds a master's degree in molecular biology and biotechnology. He has a very, very strong interest in biochemistry, microbiology, and biotechnology. He's committed to learn more about the intricate workings of life at the molecular level. And he is a PhD student who is going to be presenting to us a talk on protein concentration, anti-helminth activity, and microbial contamination of the laboratory producing encapsulated batches. So Manukus, the floor is yours. Let's give him a hand of applause as he comes.
Thank you, Chair, for this wonderful presentation. I'm going to introduce you to the output of my research. Term quality assurance of laboratory produced chitosan encapsulated bromelain through its protein concentration, its enzymatic activity, and also the level of microbial contamination in the initial product and the final product. Next. Yes, in most of the sub-Saharan African nation, livestock can help to secure food and nutrition. But in most of those countries, this livestock production is facing some challenges. Among them, you have the livestock diseases, such as emetosis, coccidiosis, and mastitis. And those livestock diseases can cause a decline in growth, productivity, and mortality, which leading to enormous losses for farmers. So to treat those livestock diseases, farmers are relying on the use of chemotherapeutics, but those chemotherapeutics also are facing some challenges. Among them, you have the drug resistance. So there's a need to formulate a new eco-friendly drug to treat those diseases. And chitosan encapsulated bromelain has been shown in literature to have an important role in the control of this livestock disease through its in vitro and in vivo activity. Among them, we have enzymatic activity, anticoccidial activity, and antimicrobial activity also. And also due to its low toxicity in some preclinical study in goat, chitosan encapsulated bromelain can be a suitable drug to treat those livestock diseases. And so through this study, the production of nano-encapsulated bromelain will be should be optimized and to ensure its reproducibility and also the quality of the nano-encapsulated product at the end should be assured. Next, please. As the objective, I defined the main objective of this study to determine the quality of laboratory chitosan encapsulated bromelain and the in vitro level of microbial contamination in the initial product and the final product. Next, please. So the methodology is defined in 3 steps. The first one, which is based on the extraction and the nano-encapsulation of the bromelain, I purchased my sample, which are pineapple from Gatundu farm. Then I brought it in the laboratory. I treat it with 1% hydrogen peroxide. Then I extract the bromelain in those pineapple peels, following the previous procedure. Then I nano-formulate it using 1% chitosan matrix. The second one, which is to determine the enzymatic activity of the nano-encapsulated bromelain, I collected the worm from Ruiru slaughterhouse. Then on those worms, I did 3 tests. The first one is to do the egg hatching assay. The second one is to do the larval mortality assay. And then the third one is to do the adult worm assay on those from this nano-encapsulated bromelain. The third objective now is to determine the level of microbial contamination. So I use the initial product, which is the crude extract, and the final product, which is the nano-encapsulated bromelain. I did the total plate count. Then I isolated some bacteria from those crude extract and the final product. Then I ran the gram staining to determine the type of bacteria I will have. Then after that, I did some biochemical tests following by some molecular biology tests, and also I sent the sample for sequencing outside of Africa. Next, please. Yes, the first result, which is based on the concentration to see the physical-chemical variation among the batches of the nano-encapsulated bromelain I did, I found that the concentration in the crude extract was around 4 milligrams per ml, while the one for after dialysis to obtain the pure bromelain, I found that the concentration was 3 milligrams per ml. And coming now to the nano-encapsulated bromelain, I found the concentration was around 1 milligram per ml. And I observed that there is no significant difference among the batches coming from crude extract, dialysis, and nano-encapsulated bromelain. And just the procedure I use is more consistent, and the bromelain concentration is similar with the previous study done by some previous author. Next, please. I did now the test for the enzymatic activity. I found that I did in 2 ways. First of all, I used the commercial bromelain, and then my extract, the one I extracted in the lab, and I found that the commercial bromelain is more active on larval growth than the extracted bromelain. And the IC50 of the nano-encapsulated bromelain was 1 milligram per ml, which is able to limit the growth of those larval mortality, larval parasite, while the one for the commercial bromelain is 0.6 milligram, which is able to limit the growth of those larval of the parasite. And these results are similar to the previous study done by some previous author. And also this result shows that the nano-encapsulated bromelain, which has the IC50 of 1.3 milligram, is active against the larval of the parasite. Next, please. I did also on the adult worm, and I found that the IC50 of the extracted bromelain, the one I did in the lab, is around 0.245 milligram per ml, while the one for the commercial bromelain is around 0.9 milligram per ml. And there is no significant difference among the batches coming to all the 4 batches I did. And I found that those results are similar with some previous study also. And in that case, the nano-encapsulated bromelain is more active than the commercial bromelain. Next, please. And now I isolated some bacteria from different batches. So this table shows that from the batch 1, I isolated some type of bacteria. The batch 2 also, I found some bacteria. The batch 3, I found some bacteria. And the batch 4 also, I found some bacteria. So in total, I obtained around 17 strands of bacteria in the crude extract. While coming now to the final product, I wasn't able to find any contamination here. Next, please. And I ran some biochemical tests, and I found that most of my the bacteria I isolated were gram negative, and all of them are catalase positive, and other tests are shown that like the test of indole, I found that none of the bacteria are indole positive. Next, please. Now, after that, I extracted the DNA from those 17 bacteria. So from those 17 bacteria, and I ran the 16S, I amplified the 16S region. Now I found that all the bacteria are well amplified with the band of around 1600 kilobase per, and I sent it for the sequencing to macrogen. They ran also the PCR there, and we obtained the similar result. And then I used those chromatograms they sent me to plot the phylogenetic tree. At the end, I found that most of the bacteria are enteric bacteria, but just few are the common pathogen. Next, please. And to conclude, the concentration coming to the bromelain extraction, the concentration of the bromelain in crude are close to the previous study and did not vary with batches, showing that the lab production produced consistent result and can be used for commercial production if you want to commercialize the product. And I did also some FTIR analysis, but I didn't show the result here. And this FTIR result, I showed that the bromelain was well encapsulated in cathode, and there was no variation between the batches. And coming now to the enzymatic activity, the study confirms that the importance of plant extract, particularly non-encapsulated bromelain from pineapple peel, can exhibit the enzymatic activity. And secondly, the encapsulated bromelain was particularly effective against adult worm compared to eggs and larval mortality, which due to the presence of a cuticle composition in the adult worm, and the activity level was consistent across all the 4 batches. And coming now to the microbial contamination, I found that there's a high number of the viable bacteria contamination in the crude extract, but none in the final product. And those identified bacteria in crude extract include also common pathogens, suggesting that there is a contamination during the production and also the harvesting. The final product is safe to be used for animal treatment. Next, please. I thank, first of all, Jomo Kenyatta University for the infrastructure, and also the Pan-African University and the BioInnovate project for supporting the work. And also I thank my supervisor, which are Professor Naomi Maina and Dr. John Kagira. On this, I thank you.
Thank you very much, Manikus, for that presentation. Please, I ask you to note down your questions. Hold. We will have 2 more early career scientists presenting, and then we can ask questions as a group. And then now, presenting to us virtually, we have a freshly minted Dr. Masarat Elias Dureso, who graduated with his Doctor of Philosophy in Plant Breeding in July 2024. I think he's the newest doctor in the room, and we can give him a hand for graduating. So he was born in Western Ethiopia and has studied in Wolaita Sodo University, where he graduated with a Bachelor of Science in Crop Science. He then later he graduated with a Master of Science degree in plant breeding from Wolega University, after which he joined Oda Balutum University as a lecturer in the Department of Plant Science in 2015. And he served also as a program coordinator on research extension and publication. And in 2019, he joined Malaria University to do his postgraduate in the PhD in plant breeding, and I'm happy to note, and we are all happy to note, that he finished just last month and graduated. He's going to be presenting to us a talk on multi-locus genome-wide association study reveals genomic regions underlying root architecture traits in Ethiopian sorghum germplasm. Masarat, you're online, please. You have 12 minutes.
Thank you, Professor Anne, and the organizing committee. So I'm going to present on the title mentioned by Professor Anne. Next slide, please. The presentation ran from the introduction, and finally I will give the way forward. The next slide, please. Next slide, please. As you know, sorghum is a major crop, diploid, majorly produced in eastern parts of Ethiopia. It is very important as food crop for millions of people. Next slide, please. Drought is one of the major limiting factors in most of arid region on sorghum production. So next slide, please. So in order to overcome the problem due to abiotic factors, different studies have been conducted. Next slide, please. I have prepared another PowerPoint anyway, I will continue. So this study is conducted with the objective to identify the genomic region underlying the root system architecture, especially the root angle. So this study is conducted at Jimma University. The phenotypic root phenotyping was done in greenhouse in 2021. Next slide, please. This is a phenotypic experimenting procedure we followed. We did using root chamber, high throughput root phenotyping platform, composed of 10 hubs and 500 slots, or what we call it row column. We plant 3 seed at the middle, and finally tilled out to one particular plant, which we collected the data from. Next slide, please. This is the image. Can you go? Please go on. Please continue, Continue, please. So we collected data for root angle, root length, and root number and the dry weights for genome analysis. This is the image showing that we use this machine to take the root angle image. Next slide. This is while we are taking the exam. So the data for root phenotyping is done using a real model, R package. Can you go? Next slide. So here is the GWAS content. So the SNP data we collect, we used the SNP data done by the Wisconsin University, which is situated in Purdue University Respiratory. So finally, after getting the SNP data from Purdue University, we filtered with MAF less than 0.05, missing value, greater than 25. And finally, we did about 265,944 SNP data for genome-wide analysis. Next slide, please. We used, come back, please. One page. The first, We use this model, ML-GWAS model, which is recently developed by Chinese scientists. So it has 6 models. Next slide. So, okay, anyway. Co-localization, we use QTL Atlas database. To identify the candidate gene, we use Phytosome website on Biomart tools. For more detailed explanation of the gene, we use sorghum-based data online. Can you go on? Next slide. Yes. This is a analysis of variancy for phenotypic data. There exists significant difference between the root architecture studied. Can you go on? So Ethiopia sorghum germ plants harbors great diversity for root system architecture. So the same thing is similar for underground root system architecture. There is a significant difference, which is very important for studying for drought or in moisture deficit environments. Next slide. This is the frequent the frequency distribution of RSA trait studied for GWAS analysis. Can you go? Here it is, the distribution of SNP across sorghum genotypes, maximum in chromosome 1 and the lowest in chromosome 7, while the marker density is highest in chromosome 7 and the lowest in chromosome 2. Can you go? So in this study, in this study, we find about 215 quantitative trait nucleotides by using all the 6 ml GWAS model and the threshold of load greater than or equal to 4. Among the identified QTN, 50 were for root number, 38 for root angle, and 101 for root dry weight, and 26 for number of root lengths. Among these 215 QTN, 17 of the QTN are identified by 3 or more than models. Chromosome 5 possessed a large number of SNP identified by more than 3 models. There is a load scores. Can you go to the next slide? So in the comparison of the 6 ml GWAS model, the first pillar is identified about 16 QTN. also identified about 15. These are the 3 methods among the ml GWAS model. Array package is the most dominant and useful approach than other models we used. So the reliable QTN identified in the current study advanced genetic improvements of the RSA trait in the future. Similar to our research findouts, different scholars also spotted the same results. Can you go for the second? Next slide. Yeah, again. Again. Here, largest peaks shows the existence of highly significant SNPs according to the Manhattan plots. The same is here. The QQ plots indicates there is a relationship, but only deviate in the case of root lengths, unless the expected and the observed are more similar. Next slide. Among the 17 QTN we identified, 11 of them are co-located with the previously identified by different scholars. The rest 6 QTN we identified are new discoveries or novels. So these 11, 4 out of 5, 4 out of 6, and 2 out of 4 for root dry weights, and 1 out of 2 for root lengths are co-located with the previously reported by QTA different scholars. Next slide. This is a map chart indicating that some of the QTN we find are found in the intragenic region. I think you passed on one slide. Can you go back again? I'm sorry. Here it is, yeah. So among the previously identified QTN, this S5, which is found on chromosome 1, and the chromosome 1, 9, and the chromosome 5, again, chromosome 10, are found in the intragenic regions. That means they are found in the gene sequencing, boundary of gene sequencing. Next slide. So these Q10 are specifically located within the boundary of the gene and they can have direct impact on the expression, especially the phenotypic expression of the plants. Next slide. Similarly, different scholars also reported. So in our study, we find about 118 genes. Can you go on? Among these 118 genes, 46 of them are uncharacterized or unknown proteins, while 72 of them are have a specific gene expression. So further description of uncharacterized protein is required to reveal the exact relevancy of candidate gene in the biochemical pathway. Next slide. So we identified about 17 Q10 among 215. 17 of them are, out of the 17, 11 of them are co-located with the previous identified QTL. The finding of QTL within the intragenic regions of the gene is significant in this study because it suggests that these genes variation may directly affect the function of these genes. So characterization of sorghum germplant collection from Ethiopia is high in resolution. It's essential for presenting. Okay, anyway. So go on, you can finish. So the genomic diversity is, reported in this study underlies the value of sorghum genetic resource from the country of origin and the diversity. So in addition to the staggering traits that was discovered in Ethiopian germplant and they worked by different scientists throughout the world. Here in this study, we find another alternatives for drought tolerance, which we call it the root angle, with 6 different genes for the first time reported. So further study on the breeding tools or root system architecture is very important. So sorghum and the other crop need to be exploited for to make marker tools, especially for traits such as root architecture is very important in the future. Next slide. Thank you. So I just prepared another slide and sent to you last night. This one is the one I sent almost 3 days ago. Sorry for some disturbance. Thank you.
Thank you very much. And I'm sorry we were not able to see that in time for this presentation, but it was very, very loud and very clear. Significant results there, discovery of active genes for drought tolerance, which is key for us, especially now with all the changes that are happening in our environment. Thank you very much. Please stay on the line. We will have a question and answer session after our last presentation. Another newly minted doctor, Dr. Beatrice Gashie. I'm sure you're still getting used to the new title. She graduated just at the end of June with her PhD in molecular biology from the Pan-African University Institute for Basic Sciences and Technology. Her research focused on identifying molecular markers associated with resistance to anti-malarial drugs in the rodent malaria parasite Plasmodium berghei and human malaria parasite Plasmodium falciparum using plasmogem factors and CRISPR-Cas9 gene editing technology. So Beatrice is going to give us a talk on fitness cost and a marginal increase in lumefantrine susceptibility of Plasmodium berghei system desulfurate for knockout of parasites. Beatrice, you're welcome. You have the floor. Let's give her a round as she comes up.
Good afternoon, everyone. As mentioned, my name is Beatrice, and I'd like to present part of my research that I did for my PhD, a small part that's in relation to this conference, and it's entitled Fitness Cost and a Marginal Increase in Lumefantrine berghei Cysteine desulfurase surface knockout parasites. Next slide. I'll give a brief introduction on malaria. Malaria remains a disease of significant public health concern, especially in Africa. And it is caused by female Anopheles mosquitoes during their blood meal. There are approximately 156 known species of Plasmodium that infect different categories of vertebrates. However, only 5 species are currently known to infect humans. These are Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and Plasmodium knowlesi. However, Plasmodium falciparum is the most virulent species, is the most virulent species, and is known to cause most of the mortality and mobility related to malaria. And Plasmodium berghei, let's go back. Plasmodium berghei is a species of Plasmodium that infects rodents, such as mice. And this species is especially important because it's used as a surrogate for humans parasites are closely related in terms. As I mentioned, Africa as a continent is still bearing the largest brunt of malaria infections globally, with children below the age of 5 and pregnant women being the most vulnerable groups to malaria. I hope the slides are clear. But I'll explain. According to the World Malaria Report that was published by the WHO in the year 2022, there were 95% of malaria deaths and 94% of malaria cases occurred on the continent. And out of this, 75% of the total malaria deaths occurred in children below the age of 5. And the other vulnerable group, which was the pregnant women, as I mentioned, 36% of these women were exposed to malaria during their pregnancy. And this led to a number of adverse effects, such as low birth weight of the children, preterm births, and spontaneous abortions. As we can see from these diagrams, most of the countries that are having the highest burden of malaria are in Africa. We have Nigeria, DRC, Congo, Angola, and others. Next slide, please. A number of strategies have currently been put in place for malaria control methods. One being vector control methods. These include deployment of insecticide-treated bed nets. I'm sure most of us are familiar with this. And indoor residual spraying, especially in regions of moderate to high malaria infection or transmission. Another key tool is accurate diagnosis We currently have rapid diagnostic tests that are used to accurately diagnose patients that have malaria. Another method is chemopreventative methods. Here we have two main methods, that is intermittent preventative therapy in pregnant women and children, that is IPT, and also seasonal malaria chemoprophylaxis, and this is mainly rolled out in areas with seasonal malaria infections, such as the Sahel regions of Africa. And recently, the WHO approved 2 malaria vaccines for rollout. This is pretty recent. The first one was the RTSS vaccine. This was rolled out in 2021. And the second one was the R21 Matrix M vaccine that was rolled out last year. However, in cases where any of these control measures fail, that's where active case management is instituted, and this is done using anti-malarial drugs. Next slide, please. Currently, the first-line drug for uncomplicated P. falciparum malaria is based on artemisinin combination therapies. So these drugs consist of 2 main elements, an artemisinin derivative that typically has a short half-life, and this is coupled to a partner drug that has a longer half-life. Some of the most commonly used partner drugs, especially in Africa, are lumefantrine, amodiaquine, piperaquine, but we have others such as sulfadoxine, perimethanine, and mefloquine. Next. And one of the largest problems we currently have in tackling malaria or reaching malaria elimination is the development of antimalarial drug resistance. This resistance has previously been reported in former first-line drugs, such as chloroquine and sulfadoxine perimethamine, or Fancida, as some of us would recall from the development of resistance to these drugs. ACTs were instituted, as I mentioned in the previous slide, but there's a growing concern about the development of resistance to ACTs because recently there's been resistance that has been detected in East and Central Africa, countries such as Uganda, Rwanda, and last year in Tanzania. So there's a concern about the development and spread of resistance to ACTs. This comes to the focus of our study. This because we need to understand underlying mechanisms that are responsible for resistance in malaria parasites, and we need to identify genetic markers of resistance in addition to identifying new drug targets that can be used to develop the next generation of malaria drugs. Next slide. In this talk, I'll focus on one of the genes that we studied. This is the cysteine desulfurase surface gene. This gene is part of the iron sulfur biogenesis pathway. This pathway is very important because it provides iron sulfur clusters for intracellular redox reactions. And these reactions are very important in parasites of the apicoplex genus such as malaria. The other importance of this gene is because other genes in this pathway have previously been identified as modulating drug responses in previous studies, especially those done in Africa. Next slide. We had 2 broad objectives in relation to this. This was to determine the growth rate phenotype of Plasmodium berghei parasites after the knockout of this gene. And the second one was to determine or to measure the impact of the knockout on lumefantrine responses in P. berghei parasites. I'll rush through the methodology. We began by preparation of the gene modification vectors. These gene modification vectors were known as plasmogem vectors. And this was done by culturing of the agar stubs that contain the vectors in terrific broth that was supplemented with kanamycin. After this, we did a plasmid isolation using a commercial kit, followed by restriction digest using NOT1 restriction enzyme, and finally ethanol precipitation. After this, we moved on to prepare the parasites. This was done using Schizont culture media, and the parasites were purified using an eco-dense gradient. After this, we transfected the parasites with the vectors, and we used the nucleofector Amaxa kit. Once we transfected the parasites, we infected mice began selection using pyrimethamine drug, did genotyping, and finally a drug sensitivity assay. Next slide. The preliminary results we got, the graph, the figure on the left, this was the expected restriction digest pattern we got after gene electrophoresis. And what this showed was that the vector backbone was released. Therefore, we were able to get the insert that we needed for the knockout. And it also showed the integrity of the knockout vector prior to transfection. And once transfection was done, we were able to genotype using 3 genotyping primers. And this was done on both knockout parasites and wild-type parasites. And what this showed was the presence of the unmodified loci in the wild-type parasites, which was absent in the mutant, and also presence of the marker cassette in the mutant versus the wild-type. And we also were able to show the correct integration at the right position in the transfected parasites compared to the wild-type parasites. Next. Finally, we went to the parasite growth phenotype. The graph on the left shows that the growth rate phenotype was significantly reduced in the transfected parasites compared to the wild-type parasites. The graph on the right shows that there was a slight reduction in parastemia of the transfected parasites versus the wild-type parasites. And what this showed was that there was a marginal increase in the susceptibility to lumefantrine in the transfected parasites. Next. A brief summary of this particular study was we were successfully able to generate transgenic parasites that had the cysteine sulfase, that had the cysteine desulforase surface gene knocked out. And this is very important because it can be used for consequent studies using a variety of different drugs. And one of the major findings was the reduction in the growth phenotype was especially important because this suggested that this gene had a significant contribution to the fitness of the parasite. So what this means is when this gene was knocked out, there was a fitness cost on the parasite. And this implies that this gene is essential, especially in the asexual blood stage of the parasite. However, given all this, we still need further validation in order to confirm the specific role of this gene, first in modulation of lumefantrine drug susceptibility, and also its exact role in the asexual blood stage of the parasite. The final slide. And I would like to acknowledge the Pan-African University, the Jomo Kenyatta University and KEMRI, and also like to acknowledge the Wellcome Trust Sanger Institute, who were gracious enough to provide us with some of the gene modification vectors. Thank you.
So I'd like to invite all our presenters, the ones who are here physically, to come and sit here in front. Manicus, please come here. And this is your opportunity to ask questions. Any questions from the floor, as we also look for the questions that are there online. I can see 2 hands, 3, 4. So you can direct your questions specifically to the presenter.
Okay.
I'm missing Eunice. Eunice, yeah, please come.
Thank you very much. I'll introduce myself first. I'm Dennis Muhongo, a very proud student to Professor Anne Muigai. She supervised me in my master's and also my PhD. I currently lecture at Alupe University. I'll begin my question to the first presentation about the use of insects in as biopharmaceuticals. Maybe the first question is why insects? Because culturally you realize there are some communities that won't eat some insects. So then how is your level of applicability across? Another thing is on still the same aspect of sustainability. Assume you mentioned termites. Where I come from, we eat them. And we eat them whole, which means if it is having a biopharmaceutical product, then I've got to eat in the morning, look for the same in the afternoon, and possibly in the evening again. So I'm thinking then, why not just use a fruit plant where I can pick a fruit pick a fruit and pick a fruit, or not? Why not use a plant where I can eat a leaf, if it's, for example, sukuma wiki? Because if it's an insect, it's the same at different times, unlike a plant where I pick part of it in the morning, part of it tomorrow, and part of it later on. Again, I realized that when you mentioned your weaknesses of the work, there seemed to be more than the strengths. So do you think then your policy recommendations can actually take off? Because you already have weaknesses that are more than the strengths of what you are suggesting. And the last question to the malaria study, you talked of RDTs being accurate diagnostic mechanisms. I doubt if we can mention the word RDTs and accurate in the same sentence, because I think the slide method is the gold standard. So I think the RDTs will just diagnose, but not accurately. Thank you.
Thank you, Dennis. And next question, maybe just limit it to one question. Yes, go ahead. Oh, please, here and then there.
Yeah, so my name is Michael Landy from IIT at ILRI. I'm also a PhD student at SLU. So my question is on the sorghum presentation. So a little bit curious about the QQ plot he presented. My question is, you already showed that there was a deviation of p-values as to the expected. So did you consider like correcting or adjusting these p-values and see if your model really works on the root length? And then also, I was also curious about the Manhattan plot, even though you skipped on this slide, just to know what kind of threshold you use to get the significant SNPs that you show in the Manhattan plot, and how you corrected or considered if you are seeing any false positive or false negatives in the SNPs you got. Yeah.
Thank you. That was directed to Dr. Masarat, who is online. I hope you have been able to capture that. The next question, please.
Okay, thank you. My name is Derek Ouma from Kenyatta University. Yeah, and my question actually goes to the first presenter and the last one. So on the first presenter, actually, I'd like to know if, considering now the approach of your study, yeah, is it possible to make like a broad-based drugs? Now, if I consider now the insects, for making the drugs, and then another concern I had was now with the ongoing, climate change, I believe there's a mutation in the genomes of actually the insects. So in case, there's a mutation in the process, is it likely to affect the results of your study? So, and then to the last presenter, so there was actually I'd like to know if, the RDTs normally target the antigens, in malaria diagnosis. So is it possible now to try to take, the ideology of the RDTs now, but we target the molecular aspect. So we have like, we target the, instead of antigens, now we incorporate now the genomes from your study, and then, try to put it in, the rapid kits, and now we do the rapids, but target is on the, the molecular genes. Thank you.
I have a question for you.
Sorry. Good afternoon. I'm Dr. Rebecca Karanja from Kenyatta University, an alumni of this university. Professor Moigai was my mentor. My question goes to Eunice Mulango. The first question is whether mass production of the insects for biopharmaceutical purposes can have an impact on biodiversity. Can they get invasive? 2, what challenges would be associated with harnessing insects for biopharmaceuticals? And 3, what regulatory and biosafety issues need to be considered when using insects for biopharmaceuticals? Thank you.
Thank you. I have a question for Eunice and Manicus. To Eunice about the insect. It's all joined the biodiversity aspect. And I wanted to ask the comparison to plant species that have been used previously for drug development, like the case of Prunus africana. We noticed that after some decade, the plant went into distinction or category. So I want to know, is this something that can happen to the insect? Can this be incorporated in the policy in terms of sustainability and the conservation of those insects of interest? So that is for Eunice. For Manicus, you mentioned about the sequencing you did before and after. And before that, you did some cell culture. And you noticed that after the encapsulation, you didn't find any contamination. We have some microorganisms that are not able to be cultured. They are not culturable. Did you also consider sequencing the after encapsulation to see if you will have any gene that or pathogen that were not able to be cultured? Thank you.
Thank you for that. I noted that there's a question online from Tobias Innocent. Please go ahead, ask your question. You're unmuted.
Yeah, thank you very much. I've been following the presentations and I'm delighted to be part of this. I should have been there physically, but just noted during the first presentation, some logistics of being physically present was what delayed me. Otherwise, I had acceptance to be in Ethiopia. Now, I have earlier dropped a question about the possibility. Anyway, that one may be taken by the keynote presenter from Kenya. Maybe will answer me later about the possibility of getting fellowship for online collaboration. Maybe one, a scientist from Nigeria, myself, for instance, coming over, where perhaps the facilities are there to join and develop capacity further by way of enhanced development. That's by the way. Then two, it's about concern about possibility of sharing these presentations so that we'll sit back and be able to digest them and get something better out of them. Then my third question now, third comment on our question now goes to the man that produced the bromelain. I think that should be the first presenter, the man in this session now where questions were not asked. You know that from the organic sources like pineapple, which is the raw base from where you started, given the admixture of so many things there, how did you handle the, of course, it's known that apart from the therapeutic possibilities, there is enzyme activities. So how did you handle the pH issue to ensure stability? I think I have a personal concern for this. I don't know if I'm clear. Hello, am I clear enough?
Very clear. Thank you. We've got a question. Hopefully we'll be able to address it in just as a clear matter. And I would like to invite our participants, Elias, Dr. Elias from Ethiopia. You can answer the question on sorghum. Maybe they can answer the questions first and then we come to another round. Dr. Elias, are you online? Okay, as we wait for him, then we'll go in this order: Eunice, Manicus, and Beatrice.
I'm here, Professor. I'm here, Professor.
Yes, there was a question on sorghum. Did you hear it?
Yes.
Okay, please go ahead and answer.
Okay, thank you, Professor. I want to say something that I am expected to be there, but due to lack of exposure for air ticket and accommodation, I didn't avail myself in Kenya. Let me respond on the question. The question is very good. Regarding the quickie plots, we did with 3 different models. So that is the result of 3 models. There is a deviation on the root lengths, that's clear, but we considered because that is a result of 3 different models we used. For example, if it is with one model, for example, we did also with TASR. In that case, it is totally coincide with the expected. But here, this result is the result of 3 models we considered for analysis. The other thing regarding the Manhattan plots, we considered the load score greater than 4, which is very important and advisable to get reliable QTN, major QTNs. For all, regarding the phenotypic and the GWAS paper are published on the rhizosphere and also on plant genome, so you can easily find and use for personnel. Thank you. I've finished, Professor.
All right, thank you very much for addressing that question. I hope it has been answered adequately. We'll go on to Eunice.
Thank you for your questions. The first question was why insects? And I can combine it with the other question, why some people have not accepted the use of, some people don't accept the use of insects. So it's just a proposal, a research proposal that it can be an alternative source or an alternative way of exploring for biopharmaceuticals. And even a good example is the use of fish for production of omega-3. Insects could have unique compounds and unique vitamins and unique antibodies that could not be found in any other source. And so it's a proposal that scientists can explore that route and find out what is unique about insects and what can be gotten from insects for biopharmaceuticals. And there's something called public awareness for those people who might not be able to accept, can be educated and explained to the importance maybe of the use of insects. For example, the omena has a very good compounds, the omega-3 and all that, but not everyone has accepted. But time and time again, people have been explained to the importance of consuming something like omena, and recommendations have been given, even for small children to take omena. So such thing can also be employed to use of insects. For example, the termites that are usually used, consumed in Western, people can be educated and told about the importance. So the other question is about the weaknesses, and some insects don't have a lot of weaknesses when you're using them or exploring them. For example, the black soldier fly that has been farmers are rearing and breeding is not complicated. You just harvest it at the larva stage and use it for different purposes. So some insects that are not as complicated can also be used for this purpose. And then is it possible to use insect for broad-based drugs? Yes, it is possible. Some companies and research institutions like ICIPE can take up this and begin employing these kind of research and study, and then it can be materialized. And yes, mutations can affect everything. Mutations can affect insects, human beings, every kind of creature. And research, the scientists just need to know how to adjust to that. And challenges to use of insects as biopharmaceuticals, there is the issue of mortality. Insects can have diseases and die. And then the regulatory research has to be done, and policy recommendations done and taken up to the Ministry of Agriculture. Then from there, they can begin to implement the policies. And then concern the biodiversity, use of insects can, the way the domestication of bees has been done, there is a way that bees are usually kept in a certain place where they will not affect the environment. Yes, they can affect, but they need to be regulated on how they are domesticated. I think that's all I have.
Thank you very much. Next.
Thank you. To answer the question from Nigeria, how did you handle the pH issue during the production? During the production, when I start with the crude extract, the dialysis, and the nano-encapsulation, I measure the pH. All those pH are measured. And for more output, you can find it in my thesis. Maybe someone who needs this or those results, I can share with him. For the after encapsulation sequencing, I did the total plate count for the crude extract and the final product. I did it for the crude extract. I incubated it for 24 hours. I saw a growth of bacteria, but for the final product, which is nano-encapsulated bromelain, I left it for more over the time, 72 hours. But at the end, I was not able to see any contamination. So I decided not to proceed on the final product because it showed that the product is already safe to be used and without contamination.
Sorry, please. I do not know whether, sorry, ma, Prof, he said, I didn't understand what he said. I can find it where. Is this paper published? If yes, give me the source. I'm interested to see how you handle them, because given the crudity of the source, it is likely there are going to be variabilities in terms of pH. And I personally, because I have a work that is ongoing in this direction, of course, with respect to enzymes now. So I want to find out how you said you handled it. Otherwise, let me have the source of your publication, because I didn't understand what you say, where you say I should go and find it.
Okay, there's a paper I published already online on World Veterinary Journal. You can find the detail there. And even maybe you can text my email address there. You texted me, so we can talk about it more.
Okay, please, I plead that these papers be shared so that I can have the email address he's talking about. I can't see it from the screen here.
Okay, you will go on Google. You take protein concentration on thermetic activity and microbial contamination in batches of nano-encapsulated bromelain under the name of Manicas Rodolpha Basebank Kotangu, and you will find my email there so we can talk.
So we circulated the book, the conference booklet. It has his name, the full name, and the abstract. I think if you take the full name, you'll be able to find him online. But you can also write to info@africabiogenomeproject.org. I will get the email and I will be able to connect the two of you. Beatrice?
Thanks. Dennis, I think I'll answer Dennis's question first. That's correct. Actually, microscopy is the gold standard for diagnosis of malaria, but RDTs are currently being used in conjunction. Yeah, that I need to make that clarification. And the question on, I think the use of our results for RDTs, this is one of the outputs we are continuing with our work. As Prof mentioned, we are continuing with falciparum using CRISPR-Cas, and one of our hopeful outputs is using it as a diagnostic tool. However, in terms of the gene, for for a gene to be identified as to be used for RDTs, it has to be highly polymorphic. Like currently it's HRP2 that's being used. So we have to confirm that with field studies that our samples are actually found and they're highly polymorphic before we proceed with that. Thank you.
Thank you. I think there was a question on Africa BioGenome Project fellowships. We have a presentation by Dr. Thangod and Professor Julian Usuji tomorrow that will talk more about the fellowships and how you can be able to apply for them, who is eligible, and the fellowships that we have that are going to come. With regarding to the presentation sharing, we will seek the permission of the people who have shared their presentations with us. Once we have their permissions, we will be able to share them to the mailing list that we have developed. So dear participants who are physically here and online, I realize it's 13:30, that is 1:30 our local time. We would like to take a 45 minutes lunch break so that we can meet here at 15 minutes past the hour, quarter past the hour. We have 3 serving points. You have all been given meal cards. I would urge people to exit. There are 2 serving points out there and a few can serve here. Remember, we have state-of-the-art recycling bin. Please, for your serviette and for the plastic paper that you will that is covering your water bottle plus your soda bottle, please come and dispose it in our recycling bin so that we can be able to be green and sustainably recycle all our waste. And then we will get a report on how much our 2 days have generated waste. Thank you very much and enjoy your lunch.
Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated. in today's video, I'm gonna take you through the whole process of ordering a product, the same website. So to start off, Africa Biosystems, the company, focuses on key sectors. Right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click on either of them, we get to find even more categories, subcategories inside, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And we are actually here on the life sciences group table page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just go straight to biosciences division and just click that. From here, I'm able to see some of the products under biosciences divisions, and quickly, I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I just come here, click this fetal bovine product. It takes me now to the product page for fetal bovine serum, FPS. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and availability. And that's how you actually make an order in this website, okay? So to make an order or even request for price, you just click on this button here, which takes you straight to the checkout section. And from here, this is where you're being asked a few questions before the order is actually placed. Yeah, so when you're here as a client, just get to fill in a few information. So I'll just do it quickly. Probably I'll just say I'm from ABC company. Then just put in an email. Yeah, ABC company. Let's say the email. Then I'll just put in my name. So maybe John. Name is John. And the last name is what? John what? Here I can either put in the phone number. I think the phone number is actually optional, but actually good, so that sales team are able to get to the client in the quickest manner possible. Okay, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. The next thing is just to place order as the client. So I'll just click here. The order is being processed, and shortly the process should actually be successful. Yeah, so as you can see, as a client, I'll just get this message. Thank you, your order has been received. And it will go now to the relevant department of the sales department, so that they can now check back with the client, gets to advise exactly how much the costings are. Yeah, so that's it for our presentation. I hope everything was helpful.
My name is Clayton Miller, and I am the application specialist for genomic services here at Eppendorf. In 2020, AfricaBP introduced a flexible treatment system on the African continent, which allowed us to produce high-quality data 8 times more than we would in the simple system.
Professor Kanganezeni Makori, a Deputy Executive Dean at the College of Agriculture and Environmental Sciences at the University of South Africa. So this is very excellent news for us and a good opportunity for us seeing the PacBio platform actually growing in South Africa, especially now the acquisition of PacBio Review. Thank you.
My name is Matati, an NGS lab technician here at Minaba Biotech. Using the new review system, fewer kits are required in library prep up to sequencing across the various applications we provide. Using the new streamlined workflow, library prep is quick and efficient.
Hi, my name is Howard, part of NGS team at Global Biotech. And one of the stuff that handle data that comes up into, I think one of the advantages of the system is that not only does it produce long, high quality reads, but when you actually work with these reads, it gives you one of the most comprehensive views of the genome. From the review data, not only do we have access to the genomic landscape of the genome, but we also can zoom down into single nucleotide variations, which is very important for a variety of applications. So I'm very excited with the data that comes off this instrument and for its future applications.
In addition to the review system, which is our long-read platform, the ONSO system is based on the sequencing by binding chemistry, which is a major contributor to the type of data that you get. By the type of data, I mean the Q40 plus level of arches. The independent process lanes and the external clustering brings a level of flexibility that has never been seen in the short-read market before. Open arches will allow our customers to sequence at much reduced depth and also reduce the cost of sequencing. We are so thrilled to have this system and start offering the services to our customers across the African continent. Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated.
In today's video, I'm gonna take you through the whole process of ordering a product, the same website. So to start off, Africa Biosystems, the company, focuses on key sectors. Right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click on either of them, we get to find even more categories, subcategories inside it, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And we are actually here on the life sciences group category page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just go straight to biosciences division and just click on that. From here, I'm able to see some of the products under biosciences divisions, and quickly, I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I can just come here and click this fetal bovine product. Takes me now to the product page for fetal bovine serum, FPS. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and availability. And that's how you actually make an order in this website, okay? So to make an order or even request for price, you just click on this button here, which takes you straight to the checkout section. And from here, this is where you've been asked a few questions before the order is actually placed. Yeah, so when you're here as a client, just get to fill in a few information. So I'll just do it quickly. Probably I'll just say I'm from ABC company. Then just put in an email. Yeah, ABC company. Let's say the email. There I'll just put in my name. So maybe John. Name is John. And my last name is Wad. John Wad. Here I can either put in the phone number. I think the phone number is actually optional, but actually good, so that sales team are able to get to the client in the quickest manner possible. Okay, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. The next thing is just to place order as the client. So I'll just click here. The order is being processed, and shortly the process should really be successful. Yeah, so as you can see, as a client, I'll just get this message. Thank you, your order has been received. And it will go now to the relevant department of the sales department, so that they can now check back with the client, get to advise exactly how much the costings and everything. Yeah, so that's it for our presentation. I hope everything was helpful.
Hello there. My name is Ethiopia, and I am the application specialist for genomic services here at Eppendorf. In 2020, AfricaBP introduced a flexible treatment system on the African continent, which allowed us to produce high-quality data 8 times more than we would in the simple system.
Professor Tanganezeni Mapori, a Deputy Executive Dean at the College of Agriculture and Environmental Sciences at the University of South Africa. So this is very excellent news for us and a good opportunity for us seeing the PacBio platform actually growing in South Africa, especially now the acquisition of PacBio Review. Thank you.
My name is Matati, an NGS lab technician here in our biotech. Using the new review system, fewer kits are required in library prep up to sequencing across the various applications we provide. Using the new streamlined workflow, library prep is quick and efficient.
Hi, my name is Hamilton, part of the NGS team by Global IT. And I'm one of the staff that handle data that comes up with this. I think one of the advantages of the system is that not only does it produce long, high quality reads, but when you actually work with these reads, it gives you one of the most comprehensive views of the genome. From the review data, not only do we have access to the genomic landscape of the human genome, but we also can zoom down into single nucleotide variations, which is very important for a variety of applications. And so I'm very excited with the data that comes off this instrument and for its future applications.
In addition to the review system, which is our long-wing platform, I'm thrilled to introduce to you the new PacBio system, which is a short-wing platform. The ONSO system is based on the sequencing by binding chemistry, which is the major contributor to the type of data that you get. By the type of data, I mean the Q40 plus level of accuracy. The independent process lanes and the external clustering brings a level of flexibility that is never seen in the short-wing market before. The accuracy will allow our customers to sequence and much reduce the depth and also reduce the cost of sequencing. We are so thrilled to have this system and start offering the services to our customers across the African continent.
Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated. In today's video, I'm going to take you through the whole process of ordering a product, the same website. So to start off, Africa Biosystems, the company, focuses on key sectors. Right? So for us to get to explore these key sectors, just put your cursor just directly on the navigation bar, put your cursor on top of products. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life sciences solutions, there's surgical, there's analytical solutions, there's forensic solutions, there's clinical diagnostics, service support, and lastly, we have general products. If we click on either of them, we get to find even more categories, subcategories inside, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And we are actually here on the life sciences group table page. When you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just go straight to biosciences division and just click that. From here, I'm able to see some of the products under biosciences divisions, and quickly, I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I can just come here and click this fetal bovine product. It takes me now to the product page for fetal bovine serum, FPS. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and availability. And that's how you actually make an order in this website, okay? So to make an order or even request for price, you just click on this button here, which takes you straight to the checkout section. And from here, this is where you're being asked a few questions before the order is actually placed. Yeah, so when you're here as a client, just get to fill in a few information. So I'll just do it quickly. Probably I'll just say I'm from ABC company. Then just put in an email. Yeah, ABC company. Let's say the email. Then I'll just put in my name. So maybe John. It's John. And my last name is Wad. John Wad. Here I can either put in the phone number. I think the phone number is actually optional, but actually good, so that sales team are able to get to the client in the quickest manner possible. Okay, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. The next thing is just to place order as the client. So I'll just click here. The order is being processed, and shortly the process should actually be successful. Yeah, so as you can see, as a client, I'll just get this message. Thank you, your order has been received. And it will go now to the relevant department of the sales department, so that they can now check back with the client, gets to advise exactly how much the costings are. Yeah, so that's it for our presentation. I hope everything was helpful.
Yeah. In 2020, Internal Biotech introduced a flexible treatment system which allowed us to produce high-quality data 8 times more than in the single system.
Professor Tanganezeni Mapoli, Deputy Executive Dean at the College of Agriculture and Environmental Sciences at the University of South Africa. So this is very excellent news for us and a good opportunity for us seeing the PacBio platform actually growing in South Africa, especially now the acquisition of PacBio Radio. Thank you.
My name is Matati, an NGS lab technician here at Illumina Biotech. Using the new review system, fewer kits are required in library prep up to sequencing across the various applications we provide. Using the new streamlined workflow, library prep is quick and efficient.
Hi, my name is Hamlet, part of NGS team at Global Biotech. And I'm one of the staff that handle data that comes up with this. And I think one of the advantages of the system that not only does it produce strong, high quality reads, but when you actually work with these reads, it gives you one of the most comprehensive views of the genome. Review data not only do you have access to the genomic landscape of the genome, but we also can zoom down into single nucleotide variations, right, which is very important for a variety of applications. So I'm very excited for data to top of this instrument with switch applications.
In addition to the radio system, which is our platform, I'm thrilled to introduce to you new PacBio system, which is AfricaBP. The auto system is based on the sequencing by binding chemistry, which is a major contributor to the type of data that you get. By the type of data, I mean the Q40 plus level of options. The independent closed lanes and the external clustering brings a level of flexibility that is never seen in the short market. Or ARCs will allow our customers to sequence at much reduced depth and also reduce the cost of sequencing. We are so thrilled to have this system and start offering the services to our customers across the African.
Welcome to AfricaBiosystems.com. This new website is rich in resource, simple and intuitive, with relevant content that is continuously updated. In today's video, I'm going to take you through the whole process of ordering a product. So to start off, Africa Biosystems, the company, focuses on key sectors. So,
hello everyone. Lunch is over and let's come back for the afternoon session. If you're still eating, you can come in with your food or your plate. Continue enjoying, eh? So we get started for the afternoon session. And this is on the emerging technologies in genomics and scientific innovations. And in this, with the line of AfricaBP, which is focusing on the digital innovation in Africa for a sustainable agri-environment. The presenters for this session will take us through the cutting-edge scientific innovations that we can apply within the continent through collaboration to achieve the sustainable goals. The first presenter for this session is Dr. Cecilia Waroyo. And Cecilia is a genomic scientist, an entrepreneur, an educator committed to advancing biology, molecular biology, virology, and genetics in Africa. As co-founder and director of the African Genomes Center and Consultancy Limited, and director at NextGen Molecular Lab Kenya, she has significantly impacted the genomics landscape. Her achievements include leading the African Genomics Center and Consultancy Limited in managing the COVID-19 pandemic in Kenya through various variant sequencing and surveillance, delivering high-quality full genome SARS-CoV-2 sequences. At NextGen Molecular Lab, she has driven innovation in molecular and genetic testing, addressing diagnostic challenges like bacterial sepsis with advanced methods such as shotgun metagenomic sequencing. As a lecturer at Jomo Kenyatta University of Agriculture and Technology, Cecilia shares her practical knowledge inspiring students to apply their skills to real work problems. Her research on virus detection in bats in Kenya has underscored the importance of disease surveillance. Dr. Wario envisions a future where genomic applications in Africa achieve new heights, particularly in precision and personalized healthcare. I hope the online team as well is with us. And here we have Cecilia.
Thank you so much, Kuja, for that introduction. So I'm delighted to give you a talk today on integrating genomics into innovation and entrepreneurship. My journey began at Jomo Kenyatta University of Agriculture and Technology, where I studied biochemistry and genetics. And then I continued to pursue genomics in higher learning. And this has led me down an interesting path. Next slide. So as scientists, which most of us here are, when we get into the career of science, our basic mindset is we get into either basic research or applied research. And our idea is we want to solve problems that are affecting the society, right? We're looking around and thinking, okay, there's malaria. What do I do about the insects? What do I do about the parasites? What can I do about drugs around this and vaccines, right? And there are different levels. Basic research is where we get our knowledge built. That is where we do a lot of questions asking, why is this happening? How is it happening? When and who is contributing? And we build our knowledge database. And then from that, we move to applied research, where now the knowledge that we have, what do we do with it? How do we apply it to solve the problems of the society? We generate new products and approaches to solve our problems. And then the next level is we have a vaccine or we have a drug that starts to treat disease, for example. So I would like to open with that saying from Thomas Edison, which says the value of an idea lies in the using of it. If you just have an idea and you sit with it, it does not help society. It does not become valuable for us, right? So I like telling stories. So I'll give you a story about Dr. Jennifer Doudna. Sorry for the typo. I'm sure some of you know her. Someone here mentioned that they used CRISPR technology in their lab to do their work, right? So Dr. Jennifer Doudna with her colleagues at the University of California, Berkeley, while doing the basic research of understanding an immune pathway in a bacteria, which is the CRISPR-Cas9 tool, they realized that this tool can be implemented much further than just in the bacteria, because they saw it's a tool that is able to cut DNA very precisely. And they asked themselves a question, what if we can apply this tool to also solve problems that exist in our real life, right? In maybe coming up with therapeutics around genetic disorders. So this is basic research, right? Studying bacteria and the mechanisms that the bacteria uses to fight viruses, the immunity of the bacteria. So from the idea that we can do more, we can do application, they went on and started a company called Editor's Medicine. And when this idea was born, it attracted a lot of attention, and it attracted different funders, as well as companies, as well as high net worth individuals like Bill Gates. It attracted big collaborations from universities like MIT and Harvard. And although they encountered a lot of issues technically, the regulatory issues, when they crossed the boundary of that, we have results where we are transforming healthcare using CRISPR technology. So 2 cases that I can mention here today is the case of Leila, a 1-year-old girl who was born with an aggressive case of leukemia. And CRISPR technology was used to edit donor cells that were injected into this young girl, and her leukemia went into remission. Today, she's still alive thanks to the science, application, and going all the way, right? Another one is, the lady from the US who was treated for sickle cell anemia. We all are familiar with sickle cell anemia, which results in deformed hemoglobin. So stem cells were extracted from her and they were transformed using CRISPR and they were injected back into her. And this resulted in her being able to generate normal hemoglobin, and her symptoms were really reduced. So she's able to persevere through the symptoms of sickle cell. Now, the reason I give this story is because the theme of my talk is for us to understand the opportunities that exist around science. Yes, some of us will go into academia. Some of us will go into industry, which is where I am today. Because the reason we do research, the reason we invest so much in research is not so that we publish and have high impact factors, right? It is because we want to solve the problems that exist in society around us. I will give another story of this gentleman, Lauren Fletcher. So this is a former NASA engineer who built a company called Biocarbon. And the idea of his company is to be able to do reforestation en masse, like large-scale reforestation, because there's a lot of deforestation happening, right? So we need to be able to sequester carbon for climate change action. And his innovation is brilliant because he combines a multidisciplinary team to build a drone that is able to shoot seeds into the earth at the right depth for the seeds to be able to germinate. And they've been able to regenerate quite a number of deforested areas. Yeah. So the perception I want you to get at the end of it is you can do more from the bench. We need to go beyond the bench. Next slide. Now, what is genomics? Most of you here know what genomics is, and genomics does not exist in isolation. Genomics is when we study the DNA of an organism in its entirety. There are different aspects that are associated with genomics, which include metabolomics, where we study the metabolic pathways. There's proteomics, where we study the proteins. Of course, bioinformatics, as we mentioned here. So genomics cannot exist as an independent discipline on its own. It needs all these partners to tag along and make it more useful. So genomics in itself is revolutionizing many industries in the space of healthcare, agriculture, science, environment, bioremediation, and conservation efforts. Next slide. So I'll touch on each very briefly because you can come for the class when we are to go in depth. So in the space of healthcare, genomics is applicable in the space of personalized healthcare and precision healthcare, as well as from a public healthcare angle. So in personalized healthcare, genomics has been applied to understand the whole human genome, for example. We all know that the human genome was fully sequenced by the year 2003, and we're building up on that as we go. And with this knowledge, with this information, we've been able to identify disease associations from the genetic point of view. And because of that, we are able to do accurate disease risk prediction, genetic diseases, and then we can modify lifestyles to do prevention, right? Then we're able to do disease diagnosis. A case and example is in COVID, for example, because we were able to sequence the whole genome of COVID. When the pandemic happened, we were able to develop tools rapidly that were able to allow us to quickly diagnose, quickly trace the variants, and be able to manage the pandemic efficiently. A case that I'll give is in the chronic myeloid leukemia, where genomic sequencing was able to identify that a mutation that is a fusion between the BRC and ABL gene causes the expression of a tyrosine kinase, which causes overexpression, leading to the tumor formation. So with this, a therapy was able to be developed that is very targeted, that attaches itself to the receptor point of the ATP. So it disrupts the pathway, which means it prevents the tumor formation. So by us understanding the pathway, we were able to develop a targeted therapeutic. The power of genomics, proteomics, and all other omics associated. Public health, genomics is a very important tool for surveilling disease, like understanding dispersion and distribution of organisms in terms of infectious diseases, for example, as well as risks of chronic diseases within populations. Next slide. And where does this lead us in terms of application, entrepreneurship, and innovation? So this allows us to enter spaces of disease mechanisms research, marker discovery. We can do drug research and design, therapeutics, as well as conduct clinical trials. And we can also be in the space of looking at the ethics, the legal and social implications of applying genomics. In the industry, there are various things that come out of healthcare genomics. So there's diagnostic testing, developing the tools, offering the services of testing. We have the pharmaceutical industries benefiting from genomics, which means they are able to develop targeted therapeutics and diagnostic tools as well. And then the research tools and development services. We have our tech companies here with machines and equipments that have been built through multidisciplinary knowledge, right? A genomics person will say, this is the sequence. A chemist will say, this is the luciferase that will help us identify this. And then an engineer will need to build the machine that does that, right? Next slide, please. In the space of environment and conservation, I like to tell stories or use examples. So here I will give two examples. One is an example of an oil spill. Through genomics, we identified a bacteria that can degrade oil very well, hydrocarbons. Then through genomics, we've been able to increase the efficiency of this bacteria to degrade the hydrocarbons, which means in the cases of oil spills, we are able to go in and bioremediate the environment that has been affected efficiently. Another story that I will give is on the conservation angle. This is in Australia. So the Tasmanian devil is an endangered species, and it was endangered because of a cancer that was very contagious within the population. And through genomics, the study of the genome of the Tasmanian devil, as well as the genome of the those Tasmanian devils that had resistance to the cancer was profiled. And then scientists were then able to breed Tasmanian devils that are resistant to this cancer, and have brought up the populations of this Tasmanian devil significantly. This can be replicated across different organisms. We have heard today about drought-resistant sorghum, right? So it can be replicated across organisms where we understand traits and we are able to adapt other varieties to be able to carry those traits. Next slide. All right, in the space of bioprocessing and manufacturing, we generally apply two different approaches. You can start from the end coming back. What product do you want? What pathway can get you there? Then what infrastructure do you need to build? An example, biofuel produced from yeast, for example, let's say bioethanol. We know yeast does fermentation. We want yeast to be able to degrade certain matter to be able to produce bioethanol efficiently. What modification can we do to make that occur more efficiently? Essentially, increase the pathway that leads to our bioethanol, bioprocessing. That's an industry that also benefits from genomics. I'll give another example. The one on the right, I don't know how many of you have heard of spider goats. Well, I'm here to tell you about it. So scientists decided we need to enhance how fast we get silk. Spiders take time. You have to farm all this and then harvest the silk. So the scientists studied which gene specifically codes for the yeast, for the silk protein. And then they were able to move this gene into the section of the goat that produces milk. So when you get the milk from the goat, you can then isolate your protein from the milk and make silk. And today, this is the silk that we use in surgery. This is what we use to stitch up. Before, you'd be stitched up and then you'd have to go back and get it removed. But now, because we're using a protein, it just meshes into your body. You don't need to go back to remove stitches. Innovation, right? Next slide, please. I'm being exciting because you just had lunch. All right, so in the space of food and agriculture, I'm sure most of you have heard of the GM crops, which have been modified to increase either yield or nutrition or resilience to climate change. This is an area that genomics has contributed to. And in the space of animal farming, we are able to do precision breeding. This has also been mentioned here today. When we look at animals, we are able to breed animals that can produce higher volumes of milk or more meat. They grow bigger. Yeah. So genomics is the beginning of that journey towards improvement of varieties. Next slide, please. Next slide. I have mentioned this one. All right. All right. So this is my second last slide. What I'd say to us today here as scientists is that I feel that we need a paradigm shift. Sometimes we're locked up in our labs just tinkering with our pipettes and slides, and we forget that the reason we began this journey is to solve the problems that are around us in society. So don't let your research stop at stage 1 or stage 2. It needs to come all the way down to the product, solve the problem. And this is where the money is when we talk about entrepreneurship. This is where you cash in with your vaccine. This is where you cash in with your biopharmaceuticals from your insects, all right? We don't need to be poor, as we were told. So go all the way. Let us solve the problems that exist around us using the skills and the tools that we have at our disposal. Thank you. Questions? Later.
Let's appreciate Cecilia for the good presentation. Thanks, Cecilia. Our next presenter is Kelvin Muriuki. And Kelvin Muriuki is a Kenyan innovator and an entrepreneur who commenced his career at Silland in 2011, creating digital content. He later contributed to African streaming expansion at Mudundo while pursuing bachelor's in communication studies with a major in design communication. At Radio Africa Group, he led content innovation for the Songa app. And in collaboration with Bob Mwangi, they co-founded a climate tech startup called Tech Earth in 2021, where they aim to document climate action through data-driven sustainable waste management, combating climate change effects, and potentially revolutionizing emission-heavy sectors. And I think we can see Taka is here with us, and here we have Kelvin. Kelvin, karibu.
Good afternoon, everyone. My name is Kelvin Muriuki. I'm the CEO and co-founder of TechEarth. And I feel like I'm out of my normal space because I'm a social scientist. And here, you're all in a very different field. But we're hoping that we can learn as much as possible from you as we have learned within the last couple of months. And so my main focus of what I'll be presenting today is what we're doing as TACAth, but also how science is an accelerator of climate action. You can go to the next slide. So first things first, I'm going to introduce what we do as TACAth. And we are a climate tech company. So that means that we're building technology to resolve climate change. And that means that what we've then built is an incentive-based recycling platform that is focused on appropriating value into what we consider as waste. And how we're doing that is basically broken into about 4 stages. The first thing is to be able to collect the waste from consumers. And so what we've done is incentivize the consumer such that when you're done consuming your soda, your water, anything that's packaged, that packaging material, return it back to us and we'll give you some money back, right? I hope I got your attention, kidogo. Yeah, so if you're able to give us that money, if you're able to give us your product back or the packaging product, we then deliver that product to a recycler. And we have different recyclers for different products. And as we do this, the money that we give you, you have to redeem it with specific retailers. That way we keep on, adding value to that consumer value chain, right? Because there's the Mamaduka who still needs you to go back tomorrow. So that's our whole focus. And as we do that, what we've then done is innovate based on what our consumers are telling us. And it has allowed us to document what we're collecting. So for every single bottle that you throw in the bin, we're able to tell that this is plastic, this is glass, this is this, and it's coming from all these different people. So that's who we are as TACAth. But what I'd love to do today is talk about science. So please, next slide. Yes, so the main problem that we're focused on is climate change. And I believe we have an understanding of climate change, but our focus is to be able to look at climate change from the perspective of human-ended drivers. So what are we doing as humans to accelerate what is happening within the world and how that is affecting us on a single daily basis? So this is just a small preamble on what we think climate change is, but what is important from a science perspective is we would never have this data if it wasn't for scientists, right? So next slide. Yeah. And so what we've done is be able to understand the problem. And what is usually interesting when you understand the problem of climate change is the different sectors where emissions are coming from. So the biggest sector there that has 72% is energy. Then you have industrial processes driving 6%. Then, please go back. Yes. Then you have agriculture, waste, land use, change and forestry, and then you have smaller bunker fuels. So what is interesting about this data, as I've mentioned, this is data that can only be assessed and contributed from science. But what we then do is looking at this problem is being able to take it to the next level, and it's to contextualize this problem. So when you think about this from an African perspective, what does it mean for us to be a contributor of climate change, or rather for greenhouse gas emissions? So what we did is we took this massive problem and then contextualized it. And when we contextualized it, for us, it was, first of all, understanding that Africa within climate change, one thing is, in terms of energy, we're not doing too bad. We're heavily driven, Kenya being an example, we're heavily driven by renewable energy. Kenya has attained 90%, and they're pushing for 95%. And the rest of the countries within the sub-Saharan Africa are pushing for renewable energy. But when you contextualize within the continent and within the region, what you understand is there's always data that will be generated, but that data has to be localized for you to be able to solve a problem. And what we've done is by localizing this data is we understood that Africa or sub-Saharan Africa had not built an adequate waste management system. Since we were kids, we've been throwing waste in pit latrines, in living it in landfills, and we've not built an adequate waste management system. So what's happening is, as time goes, we then continuously increase the amount of tonnage that we are generating from municipal solid waste, which is waste that comes from your normal house, your normal bin, and for example, what we've consumed there. So what we then did is to be able to look at that and then develop a solution. And this solution is not built on just us. It's built on research that has happened before and platforms that have been built ahead of us. So what we've done is build single-use or single-stage architecture platform that uses YOLO. YOLO is an AI model that relies on image classification. It basically takes an image. It's called YOLO because the computer only looks once. So it's computer vision. But what it then does is, as it looks at the product, you train it based on the products that you're existing within your ecosystem. So when people talk about waste within Africa, what does that mean? And that's what we went and did. We went and understood what is waste within Kenya, what is waste within Uganda, what is waste within Tanzania. And we've then created items or a unique data set of every single product that we've interacted with. And that then creates our library and allows us to develop a solution. And the solution is what you're seeing behind you there. And it's a model. It'll predict what waste type you're throwing in. And then once we predict that, we're able to verify where that waste is ending up. And this would only have been possible because of the advances in computer science and artificial intelligence. Now, being able to collect that waste is only one part of the problem. We then have to figure out what happens to that waste. And one of the things that we've realized, because to go ground, or as they say, we're on the ground. And what we've then done is to collaborate with as many partners as possible to understand how we solve for each waste type that we're collecting. So the beauty of this, and maybe this is where we might find a common ground, is that all of you here rely on data. I've seen the questions that you ask, very tough questions, but you all rely on data. And what we've then done is understand that data of what we are collecting in terms of waste, right? And as we understand that, we then figure out who is going to recycle each and every single waste type that we collect. And that waste type, we have different recyclers. So for example, we have Boredo Engineered Waste Management Solutions. They take LDPE and turn it into, what do we call it? They turn it into new nylon paper that can be reused for different things like building. You can use it to overlay dams, and you can also use it for what? For greenhouses. Yes. Then you have Mr. Green Mr. Green and T3 do PET flakes. So they take the bottles, for example, the water bottles, the soda bottles, and they'll convert that into PET flakes for reuse. Then you have Bottle Logistics who are doing glass. You have Munga who does cans and metals, and then you have Kamongo who does waste paper. But all these people represent collaborations that are required because we cannot do everything. We cannot build everything. And you guys have gone ahead to even do more research. So as we do this on the ground, there's more stuff that you're consistently re-energizing, right? But what happens then is there's things that we cannot recycle. So what happens to that? And that drives innovation and drives our research and development aspect. So we've put in some amount of effort into figuring out what to do with waste types that cannot be recycled. So something that's very interesting is, for example, your soda bottle or your water bottle. So you have three different types of plastics here. You have the bottle top, which is HDPE. You have this bottle itself, which is PET, but then you have this label. Everyone thinks it's paper, but it's also a polymer of plastic. And what happens is you have a recycler for this, you have a recycler for the bottle top, but you do not have a recycler for the label. So there's no way for this company to then say that they're 100% going green because they still have ways that they've not figured out what to do. And that's why we then lean on you guys. So luckily for us, we know a few scientists and we've been doing some research, specifically scientific research, into focusing on how to solve for non-recyclable items. And some of the projects that we've done has been one, transforming organic waste into bioethanol. So organic waste makes up a huge amount of what we generate. It makes up a huge amount of, for example, even what will be generated from this event. When you see the report, organic waste will be tops as compared to the water and everything else. So what's interesting is that then we need to solve for that organic waste and put it into a structure that allows it to create value. And what we've done so far is to create bioethanol out of that. And I think Dr. Cecilia has mentioned it because she's our resident research, but she's mentioned it. And it's purely a focus of bioprocessing through fermentation to then create that. And then the most interesting thing is there's already immediate uptake. So there's a company called Coco. I don't know if you know Coco. And Coco sells gas, and that gas is bioethanol. So one of the things that they do need is actually more people researching into bioethanol because they need to uptake as much as possible. They are importing bioethanol while we still have organic waste. Now, the other thing was used cooking oil. So used cooking oil is a huge problem for a lot of the companies that we work with, hotels, and even the manufacturers such as Bidco. And some of the research projects that we run has been to see, could we turn this into biofuel, specifically with a process that I find it hard to see this name, but I'm pretty sure you guys are transesterification. Yeah. Yeah. So that's what we've been using to focus on that. And we've managed to be able to generate very good results. And we're currently going through the KEBS standardization structure. Then we have another research project that we've been running, which is to convert now non-recyclable plastics, such as this label, into biofuel. And this has also gone very well with a focus on pyrolysis, but trying to avoid syngas. So it's a lot of work that goes in there. But what we believe is the continuous collaboration between entrepreneurs and scientists will every single day drive innovation that we could not have possibly thought of. Because where you guys spend a lot of time in the lab, we're spending time selling to the customers, and the customers do really need the research and the work that you guys are doing. And sometimes it's very interesting. You're researching something totally different, but when I present a problem from a business perspective, you understand what it is from a molecular perspective, and you can be able to break it down. So with that, I would like to conclude my presentation, but I believe there's a lot of focus for science, and we could definitely use science as an accelerator for climate action. Thank you.
Thank you very much. Just let's give him another round of applause for even taking the effort to ferry the recycling bins here for us to be able to envisage. So our next presenter give me a minute, I get to is Dr. Oliver Fedrigo. I believe he's going to present online all the way from the United States of America. Dr. Fedrigo began his academic year at the Museum of Natural History in Paris, studying comparative anatomy and marine biology. He earned his PhD in genetics from the Iowa State University in 2003, where he focused on Islamobunch phylogenetics. He then conducted his postdoctoral research at Duke University in the US, specializing in genome regulation and comparative genomics. Then he became the director of the Duke Sequencing Core Facility between 2010 and 2016, where he enhanced its throughput sequencing capabilities. In 2016, Dr. Federico joined Rockefeller University to establish and lead the Vertebrate Genome Lab, and we have been collaborators with Vertebrate Genome with the Vertebrate Genome Project when he was there, where he oversaw the production of high-quality genomes, contributing over 25 influential papers and sequencing about 200 genomes. Through conservation efforts for species like the Vakila and the Capo. Since mid-2023, he has been the director of genomic resources at Colossal Biosciences, leading genomic analysis and guiding de-extinction of species projects. This is including the Dodo project. So Dr. Federico is a respected genomics expert with a focus on evolutionary and conservation biology. We're really esteemed to be here to be able to listen to Dr. Federico giving a talk on advances in biotechnology and de-extinction, assessing economic benefits and challenges.
Thank you for this very kind introduction. Just give me a minute. I have a problem sharing my screen. All right. Can you see my screen? Fine.
Yes, it's very clear.
Thank you. All right, good afternoon, everyone. My name is Olivier Federico, and I serve as the Director of Genomic Resources at Colossal Biosciences. I would have loved to be here with you this week, but my schedule didn't allow me to do that. But I'm sure I will find another time to visit and to interact with you guys. I'm incredibly grateful for the opportunity and the invitation, especially Professor Anne Mugai to invite me. Today, I'm going to talk about the economical impact of extinction and conservation. And I'm hoping that after this presentation, you will see the link between conservation, the extinction, and economy. Let me close this window here. Oh, damn it. Sorry. I cannot figure out how to close this. All right. More than 30 years ago, in 1993, E.O. Wilson, you probably all know him, already observed a very scary trend. He stated in his seminal book, Diversity of Life, if past species have lived on the order of 1 million years, in the absence of human interference, a common figure for some groups documented in the fossil record, it follows that the normal background extinction rate is about 1 species per 1 million species a year. Human activity has increased the extinction between 1,000 and 10,000 times over this level in the rainforest, but reduction in area alone. Clearly, we're in the midst of one of the great extinction spasms of geological history. So 30 years ago, he has already observed this trend. And unfortunately, as you can see on the plot from, let me stop sharing and here we go. As you can see on the plot on the bottom right, by Ceballos et al. In 2015, published in 2015, this alarming trend shows no sign of slowing down. And in mammals, in birds, but also in vertebrates in general, the extinction rate is increasing. The number of extinct species is skyrocketing across the tree of life. The whole scientific community agrees that we are either on the brink or on the midst of a mass extinction event, the sixth one. So if you deep dive into more details about the sixth mass extinction, it becomes more scary. And here are some number of IUCN. Currently, staggering 38% of all species are on the brink of extinction, and 50% of primates and 20% of all mammals are at risk of extinction. About 50% of the mammal species are already in decline. This crisis is not only limited to animals. Plants are affected as well, with a roaring 68% of species are facing extinction of plant species. So the United Nations has recently declared that nearly 1 million of plant and animal species are on the edge of extinction. Studies like the paper from Thomas et al. In 2004 predicted that by 2050, roughly 30% of all species could face extinction. Other studies are less optimistic and even estimate this to be at nearly 50%. So this is a pace hard to keep up with, and honestly, that exceeds the capacity of traditional conservation efforts. We cannot reintroduce or backbreed animals that fast. So this is a big issue, and beyond our moral obligation to preserve our environments and the intrinsic value of nature, we all love nature, we all care about our environment. There are tangible economic ramifications as well. For example, pollution, resource overexploitation, and biodiversity loss directly impact human health and the global economy. So here a quick summary slide about what I mean is like numerous industries rely heavily on the extraction of resources from forests and oceans, as well as the essential ecosystem services like clean water, pollination, and climate stability. So according to UN Principle of Responsible Investment, UNPRI, more than half of the global GDP, totaling about $44 trillion, is directly or indirectly connected to natural ecosystems. That includes food, materials, and energy sources. In addition, the Boston Consulting Group, or BCG, estimated that the annual cost of declining ecosystem is totaling about $5 trillion worldwide. So again, the economic consequences of species loss and collapsing of biodiversity are not just environmental concerns, but are pressing economical issues as well. More specifically, biodiversity loss threatens ecosystems, affecting production capacity and disrupting supply chains. Ultimately, the collapse of ecosystem will drag down businesses, industry, and entire economies. I know it's scary, but we have to be aware of that. And just to give you a few examples, consider these three potential impacts. Fundamental to food security. Well, animal pollinator species are crucial for agriculture. 15% of these species face extinction. If we don't stop this trend, global food security will be jeopardized very quickly. Emerging diseases. More than 75% of new diseases are from zoonotic origins, right? We all experienced that very recently. It's mainly driven by human activities, and more resources are needed to fight these new diseases, right? So that creates some sort of feedback loop leading to future pandemic. Halting deforestation and species extinctions is vital to prevent such crises. And connected to that, the medical breakthroughs about it's pretty impressive because in cancer research, about a quarter of all newly approved cancer drugs come from natural resources. So if we don't protect the environments, then these vital medicinal breakthroughs are at risk of never exist. Okay, so I drew for you a picture, a very dark picture, and I would like to give you a small hope here. At Colossal, we're working on de-extinction, and how is that connected to economy and conservation? And I hope in the next slides I will be able to convince you. So what is de-extinction? So there is several ways to define the extinction, but at Colossal, because there are three main ways to approach the extinction, right? We can do the extinction by backbreeding, by cloning and by genome editing, or any combination of those. So from colossal perspective, our company, de-extinction is a process of creating an organism that resembles and genetically similar to an extinct species by resurrecting its lost lineage of core genes to fill a vacant ecological niche. And that's a very important concept because we not try to recreate Jurassic Park, we try to fill biological niches, ecological niches that will be helpful for the environment. So genome engineering technology already exists to make these species thrive more today and make them more resilient. But not only we're going to recreate these well-adapted species, but we're going to make them better to be able to be more resilient and make ecosystem works more optimally. So why am I talking about the extinction while discussing conservation efforts and economy? Well, our approach at Colossal for restoring extinct species has many ramifications. And these ramifications are coming from our mission of developing a large array of tools, techniques, and technologies to tackle this problem. So, for example, here for the Mammoth project, we develop assisted reproductive technologies. We optimize multiplex gene editing. We work on disease, and because we don't want these animals to be prone to disease, so disease mitigation. We perform biobanking, organized biobanking. We use new computational approaches such as machine learning and AI. We provide some conservation genetics new tools. And we're generating data that can directly benefit conservation ecosystem restoration. We're producing a vast amount of data that is not only for the species we're working on, but also other species that are connected to it. So our scope and reach goes beyond the extinction, really. So I'm hoping that with the following slides, I will convince you again that the extinction project can have a significant impact on conservation and consequence on the economy. So Colossal, you may or may not have heard about us. It's a company that has been created about 2 years ago, medium-sized company, about over 30 employees. We collaborate a lot with a large set of academic partners and conservation partners. And we have 3 main projects we're working on: the woolly mammoth, the Tasmanian tiger or thylacine, and the dodo. So you may have heard of these projects, especially the mammoth project, but what you may not know is how they intertwine with conservation efforts. And I'm going to walk through each project with you. So the woolly mammoth project is probably what you've heard the most. The project is led by Eliana Isoli. It's the first project that Colossal announced and started about 1.5 years ago. So it's way in progress. The woolly mammoth went extinct about 6,000 to 14,000 years ago. And well, that's old, right? But not that old. Fortunately, we can access to a large number of samples and usable DNA that we can do our work with. So we're generating the mammoth genome and things like that. The goal of this project is not to recreate an identical mammoth, identical to the wolves that were on Earth like 6,000 years ago. Instead, we want to create a mammoth proxy, or more specifically, a core-resistant elephant with all the biological traits of the woolly mammoth. And this mammoth proxy will fill the vacant niche of the original woolly mammoth. That sounds crazy, right? But why do we want to do that? Well, I described in Ratanen et al. In 2022 in the Nature paper, in the last 40 years, temperature in the Arctic have increased 4 times more than the global average, accelerating the melting of the permafrost. If the permafrost melts, it releases CO2 that was previously sequestered in the ground. And the release of CO2 is a significant contributor to the greenhouse effect. So the idea of the mammoth proxy is to have this new organism trample moss, shrub, and uproots in some areas, thus creating an ecosystem of grass and not tree. It sounds counterintuitive, but grass absorbs less sunlight than trees, keeping the ground temperature cooler and preventing the release of CO2. So you can imagine all these large mammoths roaming on a clear terrain of grass, and they will pack the snow above the soil, insulating the frozen ground and helping maintain the permafrost. So how does it sound? A little unrealistic. Well, we can see a glimpse of this effect in this project called the Pleistocene Park that has been experimented in Russia. They reintroduce large mammals to an area. So they reintroduce camels, moose, fur goats, fur oxen, and plain bisons. So a study published by Beer et al. In 2020 shows that the introduction of these large mammals in the park reduces the soil temperature by up to 8 degrees Celsius. That's a lot, right? So these very large herbivores are not as large as mammoths, of course. So the question is, okay, it may work, but do we have an ecosystem that will be able to sustain a mammoth proxy, right? Ultimately, that's what we want. While mega herbivores' capacity critically depends on digestible biomass available for foraging intake. We recently published a study in scientific reports estimating that the North Slope of Alaska could, in theory, support about 72,000 woolly mammoths. It's actually more like between 64 and 80,000, depending on their size. This is plenty. We won't produce 72,000 mammoths, but this is plenty of space to be able to rescue the permafrost. All right. Well, this is a mammoth pipeline. Let's skip a slide. Yeah, I think I skipped a slide, but that's okay. This is a rough mammoth pipeline. So our pipeline works this way. We acquire and sequence mammoth samples. We generate T2T genomes of Asian elephants and others, because that will be a template. We identify core genes and identify candidate edits. We do cell and genome engineering, and we work on surrogacy, like artificial insemination and artificial worms. That's a project we work very actively on this. Then what we do, we collaborate with a lot of specialists from zoos and zoo partners to make sure that these animals are raised and in very proper environments and ethical environments. And eventually, we're going to collaborate with conservation partners for the rewilding. All right. So in another like rough overview of our pipeline, to repeat a bit what I was saying, but along the way, de-extinction paved the way for a suite of scientific advancement that would benefit other species, right? So we call them the beneficial species. To work on the mammoth project, we're going to sequence genomes of the forest elephant, the savanna elephant, the Asian elephant. We're going to work on conservation genetics of African elephant or Asian elephant. We do some biobanking. So it directly impacts other species that are not the mammoth proxy. And I forgot to say, we're surrounding, of course, ourselves with experts from the field in conservation and zoo. So we, in collaboration with the VGP, the Botanical Genome Project, we generated 2 very high quality genomes of the African elephant and the Asian elephant. And this is obviously free and all the public can access it. We actively working on the forest elephant. And as today, we finally fill the gaps of the genome for the African Asian elephants and made these genomes T2T. And what we published like about 2 months ago in 2024, a paper where we state that for the first time, we were able to derive induced pluripotent stem cells, iPSCs, in elephants through a two-step process. I will not go into details, but this really opened the door to not only for us to achieve our goal in our project or the Mammoth project, but for conservation, right? From arterial cell isolations, you can now generate pluripotent stem cells that could be used for cloning and for editing. Another aspect of our work is a collaboration with Paul Ling at Baylor College of Medicine. So the EHV, the elephant endothelial-tropic herpes virus, it's responsible for about 1/3 of elephant deaths in human care, right, in captivity, with a 65% fatality rate. This is a big problem. So it has been identified as an emerging threat to highly fragmented population of elephants as well. So it's not now there is evidence that it's not only critical for captive animals. So we collaborated with Dr. Paul Ling to develop an mRNA vaccine. And as July 29th, 2024, we test a 40-year-old Asian elephant at the Houston Zoo. Was the first elephant to ever get this first herpes virus mRNA vaccine. So that was a very breakthrough, and we're very hoping that that's something we can confirm and develop more. The second project is the Thalassin project, and I know I'm running out of time, but I'm going to go fast. The Tasmanian tiger, Thalassin 1 in 6, but in 1936, mainly due by human activity, hunting. The lead project of the Thalassin project is Sarah Ode. She's the Director of Species Restoration and Thalassin Lead. So why the Thalassin? Why the Tasmanian tiger? Well, very similar to I thought with woolly mammoth, the Tasmanian tiger plays an important, a crucial role in the ecosystem. So if the Thalassin is an apex predator, was an apex predator that was controlling population of Tasmanian devil, for example, that were controlling the forest health. So without the Tasmanian tiger, we have overpopulation of some species, and we have a risk of fire and things like that, right? So an apex predator, and there was no other apex predators in Tasmania. So that was a very important species. So we're generating a quality reference genome T2T for all the indigenous dasyurids, so related to the Tasmanian tiger, the quoll, the nut, the devil, numbat, Articinus, and Pascagale, and they're all publicly available or will be publicly available. We generated assisted reproductive technology for all marsupials and developing exopouch technology at this moment. So as I was saying, without the presence of an apex predator like the Thalassin, to eat the old and the sick of Tasmanian devils population, diseases are spreading very quickly, and others, and this is a very bad example that another speaker, like a few minutes ago, mentioned, the Tasmanian devil facial tumor disease. There's a way to combat this, but it's mainly due to overpopulation of the Tasmanian Tasmanian devil population. We're also working on engineering thalassines and other dasyurid species to be resistant to bufotoxin produced by the invasive species, the cane toad. The third and last project is the Dodo project. The Dodo went extinct in 1662 due to human activity in invasive species like hunting, monkeys, pigs, rats, goats. All these animals contributed to the extinction of the dodo. Dodo is endemic to Mauritius Island, so and it's a flagship project for all of our avian conservation projects. So we use the dodo as a platform to develop new technology for other birds as well. Its closest relative is the Nicobar pigeon. So are we making a dodo? So we use PGCs, which are primordial germ cells that migrate to developing gonads and later differentiate in mature oocytes or sexual organs. So the way we do it, we collect PGCs from a topless species, like I was mentioning the Nicobar pigeon, for about day 2.5, while cells are secreted to the blood. We collect these cells, we grow them, expand them in culture so that we have enough cells to edit. We edit these PGCs, introduce dodo variants to recreate the dodo phenotypes, or fix disease alleles, or things like that. We then inject edited PGCs into surrogate host embryo. So this surrogate has genetically to be engineered so that his own PGCs will die, will apoptose. So when we inject a new PGCs, it will replace the old ones. Surrogate could be a chicken, could be a pigeon, and we let them grow, adulthood, and mate with one another. And what they're going to do, they're going to give birth to a dodo, or going to hatch a dodo. So this pipeline can also be applied for any birds, and that's what we're working on. So while we're interested in birds, like in 2023, in the USA alone, 21 species of birds went extinct, right? 21 species went extinct. About half of them were birds, and mostly from Hawaii. So all these birds need to be rescued, and we need to develop tools to be able to biobank PGCs and be able to clone them, edit them, and de-extinct them when we're ready for it. So we involve in a lot of conservation effort for birds. We partner with the Mauritian Wildlife Foundation for the Dodo project. Our work doesn't only include like the extinction of the bird, but we also include ecosystem restoration efforts, where there'll be no invasive species to reintroduce the dodo, but they also benefit other species. The example here we're working on is the pink pigeon. This pink pigeon is predicted to go extinct in the next 50, 100 years if we don't do anything. So this is a project we're working on in collaboration with the Mauritius Wildlife Foundation, and we're going to set up a lab in Mauritius. We have signed an MOU agreement with the IUCN Pigeon and Dove Specialist Group and BirdLife International to collaborate on a number of pigeon conservation projects as well. Like another bird we're working on is the tooth-billed pigeon, the Manumea, for the Samoa Islands. It's a close relative of the dodo, and it has been seen in the wild for about 10 years. So we're working on finding it and de-extincting it as well. So we have a lot of also in situ and ex situ conservation effort for the Nicobar pigeon. So another example of like conservation effort is northern white rhino. We're working hand in hand with BioRescue, dedicated international team to employ genetic engineering technologies. This effort includes sequencing all available northern rhino specimen to identify lost genetic diversity and restore it by leveraging genetic techniques. As you know, there's 2 northern white rhino left and the 2 females, right? Using genetic editing on biobank specimen is the only route we can take at this point to rescue the species. We also work in the Victorian grassland earless dragon in collaboration with Zoo Victorias to aid the recovery of this species that has been extinct since 1969, but just rediscovered in 2023. We're supporting the establishment of a conservation breeding program at the Melbourne Zoo. This population already laid 50 eggs. We're very excited about it. We perform genomic sequencing of these lost dragons to assist with breeding recommendations. We're also generating a reference genome for the species. So all this work is not done in isolation. This is our current global conservation partners, and we really listen to them and try to assist these efforts as much as we can. We believe that conservation efforts is a multi-pronged approach. We can use multiple tools, and one of them that Colosso decided to use was genome editing for the extinction, and that's something we just started, but very optimistic we will succeed. So to go back to the initial question, what is the economic impact of the extinction? Well, I'm sorry because my presentation will be choppy. It's very early for me in US, but the extinction impact the conservation through the tools we develop for the extinction can be used. Conservation will have an effect on ecosystem restoration and will have an economic impact. We develop the tools that help conservation, but also our toolkit will benefit medical biosciences, like if we develop better tools for gene editing. And that will impact the economy as well. And the extinction will also directly impact the economy, right? So hopefully with this presentation, I was able to convince you that the extinction is really part of conservation efforts and that impacts economy and can help economy quite well. On this, I thank you for your attention and I can answer any questions. Thank you.
Thank you very much, Olivier. I recognize the fact that we got you up really, really early. And we appreciate that you're looking quite awake despite the early hour. This is your time to ask Dr. Olivier questions, Cecilia and also Mr. Kelvin. Any questions? I'm ready to take a round of 4. And then, yes, we have a hand up there. We can also look at some of the questions that are coming in online. Go ahead.
Good afternoon. I'm Patricia, a student at JQuat. My question goes to Cecilia. What is your take on how the FDA is kind of approving lab-based meat? Like, yeah, the grown meat that's in the lab nowadays, chicken and beef. Thank you.
My name is Esther, to Dr. Cecilia. I think it's a question to all of us as scientists. How far can we go with the genetically modified organisms? Do you think in the near future we might reach a dead end? Maybe as we look forward to the positive impact of genetically modified organisms, what negativity can we anticipate in future? And I think the project that the African BioGenome is doing is very essential, sequencing the native plants, the native genes, the way they are, because as some of it, maybe we are looking at drought tolerance in maize, and we come up with good, very good genes, and maize can grow with less rainfall. But what if this maize now starts growing like a tree? Like maybe it's extending too wide. What is the impact of GMOs in the future? Thank you.
Thank you very much. I might ask the question on behalf of Professor. She used to ask it so much many days ago. You realize that the whole essence of genetic engineering is when some societies had achieved what we call the diversity plateau, because there was a lot of extinction in their areas that they didn't have alternatives for crossbreeding to create a new sort of something native naturally. So in the essence of the African BioGenome Project, where we are talking of conserving, yet in the same biogenome conference, we are still talking of editing. Okay, so there's a way in which nature and nurture works to give us solutions naturally. In this case, then, can't we just assay for the natural solutions instead of tinkering with what nature has given us to create alternative artificial solutions to natural problems when nature has already given us that? Then to the friend from the US, the genomes you are creating, what do you think is the impact of the founder's effect? Because maybe you are dealing with, you are trying to create genomes from populations in a particular area, yet there are other populations that went extinct in a different area. So then how sure are we that the diversity we are creating is representative sort of what was there or what is just found within that particular ecosystem. Thank you.
Thank
you. Okay.
Should I
Eric,
answer this question now, or do you want me to do that?
We have one more question and then you can answer.
Okay.
So, okay, I just have an input to Mr. Kelvin's actually presentation. And I would actually, yeah, the project actually on waste management, yeah, it's a new thing, yeah, and it's encouraging. And actually my concern is, you know, when we have such kind of, you know, projects, so is how practical can we make it? Now, when we target industries, let's say like we target learning institutions, and then we install, you know, the models, yeah. And then we also target industries, let's say the hotels, yeah. And then we also target the learning, you know, learners from primary school. Now we enlighten them as, you know, they proceed with education. And then they also, you know, it's like a projecting to the future. So with these kids, when they are raised and they know how to manage waste, we can also help, you know, in conserving environment. Yeah, in Nairobi, actually, waste management is a big problem. And I feel like before we go to the pub, yeah, let's target these industries and then later on we go to the population. Thank you.
Thank you, Dr. Olivier. There's also a question from ourselves regarding acceptability of your project of de-extinction to policymakers. And I see you have won over the conservationists. I wonder how receptive they were in the beginning, and then now the policymakers and the ones who make all the decisions in governments, how accepting have they been of the project? You can go ahead with your answers now. Thank you.
Yeah, thank you for the question. Yeah, this is a very good question. I mean, this is something we actively working on, and we try to have support and agreements with local governments about editing projects. Like I was saying in the presentation, we working with the Mauritius government right now, or affiliated institution with the government. Now, in terms of policies, it's tricky, right? Because each country will have different policies about what we can do, what we should do. And there is nothing really in place right now in various policies. And we're talking to higher ups a lot, try to figure out the best way to make this happen without violating any laws, but also promoting new political decisions that could help conservation efforts. I'm not sure. Sorry. Go
Yes,
ahead.
we had the answer. Excellent. And there were a few other questions that were addressed to you. You can go ahead to answer them.
If I can remember the questions now. Can you remind me what question there were? Sorry, I lost track.
Sorry, I wanted to know the effect of the founder's effect.
Right, that's a great question. And that's something really we actively working on, and especially in the Northern White Rhino project. When we create one animal, and even if it's a clone, there was a founder effect, and there was a huge bottleneck. And especially when you get the samples and when the animal just got extinct or in danger, you have this gigantic bottleneck and all these deleterious mutations accumulation, this deleterious load. So what we try to do using AI and other tools is to identify what region of the genomes we need to edit to restore historical variants, right? So they go to a museum, collect these old specimens, do ancient DNA work on those, identify what variants were previously existing in these ancestral populations. And we try to edit them back in the new population to avoid this founding effect, because that could be very bad.
Practical, right? Okay. Well, let us give me a chance to ask.
Oh, I was muted.
No, we can hear you. We can hear you. We just
Okay,
muted
okay.
us. Yeah.
All right. Are there any other questions? I hope I answer your questions. But
I think there's also the question about nature reinventing itself. I think Dennis also asked why interfere with the nature.
oh,
Sorry, nature has a way of reinventing itself.
right.
So why bother interfering with it anymore?
Yeah, nature, life on the way, right? Well, I mean, this extinction event that is happening right now is caused by humans, right? And that, I'm not saying that to make ourselves feel guilty. I say that because that increases the pace of the extinction, right? It's going so fast that nature doesn't have time really to adapt because it takes several generations in natural populations to adapt to environmental changes. And that's going too fast right now. And the only way we can counter that is using scientific approaches, editing approaches, try to slow it down at least, because nature won't have time to do that. That's my opinion.
Thank you. Thank you very much. Dr. Cecilia, you can answer, and then after that, Mr. Kelvin can answer.
All right. It was said you ask tough questions. So I'll start with the question on biodiversity, conservation, and gene editing by Dr. Dennis. So your question is, in the same conference, we're speaking about conservation and we're speaking about genome editing, right? So when we speak about conservation, biodiversity conservation, my take is we are speaking about understanding what is occurring in different ecosystems, how the different species and organisms are interacting, how they support each other to thrive in this ecosystem, and how we can play a role to ensure that we maintain and encourage this biodiversity in these ecosystems. Now, when we speak about gene editing, we are looking at the places where we are having gaps, we are having problems that we need to fill. And when we speak about gene editing as a function of, let's say, crop improvement, sometimes we speak about GMOs. When we speak about editing, we think that we are doing some magic and playing God. But if you think about it critically, when we do grafting, right, we take the root part of a tree and tie it up to the top part, what we're expecting to happen is actually an interaction of the genomes that will end up producing a new variety. So it is genomic editing has been happening over time, but as a function of nature. But then because of our research and build-up of knowledge, we have found tools that can help us accelerate this process, right? Of course, we make mistakes, but before the FDA approves something, before WHO approves something, it has gone through a process that is quite rigorous, from lab to trials to testing, to make sure that its impact on the end consumer, be it animal, be it human, be it environment, there's a reduced impact of any adverse effects. So in a way, I'm trying to answer the multiple questions diplomatically. So GMO, we have been doing it since our grandfathers. They just knew how to do it using the natural plant. But we have found out we have DNA, which we can edit and accelerate this process in the lab setting. But we don't do it blindly. We also apply all these stop gaps to check. Are we going to produce meat, protein, that is going to cause an allergic reaction in the consumer? In that case, let us go back to the bench and reinvent. Yes. Which other question do I have? So the FDA approving meat and other food products, medicines, for example, that are coming out of genetic modification, I believe it's such a rigorous process. And the meat that I consume, when I eat chicken or when I eat maize, I consume the protein. Yes, the DNA is in there, but it is not expressing. Otherwise, I'd grow like leaves on my ears. It is the protein that is broken down for energy, right? I think I have answered all my questions. Is there an end limit to the impact of GMOs? If we keep doing it right, we keep mitigating the impacts. Our limit is as far as we can imagine.
Thank you very much, Mr. Kelvin. Yes, here he is.
I think before Kelvin, there's a question for Cecilia. A burning one.
Yes, my name is Stephen Katana. And I wanted to ask you, because there's a point you have said that we can mint money from the crop diseases that are currently a burden to farmers. So my question goes, we humans, when pathogens infect us, our body tends to produce antibodies so that it fights back the antigen. So in the plants, I'm not a botanist, but I'm very sure that plants do not produce antibodies. So in the case that we have plants that have been infected by pathogens, let us say tobacco mosaic virus, how do we then go about producing vaccines that will enable the treatment of those plants so that in the case of reinfection, that plant does not, that plant defends itself against that infection? So how do we go about it?
Okay, thank you for that question. So generally, we know plants are infected by the symptomatic presentation, right? Either there's wilt or there's stunted growth or there's low production. What I know has happened in this space is we study the pathways through which the infection occurs. So which pathway is the microbe, for example, infecting? And are there ways to limit the efficiency of the microbe to infect? I'll give an example of potato, I think wilt, potato wilt or blight, the one that causes the potato to rot in the ground, yeah? So a research has been done as part of actually sajorec project, where the scientists look at which bacteria causes this rot, and then which bacteriophages can infect this bacteria. That is one way that they have created value out of phages. So they're able to generate these phages on the bench, bring them out to the field and spray onto the potato plant before putting it in the ground. So the phages have colonized this seed, and any bacteria that tries to infect this potato will be attacked by the phages. Remember, phages are not able to cause disease in humans. So at the end of the day, I think that is a safe pathway. So that is one. So it is a case-by-case scenario depending on what you're looking at. So you have to apply the scientific method. Even if we are going to find the money in the end, you still have to apply the scientific method where you ask your questions, you formulate your hypotheses, you test, and then when you have your results, you validate, and then can go into the production pipeline of having a product. I hope I've answered your question. All right, thank you.
And on now to Taka.
Yeah, so just to recap on the question that you asked, it was feedback on the deployment structure that is starting from a younger mindset and then growing out the deployment. And I think that's a brilliant idea. And that's also what we ideally have as a bigger goal, because the goal is to, what we're actually doing in essence, as much as people see climate action, is we are actually focused on mindset, behavioral change, right? That's what we're actually doing at the very basic essence, right? It's you're incentivizing someone in almost like a research purpose, right? You incentivize, then you see the result, and then you consistently grow based on that. So that's actually our focus at the moment. So we're beginning our deployment with Coca-Cola for mass production, and it'll be heavily within universities because we believe the younger population will have
a higher adoption rate based on what we have in terms of the users that we currently have. And then also because as this younger generation adopts the technology, they're also very quick to disseminate that to their parents and also to a generation that may not be able to understand the technology. So yes, thank you.
Thank you very much. Let's give our presenters a round of applause as we thank them for having given us this very, very informative session. Just as, by the way, we are also trying to be very green. So we've given you recyclable lanyards. So you can use it to measure things if you want, but you can actually go and use it as your new name tag. And we thank Inkaba Biotech for donating them. But you notice our name tags are different. We haven't used the plastic ones that you normally find in conferences because these are business card holders. So young scientists, you should have business cards. And if not, we have given you an incentive to make some business cards and put them in there. So you don't need to throw it away. You can actually reuse it after this conference, but come with it tomorrow. And now I want to hand over to Dr. Kuja Josiah to introduce our next speaker.
Thank you. Thank you, Cecilia, Olivier, and Muriyoki. Next speaker is Kiran Chandran. Sorry. Chandran is a genomic application specialist working at AGBL and specializing in training, protocol development, assay design, and troubleshooting. He has got over 13 years of experience in the biotech field. Kiran excels in next generation sequencing, third generation sequencing, single cell sequencing, and spatial transcriptomics. He leverages his expertise to drive innovation and support customers in advancing genomics research and achieving their research goals. Kiran is here with us. Feel most welcome to take us through your expertise.
Good afternoon, everyone. My name is Kiran Chandran, and I work as an application specialist at AGBL. I am based in UAE, and I cover most of the regions, including Africa, GCC countries, as well as like Lebanese countries. So I work everywhere as an application specialist for AGBL. Yeah, so my topic for today will be about AGBL and how the solutions from AGBL can help in precision genomics and in biogenome analysis. So that will be my presentation. Yeah, so maybe I will start with AGBL. AGBL is a distribution company. So we are distributing different laboratory equipments and reagents in different parts of the world. So our main aim is like the mission of AGBL is to improve the lives in emerging markets. By emerging markets, we mean like emerging countries like Africa, GCC countries, Lebanese countries like Turkey, Lebanon, then Jordan, these countries all, we want to improve their lives through providing like novel biotechnological solutions. So this is our aim. And for this aim, we partner with different manufacturer companies in US and European market, and we make these devices and reagents available in these emerging markets. So as a company, we are like 200 employees. We have like 19 offices. Our main office is based in UAE, so that is the headquarters. And we do have offices, like 19 offices in like different countries. This include like African countries like East Africa, countries like North Africa, South Africa, everywhere. And in like East Africa, we have like 2 offices. One is located at Nairobi, and the other one is located in Kampala. So these are the 2 offices what we have in East Africa. Yeah. Yeah, you can make it any question. Just in case you're thinking of something from his words. Okay. Anybody thinking about myself? I'm friend too. He's my good friend. So just for the speed talks, we have got 2 posters which will be telling us why you must go and see your Susan and Susan and Solo. So we have the question and then you have like why yeah, I mean and other questions for Susan and Soro? What's the intention of it? The African BioGenome Project. Maybe you can skip some of the slides which I already go through. Yeah. Yeah, next one. So these are the 19 offices which we have. And like, as I mentioned, we are like more than like 200 people. Then go to the next slide. And like around 35 nationalities work in our company, and around 25 languages our employees can speak. So it will be helpful in dealing with the different markets. Yeah, next slide. So the AGBL divisions, it is divided into multiple products, like product lines. So the first one is the diagnostics and industrial. So this line covers mostly like food pathogen detection products from our side. Then comes the life sciences. So this is the major part of our business. So it is divided into 2, like genomics side and cell biology and molecular biology reagents. And we also have a services section. Like this is, we have a lab called Genomics where we do like precision medicine tests. We do like wellness treatments, like hydrogen therapy, HBOT. So these type of treatments we do for the patients. For that, we call it as the services part. Now coming to the next, to the genomic products. Yeah, so the first genomic product which we are handling is from GeneMind. So GeneMind is a NGS sequencing manufacturer. They are based in China. They manufacture 3 different types of NGS sequences. The first one is the FastaSeq 300. So this is a low throughput sequencer, mainly used for shallow, low pass whole genome sequencing and for targeted applications. And one of the important thing about this FastaSeq 300 is like more than 85% of the bases are above Q30. The second sequencer is the GenoLab M. So this is a mid-throughput sequencer, which is comparable to NextSeq 550 in the Illumina lineup. So this mainly almost everything you can do it, including like targeted sequencing, whole genome sequencing, whole exome sequencing, everything can be done on this sequencer. Then comes the sajorec 5000. So this, when you compare with the Illumina, this is compares with the NextSeq 1000, 2000 series lineup. And this is the high-end sequencer, which is coming from GeneMind. So this, it is for all the applications. When you think about the quality scores, like 85% of the bases are above Q40 and 90% of the bases are above Q30. So it is basically a Q40 sequencer. And we also have like a liquid handler from GeneMind. So this can be used for applications like DNA extraction for library preparation, and it works well with in combination with the GeneMind sequences. So the technology for the GeneMind, it is uses the same technology as the Illumina. So they haven't changed any patents, like only few patents they have changed with the detection systems. So the technology it uses is the solid phase bridge amplification, similar to the Illumina technology. It is the same SBS technology. For the differentiating this technology from the other NGS manufacturers, they call it SurfSeq, which is like surface restricted fluorescence sequencing. So that is the name given. It is just a name, not much different from what is done on the Illumina sequences. And the other important thing, which it helps the Illumina people who are using Illumina, and if they want to convert to a different sequencer. So if they have already made the libraries with Illumina, and if it has like TrueSeq or the Nextera adapters, then these libraries can be directly run on the GeneMind sequencer. You don't have to use any conversion kits for running the Illumina libraries on the GeneMind sequencer. Next slide. So they have already done comparison with the different Illumina platforms. And like they have done comparisons with whole genome sequencing, whole exome sequencing, single cell sequencing, and all. So one of the outcome from the whole genome sequencing is like the whole genome sequencing done on GenoLab M sequencer from GeneMind at 22x coverage. It has a similar data performance to the NovaSeq 6000 done at 33x coverage. So these are all publications available, and you can get it from the internet. Yeah, some of the advantages, it is easy to use. As mentioned, it is compatible with the Illumina, TrueSeq, and Nextera libraries. And there are some end-to-end solutions available from GeneMind. The end-to-end solutions means from the sample collection to DNA extraction, library preparation, sequencing, and data analysis, everything is coming from GeneMind. So this includes like NIPT, PGT applications, whole exome sequencing. Then they have like specialized the end-to-end solution for tuberculosis called TBT NGS. And for whole genome sequencing, also they have an end-to-end solution. But the amount of data analysis is restricted for the whole genome sequencing. You can get only like in the secondary analysis part, you can get only like WAF files. And the other thing is the data which is produced from the GeneMind, it is accurate. So it is comparable to the other NGS platform available in the market. Then it is flexible, like two of the devices, the SurfSeq 5000 and the GenoLab M, it can run 2 flow cells per run with 4 lanes. But the SurfSeq, the low throughput device, it is a single flow cell per run. The main important thing with the GeneMind sequencer is the cost effectiveness. So the amount which you have to pay to buy the sequencer, it is less, which is the capex cost. And if you think in terms of the operational cost, if you are doing a whole exome sequencing on a NextSeq 550 system and a GenoLab M system, then you are paying only like spending only half the amount for the GenoLab M system when you are doing a whole exome sequencing. So the applications, as mentioned, it can do NIPT, PGTA, whole exome sequencing, whole genome sequencing, then single cell RNA sequencing, all the short-read applications, it can be done on these systems. And also they have like tie-up with a company called Wiston, which is another Chinese company. So they can make custom kits also for different applications. So in Saudi Arabia, they have made this obesity detection panel, and it is widely used in Saudi Arabia for the obesity detection. Yeah. So the next company which I want to talk about is the 10x Genomics. So it's a US-based company which is mainly manufacturing reagents and instruments for single cell sequencing and for the spatial transcriptomics. So they have like 3 devices called Chromium, VCM Cytosist, and the Xenium in situ. So the Chromium is for single cell sequencing. So mainly if you want to identify like heterogeneous cell populations in a sample, or if you want to do rare cell types and all identify and all, then you go for the Chromium system. If your starting material is like a slide with a tissue, then, and if you want to do like single cell gene expression research, then you go for the spatial transcriptomics. You have like 2 solutions, VCM Cytosist, it is mainly for the discovery, and the Xenium in situ, it is a targeted approach. So you need to create targeted panels. Some targeted panels are available for oncology research, neurology research, and all from 10x. So you can directly use it. And also you can have the possibility to customize the panels and use it on these systems. So for the Chromium and VCM Cytosist, you need a sequencer. So it's a sequencing-based technology and it is mainly used for the discovery purpose. Xenium in situ, it is a targeted approach and it uses like image-based, it gets the readout, what you get out is an image-based readout. So you don't need a sequencer. And also all the data processing, it happens on the device. So a bioinformatics support is also not required if you are using the Xenium in situ platform for spatial transcriptomics. Yeah, next slide. So Mission Bio is another US-based company which is providing the single cell sequencing products. So 10x Genomics, it is based on RNA sequencing. Mission Bio is based on DNA. So here you are doing single cell DNA sequencing. In the 10x, you are doing single cell RNA sequencing. The instrument what you use is called the Tapestry platform. So this one is a targeted approach and it is mainly for the cancer research. More than 70% of the cancer research institutes in USA are using this system for MRD monitoring and for finding out the clonal heterozygosity, for mutation co-occurrence detection, and all these they are using it. And they have like panels for the solid tumors and the hematological tumors. Then you can use it as a QC for the genome editing. If you are doing the CRISPR genome editing, then to find out the on and off targets, you can use it as a QC system. And for biomarker development, you can use it. And also recently, some of the researchers has used it to identify the latent reservoirs in HIV latent reservoirs for identifying. They have used this system as well. Also, apart from the DNA, they have like a tie-up with BioLegend. BioLegend produces oligoconjugated cell surface markers. So you can combine these cell surface markers along with the DNA sequencing. Then you can do like protein sequencing and the DNA sequencing together. That is why it is called, it is the only platform with the phenotype and genotype from the, which can do the phenotype and genotype from the same cell. So VitroMaster Diagnostica, this is a company which is based in Spain. So it is mainly used for the detection and of infectious diseases. So it is a DNA flow technology which uses reverse blot hybridization technique. So they have like 3 different devices called HS12, which is a manual device. You have to do everything manually. Then they have like a fully automated system called HS12 Auto and a semi-automated system called HS24. So they have like a different kits available for detecting different infectious diseases. For example, they have like HPV detection kit for the STD, like sexually transmitted disease, for the sepsis, which is for the bacterial sepsis, antibiotic microbial resistance that's available, tick-borne, and for respiratory diseases also they have like kits, assay kits available. Yeah, so Covaris, this is also a US-based company. So this is used mainly for, it started as a DNA sharing company for fragmenting the DNA. So the technology used is called adaptive focused acoustics or AFA technology. So this uses ultrasound to break the DNA into smaller fragments. So the advantage of this technology can, when compared to the fragmentation enzyme, is like when you use this technology, you get like reproducible results. All the DNA which is shared, it will have like almost similar sizes over the time. And the same technology, you can use it for the extraction of DNA and RNA. So like using the same ultrasound technology without changing any device, using different consumables, you can use it for the extraction of DNA and RNA from different tissues. For example, for isolating the DNA and RNA from FFP tissues, this is widely used. And also for preparing protein samples, also this technology is used mainly for the GCMS analysis. If you are preparing the protein, this is widely used, and for cell lysis as well. Next. So the Genomics, it's also a US company. So this company mainly manufactures like spectrophotometers and the fluorometers. So they have the best micro volume spectrophotometer available in the market. So you can do the RNA, DNA quantification or protein quantification using like 1 microliter of the sample using these spectrophotometers. And similar to Qubit, they have like fluorometers and reagents for doing the RNA, DNA quantification using the fluorometers as well. Your Gene Health, this is another company which we are dealing with. So they have this light bench detect. So this is a 3-in-1 device. So you can do the fragment analysis, you can quantify the DNA, and also you can do like size-based, gel-based size selection using the same device. So this is like a 3-in-1 device, which you can use it for the genomic applications, mainly for the short-read applications. But there are they have partnered with the PacBio to work as a size selection system for the PacBio long-read sequencing as well to get the 20 kb libraries. So one slide back. So we also have the biomolecular systems. So this company manufactures the RT-PCRs. So it works with the technology called magnetic induction for heating and cooling. That is why it is named as MIC, magnetic induction cycler. So they have like 4-channel and 6-channel RT-PCRs. And also they have like a liquid handler for preparing the libraries for PCR. So this liquid handler is an open system. So you can use it for preparing PCR libraries for any other system available in the market. And also some steps on the NGS pipeline like fragmentation, bead cleanup and all, these are also possible on this system. So Avicenna, it is a sister company of AGBL. So we manufacture kits mainly for the oncology NGS applications, and for whole exome sequencing, we have like CE-IVD and RUO kits available from Avicenna. Next slide. Yeah, so the cell biology products, I will just go through it. So we have like Quantrix. So this company is also a US-based company that Quantrix produces different devices, and they call it digital ELISA. So the advantage of this technology is like it is highly sensitive, and you can detect like femtogram level of proteins using this technology. So this company, the instruments from this company are mainly used for Parkinson's disease and Alzheimer's disease. Most of the pharmaceutical companies who are developing medicines for the Alzheimer's, they are using the devices from the Quantrix. Then from Cytec, we have like flow cytometers. They use it is different from the conventional flow cytometers. They use a technology called spectral profiling. So you can do more colors from a single tube. With a conventional flow cytometer, you can do only like with one laser system, you can do only 3-4 colors from a single tube. But with this system, you can do like 9 colors from a single tube. And if your laser is like 5 laser system, then you can do like 45 colors from a single tube using the Cytec systems. They have the spectral cell sorters as well. Then we have like NanoSelect. This is also a cell sorting company. So this company manufactures microfluidic cell sorters. So this is mainly used when your downstream applications are like cell culture, or if you are doing RNA sequencing, bulk RNA sequencing, or single cell RNA sequencing, then RNA integrity is very important. So if you are using a conventional cell sorter, they sort at high speed and at high pressure, which is like 60 psi. So this can damage the cells, resulting in lower viability and RNA damage. So for these applications, we recommend using the microfluidic cell sorters because they sort at a 2 psi, which is very less pressure, and 2 to 0, it is very low. And so the cell viability will be higher when you are using these type of cell sorters. Assure Biosystems. So these are imaging systems mainly for the Western blot applications. So we have that. Epredia for the histopathology applications, almost everything required for the histopathology solutions, it is available from Epredia. BioLegend for antibodies, mainly for the flow cytometry antibodies and for the Western blot antibodies. We have it from BioLegend. We also have it from cell signaling technology. Lonza, mainly for endotoxin testing and for cell culture. And Genomics we have for the cell counting as well. Yeah, that's all from my side.
Yeah. Thanks a lot, Kiran. And we are grateful for your presentation. And again, we have to say thank you for having traveled all the way from Dubai to come and attend the Eastern Central African BioGenome Project. We are so grateful. And as you have seen, there's a package with Kindran, and you have a reason to go to the AGBL desk to ask more questions. Is there a burning question? But the initial told me they have a lot to ask at their desk. So, so I hope they'll visit at your desk to ask more questions. Okay, thanks.
Yeah, I want to thank everyone for inviting us to be part of the African BioGenome Conference here. And thanks a lot.
So change of speed, and now we move to the great speed talk, starting with Susan. And Soro, you can as well come over here too.
So let me give a little background about flash presentations. I hope you have realized that we are inducting the young early career scientists into the world of research, because we need to be able to pass the knowledge to the next phase. So what happens as scientists, you can be invited to a conference, and in the conference preparations, you're told you'll have 30 minutes. When you arrive, they tell you you have 20 minutes. When the day comes, they tell you you have 5 minutes. When you reach there, they tell you stop when you're on the podium. So as a scientist, you have to be versatile. Whatever information that you have prepared for 30 minutes, you should be able to present it in 2 minutes. What is the core of your work? Why are you standing up? And without complaining, because remember, you've been invited, you've been given an opportunity. Instead of negotiating for more time, what you should do is to take that extra time. So we have 2 young early career scientists who will present their posters that are out there, and I hope you will all visit. They have to present their work in 2 minutes and tell us why we need to go and visit their poster. Who is starting? Sally. Susan, please. Let's give her a round of applause.
Thank you very much, Professor Anne. My name is Susan Wanjiko from Kenya Forestry Research Institute, and my study is actually part of my MSc work, and it was on the impact of canker and dieback on Vanguaria and Patamia discolor. These are 2 native tree species that are endemic to the arid and the semi-arid areas of Kenya, and they are of major social, economic, and environmental benefits. However, their productivity is constrained by several factors, among them being the canker and dieback diseases, which is causing a lot of damage to the tissues, causing reduced growth, and also in severe cases, death of the trees. So the aim of my work was actually to identify the phytopathogens that were affecting the infection, that were causing the infection. And also I was able to achieve these through the sequencing and phylogenetic analysis of the ITS gene region, where I was able to identify most of the fungi that were actually isolated from the 2 tree species, and also confirming the virulence of the fungi through a pathogenicity test. So this research is basically highlighting the need for an integrated disease management for increased biodiversity conservation. Thank you.
So was it clear? 1 minute, 25 seconds. Yeah, so you see, you don't need 30 minutes to present your work. You can do it in less than 2 minutes. And now we have Vincent.
Okay, so good afternoon. My name is Soro Maru. So I am an MSc student at the Pan-African University. So my work is on bacteriophages. So you all know that actually antimicrobial resistance is a global health threat, and it kills around 700,000 people annually. And according to predictions, this number is expected to reach 10 million dead by 2050 if no action are initiated. So right now, I'm proposing an alternative to this antimicrobial resistance. And my solution is phage therapy. What is phage? Phages are viruses that kill bacteria. So I isolated bacteriophages targeting multi-drug resistant Enterococcus faecalis that cause a wide range of infection in human, such as UTI, bacteremia, endocarditis. So I characterized them phenotypically and genotypically, and according to phenotypic characterization, they were very stable in different temperature from -80 to 50 degrees Celsius, and they are also stable from pH 3 to 11. And I also sequenced them, and after classification, so 77% of them were novel phages. So only around 23% were known. So, and for each phages, I tested them against a panel of multi-drug resistant fecally. So I used in total 26 of them. So, and I found that some phages were able to lyse all the 26%. And one thing about phages, they are very host specific. So, and one thing that is interesting about the phages I isolated, 3 of them were able to lyse some Ephysium phage isolate. So they had a cross-species activity against a different bacterial species. Thank you.
And that was 2 minutes, 7 seconds. So you can do it, isn't it? Yeah. I actually thought his presentation is 30 minutes, but it's only 2 minutes. So you should be able to give a 30 minutes presentation, a 20 minutes presentation, a 10 minutes presentation, and even one in 1 minute and 30 seconds. So let's give them a round of applause. I think there is enough justification for us to visit their stand, isn't it? Yeah, please visit the poster, ask more questions when you're there. They went out of their way to present a poster and have it actually out there. So let us all go and visit it. So now we move on to a panel session. This is an online panel session, and it's to discuss a very topic that is dear to all of us who are members of African BioGenome Project, on how, excuse me, how we can build a sustainable future for biodiversity, genomics, and informatics in East and Central Africa. So we're investing in terms of human resources, we're investing in terms of time, we're investing in terms of projects, but how can we make this sustainable? To be able to take us through this very, very interesting panel, we have 4 panelists. We have Dr. Marie Botes from Stellenbosch University. We thank Dr. Marie for being here this year, as she was last year. She's a lawyer by profession, has worked over 20 years as an attorney in health, law, and biotechnology. She holds a bachelor's degree of BProc, LLB, and LLM. These are law degrees in intellectual property law, and also an LLD in biotechnology law. Her research interests include regulation of regenerative medicines. These are gene therapies and stem cell research, health data governance, pandemic preparedness, digital health biotechnologies, and especially in the area of neurotechnology, such as brain-computer interfaces and their impact on the fundamental rights, including online behavioral manipulation and how it shapes debates around the digital constitutionalism. So far, Mariette was a principal investigator in 1 international research grant, has published more than 50 articles in national and international accredited peer-reviewed academic journals, and has also co-authored and authored books in the area of COVID in South Africa. She has served as a co-editor for the special edition of Digital Ethics for South Africa, for the South African Bioethics Network. As the legal designer and science communicator, Mariette has also created comics about biomedical and digital ethics to create awareness and stimulate debate among a wide variety of people. Another panelist we have is Misali Katie, who is currently the ABS Research Associate at the International Livestock Research Institute. She is a lawyer by training, an expert on ABS Nagoya Protocol, a legal specialist, skilled in legal assistance, writing, research, advocacy, and biodiversity policy advisories, also applicable to sustainable food systems. She currently has worked in the area of genomic research industry capacity building initiatives. She's the chair of the Ethics, Legal, and Social Issues LC Committee of the African BioGenome Project member, and a member of the Executive Governance Committee of the Earth BioGenome Project, the DSI Scientist Network, and the Interdisciplinary DSI Group. She's very passionate about working and collaborating with many diverse people. We also have Professor Adi Feleke Desta, who is an associate professor of environmental microbiology and biotechnology at Addis Ababa University. She earned her PhD in applied microbiology from Addis Ababa University in 2014. Her research interests focus around microbial ecology, biological wastewater treatment systems, focusing on key communities involved in xenobiotic degradation. She has contributed to many research projects, including fellowships from DAD, ABCF, UAMPS, exploring microbial detection in wastewater, plants, and nutrition recovery from human urine. Since 2016, she has monitored resistance in waste to fertilizer processing in Ethiopia, which are projects that are funded by AAU, PIA, USAID, USDA in collaboration with SLU. She's a very active researcher and a publisher. We also have Dr. Laura Bertola, who is interested in how the rapidly developing field of genomics can contribute to conservation of biodiversity. During her PhD at Leiden University in the Netherlands, she applied various genetic techniques to gain insights into the distribution of genetic diversity in lions. Afterwards, they were affiliated with City College, New York, and Copenhagen University in Denmark to do this work. Dr. Laura has worked on fundamental scientific questions, as well as applied research for a range of species, primarily found in Africa, Asia, and South America. She is particularly interested in leveraging genomic data to transform the management, policy, and development of research tools for monitoring biodiversity and forensic. So we are very, very honored to have this distinguished panel speak to us about this very, very topic that is at our heart. And to start us off, Dr. Botes will give us a presentation, after which we shall engage in some discussion. Dr. Botes of Stellenbosch University, go ahead, please.
Thank you so much, and good afternoon, everybody, from a cold but sunny South Africa. It's really a pleasure to lead this panel with my esteemed colleagues. I'm looking forward to a lively discussion. I just want to inquire, would the host please share the screen again? Or share my presentation again? Super. Okay. Just waiting for it to appear. Fantastic. Thank you. So the gist of today's panel discussion will be around building a sustainable future for biodiversity genomics and bioinformatics in East and Central Africa. Next slide, please. So first and foremost, we need to have a look at the importance of biodiversity genomics and bioinformatics. And you can divide it kind of in three categories. First and foremost, the conservation efforts, then ecosystem health, and then sustainable development. So with regards to conservation efforts, the role in identifying species is very important to understand genetic diversity and guiding conservation strategies. Secondly, the second aspect is ecosystem health. And it is important because the contribution to monitoring ecosystem health and resilience make it possible to give attention to the conservation efforts. And then the last aspect that makes this very important is sustainable development. This entails supporting sustainable use of biological resources and bioprospecting. So we will have a look at these efforts or these issues within the next couple of minutes. Next slide, please. So on the next slide, you will see where the hotspots in Africa are with regards to biodiversity. And considering that this is a conference or workshop of Central and East Africa, you can see that East Africa is really blessed with an abundance of biodiversity. There's a lot going on in the Horn of Africa, Madagascar, and in the Indian Ocean Islands, the coastal forest areas of Eastern Africa, and even the Eastern Afro-Montane. So you can see there's a lot of biodiversity which East Africa has been blessed with, but there's also a lot of structural, infrastructure, sustainability, etc. issues that we need to discuss to make this biodiversity last forever. So next slide, please. In this context, considering that there's so much biodiversity in that area of Africa, we have to look at the regional context. So biodiversity hotspots, you have seen, it highlights the rich biodiversity of East and Central Africa, but challenges remain. So we should mention the challenges faced by this region specifically, which differ from other regions, Northwest and South Africa, such as a difference in habitat loss, climate change, and limited resources for scientific research. Many of these challenges are indeed shared with other regions, but issues impacting specific habitat loss and climate change and effects on that specific region of Africa are very unique to the eastern, central eastern parts of Africa. Next slide, please. Oh, this is the right slide. So first and foremost, we need to look at scientific and technical capacity. And when we speak about scientific and technical capacity, we need to look at infrastructure. There is a need for advanced research facilities, sequencing technologies, which the speaker prior to me described so beautifully, and bioinformatic platforms. Those are very important to process the collected data so that we can gain some insights from it. To do that, we need some training and education, and this is very important for the building of local expertise through education and training programs, how to use these platforms, how to use these sequences machine, and how to analyze and interpret everything. Next slide, please. The next aspect is funding and collaboration. So funding is super important. This is how we afford infrastructure such as sequencing machines, etc., etc. And this is necessary for sustained funding from both local governments and international organizations. The reason for this combination in funding is when the one source of funding may possibly dry up or be less than the other one, you still have a fallback on any other funding resources which may be beneficial to you. And to make this work, it is also very important to create partnerships. And for this reason, the role of collaboration between academic institutions, governments, NGOs, and even the private sector are of critical importance so that we can work together, pool resources, ensure knowledge transfer, etc., etc. The last aspect is ethical, legal, and social implications. If you have all the infrastructure in place, you've been trained to use the infrastructure, you received funding to make it possible, you've now entered into collaborations to share all these resources and knowledge, there's always ethical, legal, and social implications involved when it comes to the translation of the knowledge back into society. For instance, issues that are really prevalent in this area is data ownership and access. One should ensure fair access to genomic data to respect indigenous knowledge specifically. And to allow this, we need strong but flexible regulatory frameworks so that we can develop policies that support sustainable research while protecting biodiversity and local communities. Because it is a circular ecosystem where when you have trust in the local communities, you will have access to indigenous fauna and flora that will allow you to collect data and samples to do your research. And if you collect proper data and samples to do proper research, you will be able to feed that back via benefit sharing agreements and by benefit sharing opportunities back into the community. And so the positive life cycle continues. So this is just a very brief introductory couple of ideas just to kickstart us. And I've made a couple of notes or a couple of questions just to kick off hopefully lively debate in this context. So the first question is, if I come back to the importance of biodiversity, genomics, and bioinformatics, is how can genomics and bioinformatic tools be effectively utilized to identify and protect endangered species in East and Central Africa? And considering that question, in what ways can genomics contribute to the monitoring and improvement of ecosystem health and resilience in the face of climate change and habitat loss? So how can we use this information to fight habitat loss brought about by climate change? So who is willing to dig into this first question from my very esteemed panel?
Yeah,
Professor.
I'm happy. Oh, sorry, go ahead.
Please go ahead. I cannot see hands, so please just unmute and fall right in. Thanks.
Well, I'm happy to share a few thoughts. Just I'm afraid I cannot give a conclusive answer because I think this is a very big question we all struggle about a little bit. At least I do. And one of the thoughts that I have about this is that genomics, the field of genomics, has developed so rapidly in the last 10, 15 years. It's really hard to keep up. And there are very exciting things we can do in a very kind of like on a fundamental scientific exciting level that are very interesting from an academic point of view. But then, of course, one of the motivations, at least for me personally, and I think for a lot of people here probably, is to actually contribute to conservation. And one of the things is we can use genomics to build genomic resources, and based on these genomic resources, we can translate them to tools that give us the answers that we need. Because when I speak to managers or policymakers and I ask them, what are the questions that you're being faced with? Often the questions are things along the lines like, are there lions in this protected area? How many are there? What is the connectivity? Is there connectivity between this area and that area? And the truth is, we don't need very advanced genomic data sets to answer those questions. But having genomic resources and having the insight from scientific work that we have done maybe previously can be enormously beneficial to translate to tools and tackle these kind of questions. So I like to look at genomics partially to answer very fundamental questions, but partially also to use those resources to develop tools that can answer questions for which you don't need this amount of data and this amount of computational resources, for instance, and which is cheap, cost-effective, scalable. I think that is very important, at least for my the topics that I work on.
Fantastic. That's a wonderful insight. Thank you, Laura. With other words, what I hear is that a basic mapping of which fauna and flora is available within a specific region is actually, I don't want to say more important, but that's needed first before you can apply your genomic research on them. Is that correct?
Well, baseline data are incredibly valuable, right? We need to know first what is there and have maybe also even a historical perspective of you know, what was historical connectivity or the historical state 50 years ago or 100 years ago. So these baseline data are incredibly helpful to have, yes.
Great, thank you. Any other comments and suggestions or side notes on this? And then I have another question with regards to regional context and challenges, because we're specifically looking at Central and East Africa today, is considering the rich biodiversity in East and Central Africa, what are some innovative strategies that can be adopted to address the challenges of habitat loss and limited resources for scientific research in this specific region? Anybody that can give me a little bit more insight into this question? I'm not from the region.
I'm wondering.
No idea.
Yeah. I'm wondering what the contribution of AI and drone technology and perhaps even satellite imagery can provide in terms of tapping into resources that are already existing without necessarily having to leave your desk. Are these strategies which are acceptable, and are they possible?
That's absolutely true. That sounds amazing, because you can stand on the shoulders of giants if you do that. Absolutely. I see a hand raised by Fikrutamiru Kanaya. Sorry for the pronunciation. Please go ahead.
Thank you to give me this chance. Really, this is very impressive program. In Africa, we need to have intensify our agricultural practice. The agricultural practice ongoing in Africa is really, it is extensive based practice, which our farmers engaged to deforest the available biodiversity. Without forest, we cannot talk about biodiversity conservation. Hence, we need to boost the yield, the quality of agricultural product. To boost that, genetic engineering and molecular breeding is compulsory. Before just we going to do that one, we have to conserve, we have to establish the exit to conservation mechanism, especially to collect all biodiversity, original biodiversity, and conserve them before we engaged to molecular breeding. For instance, now I am a student at in China. At every place, first they establish well-organized seed storage place. Then after that, they are just really using this technology for the community. In order to just establish this in Africa, first we have to conserve what we have on our hands before just we going to use these tools application before just molecular breeding, before genetic engineering. We have to conserve them. That's my forward. Thank you.
Great. There's lots of practical tools and on-the-ground activities that can be done to preserve biodiversity. I like that. There was a hand by Tobias. I don't see the hand anymore. Tobias, did you want to add something? No? Okay, in addition to conversation,
Yes,
conservation.
Yes, I was actually raising hand. That was
Okay, good.
Yeah, this
Go
is
ahead.
Professor Tobias Ezejofo from Nigeria, Federal University of Technology, Owerri, Nigeria. The hand I was raising was actually when the presentations were going on. I mean, the presentation that preceded this discussions phase. I don't know, it's still not belated now because it appears to be on the discussion table now. I had wanted some of those presenters to give me some clarifications. That was why the hands was up. So in any case, I'm joining the conversation. I just needed to find out, maybe it can be passed on to them. One, for the presenters on the West, those recycling systems. Here in Africa, I try to do a lot of waste to wealth, trying to convert some of these waste that constitute nuisance here into some useful products. And what those people did appears to be there. I had wanted to see whether I can get a catalog of what they do, and then all of those equipment, and also find out too whether they have a space for somebody to understudy them for a period 1 year or thereabout, months to a year, in order to internalize properly and then come back home to waste is a big problem for us here. And particularly the cellophanes, the waterproofs, are constituting nuisance here, blocking gutters and creating ecological problems. And we are thinking we can find some local organisms that will be able to see whether there's a possibility of handling them. So I would have loved a situation where I have a chance to interact with them on a closer range and find out their systems, whether in a few months to a year, with a view to coming back here
Thank you.
to
So
improve
we'll
on the work I
connect
do.
you. Yeah, thank
And
you
then
so
that
much.
is
We'll
one.
connect.
Yeah, that is one. The second one refers to the meat.
Tobias, excuse me, it's related to the panel discussion, because we will have a question and answer session at the end. If it's not
Okay.
related to the panels, you can
Okay,
ask it
no
then.
problem.
Thank you.
No problem. Let me stop it
Yes.
here on the question
Thanks,
station.
Tobias.
Yeah,
Thank
so
you.
Thank you.
But
I'll join the
to link to
conversation.
what
Thank you.
Tobias, thank you. To link what Tobias said, it may link to my next question is, over and above conservation efforts or innovations on the ground, how can regional and international collaborations be enhanced to mitigate the effects of climate change on biodiversity in East and Central Africa? In my presentation, I said how important collaborations are. So what from the panel, I would like to know, what can we do better or different or start doing to enhance collaborations that could enhance the effects or mitigate the effects of climate change on biodiversity in this specific region? Anyone who wants to try their comments on this?
I think from the African BioGenome perspective, we find that when we go to institutions, we don't have such a big challenge in the institution to forge partnerships with international partners outside Africa. We find that they already have a history and mechanisms through which those networks can be enhanced if they have not really been effective. But what we don't find a lot is Africa to Africa, South Africa to Kenya, South Africa to West Africa, and maybe tripartite. So when you're writing grants and they require you to partner 3 countries, it's easy for you to get partners outside Africa than getting inside, not necessarily because they're not there, but it's because you don't know them. You don't know who is doing what on the African continent, because we've not had a very good conversation around that. So I'd like to spur some debate about, is this my own personal perception, or is it something that is really true? Also, collaborations are personally driven. So if I am working in an institution and I meet Dr. Laura and we connect, then our 2 institutions are more likely to collaborate because of a personal initiative. And sometimes I wonder if these personal contacts are not there, would we be able to have those sort of collaborations? Thank you.
Great. Laura, do you want to add something to that? I see you nodding your head.
No, I think that is absolutely true. And I always felt that the personal element is really, really crucial. I was thinking also about something something else is that, so my field is very much focused on wildlife genomics. I don't really work on pathogens. I don't work on agricultural species. I don't work on human genomics. I work on wildlife. Sometimes I'm looking for collaborators in countries I have samples from or countries I have some kind of interest in. And it's really tough to identify people in my field in those countries sometimes. Now, I know that probably there are, for sure, the geneticists are there, skilled people who could maybe fill a gap in the project and could really help forming a consortium or forming a nice collaboration. But maybe they're working on human genomics or they're working on agricultural species. So these are not the people that I meet at the conferences I attend or not the papers I read. And so I wonder if there's something to gain, if there's more, I don't know if it's communication, but maybe we just need to get to know each other more, because if we don't know each other, then it's really difficult to initiate a collaboration.
Absolutely. It's as if we need a network and a check-in, like a social event, just to check who you are, where you are, and what you're working on. That's critical, yes. There's a hand by a day. I hope it's the right pronunciation. Please go ahead.
Yeah.
She's one of our
Thank
panelists.
you very much. Sorry.
Yeah. Please go ahead.
Okay, thank you very much for this interesting topic. Yeah, it's really a challenge to communicate and to network among ourselves as African countries in different aspects of the biogenome area or biosciences area to solve a problem. I usually think like, yeah, starting from informal connections and communication is great, but it's also, now we are in the era of, you know, electronic meetings, which does not cost very much compared to the physical meetings. So having more and more of symposia, for example, for me, I work on pathogens, but it's also good for me to understand about the animal genomes or cattle or agriculture, because when we try to solve a development problem, we usually have to collaborate. Because, you know, I may also need some social scientists for that matter to solve developmental challenges. So having a symposia, you know, is important because everybody talks about their area of interest. And then we start thinking outside our comfort areas or our specialty areas, our boxes, to be able to cross-think and kind of try to address a developmental issue. It can be conservation, it can be climate change, or any other issue. Thank you so much.
It's a great point. Thanks for sharing, because you have that different perspective as well that can inform and enrich your own thinking. Yes.
Yes. Can I jump in and just say that Professor Ade has brought in a point of versatility. I don't know how much an African scientist can be allowed to stick within one niche without being versatile, because as you say, we are developmental scientists. So when you're working on wildlife, you will find other challenges in the community. And if you have just a one-track mind, maybe you just focus on one area and not forget some of the others. So how versatile do the scientists have to be, especially in this area of capacity building and doing the actual research itself? So versatility, I just wanted to point out that was a point that came in that struck me as being quite relevant. Thank you.
Great point. Thank you so much. Tobias, you've got another hand raised. Do you want to add something to this conversation? You're muted. There you go.
Can you hear me now?
Yes, we
Can you hear
can.
me? Yeah. So I'm saying that the discussion on ground is capacity building is key to what we are talking about now, because the conversation has already started. The last speaker made a point. We're in the era of electronic communication. I am right now in Nigeria, even when I should have been there physically, but logistics of airfare and all whatnot made it, I couldn't come. But I'm here still hearing what is happening, which is kudos to the electronics era. And so we are in a world where communication has breached distance. So as I'm hearing the discussion on the ground, I have seen the various dimensions. She rightly pointed out, the coordinator or moderator was talking about, in an African setting, it's very difficult to focus centrally on one area because you have an environment that is bedeviled by too many myriads of problems. And so all of them, solving one is key to solving another. So you cannot but help looking into, a peep into the window of what the other person is doing in order to be able to solve your own problem. All of them, just like what we do under One Health paradigm of disease control. You look into the environment, you look into the animal, you look into the human. And then you now bring the socio-economic aspect, the governance, the social structure, economic policies, and then all of which will now tangentially or directly will impact on how well you do that aspect you think you are the expert. So I think the kind of conference being organized now is key to what we are talking about. Bring what you have, I bring what you have. We now kind of cross-fertilize ideas. And from there, you now know the aspect that I'm personally doing in Nigeria with respect to other areas that are tangential to what, even when I don't literally want to go there, but I'll be forced to touch one of those aspects in order to be able to solve my own aspect of the research. And so, as we are now talking, we are already ventilating our ideas on how to solve that problem, because today you now know there's a Nigerian who is a medical scientist, who is an environmentalist and a public health man. So with all these backgrounds, and eventually a professor of biotechnology, I now try to synergize all these things into all the researches I do. And so when I now ask the kind of questions I ask, it's just because listening to the presentation today, I now know there are people who are doing the kind of things that I need answers to. And so I'm now eager to get connected to them in order to tap from their own knowledge as to further my own aspect of my research. So I think collaboration can come through what we're already doing, courtesy of this Africa BioGenome Project conference or workshop. I think
Excellent.
that's my take.
I agree. I agree. Great point. Great point. There's also a hand raised by Daisy. Please go ahead.
Okay, hi everyone. I'm Daisy. I'm from MAST currently, but very soon I'll be a student at Queen's University Belfast in the UK. And my area of interest is parasites. Basically, I look at parasites that affect wildlife, also mainly focusing on still conserving them because we have parasite affecting them and many of them are dying, then it will be a conservation concern. So back to the question and the panel about collaboration. My point on this is that in Africa, the collaborations, we have them, they exist, but they are still few. It is not an area that has been given much attention, where you find very few people are collaborating within each other, whereby one of the reasons could be is because of the limited resources, most especially funding. So you find you have an African scientist. So when they're looking for collaborators, they will look for people who will have, for example, funding to bring on board. So, and then the other thing, we have very few platforms that bring us together to get to know each other and we try to work together. For example, I've attended a few international conferences, but you find you're like 3 or 4 people from Africa. You get the point? So if you've not had a chance to meet anyone, to know what they are doing, you can never think of collaborating with any of them. So maybe if we can come up with a platform and have more African meeting each other and then discuss about what they're doing, and then they can see how we can increase the collaboration. Yeah, thank you.
Great, very great point. So there's a definite need for a platform or a network to connect people. Great.
Yes.
And this is exactly like someone said in the comments, this is exactly what this workshop is. So perhaps we need to just continue this network going forward. So a lot of the conversations also talked about capacity. I heard the word capacity, which brings me to scientific and technical capacity. And I've got 2 points here. So what I want to hear from the panel is, what are the most critical infrastructure needs for advancing biodiversity, genomics, and bioinformatics in East and Central Africa? And how can these be addressed? Because we all say we need capacity, we need infrastructure. Exactly what does that mean first? And the second is, how can local expertise in genomics and bioinformatics be developed and retained in this region to ensure sustainable growth in these fields? So anybody who wants to enlighten me with regards to those 2 points, what critical infrastructure needs do we have? And how can we home grow expertise and retain them? Who wants to go first? What is your
I think I can.
Yes, please go ahead.
That as long as we don't provide the incentive for institutions to buy instruments and place them within reach of the scientists locally, we will continue to send our samples outside Africa. We will continue to rely on others to sequence for us, to analyze for us, and therefore we are, number one, not driving the local economy, because once an instrument is placed locally, there is a direct impact on the local economy. There'll be somebody who will be employed, there'll be reagents that will be shipped in, there will be trainings that will be done. So there's an actual benefit. But also, if we ourselves as scientists locally don't do our bit to make sure that those instruments are busy, if we continue sending samples to Europe and America and avoid the local institutions because it is cheaper to send outside, then what we do is that we harm ourselves in the long run. Of course, it will be cheaper to pay for DHL or one of the courier companies to send a DNA sample to somewhere in Europe for the sequencing to be done, maybe because they have high throughput labs there, so the actual cost of the run is cheaper than here locally. But if we don't do that, then we will not be able to build capacity locally. Secondly, my view about this is that we need to build the capacity locally, but we also need to employ them, because we have institutions don't advertise for positions. So if we train a postdoc or we train a PhD and they don't get a job and with enough money to pay for their needs and their family, then they will look for greener pastures abroad. So we need to give them good positions in farm institutions and be able to pay them so that they can have the incentive to stay home. Otherwise, people will forever be looking to go to Europe and America. Thank you.
Excellent points. Excellent points. Anybody wants to contribute to that from your own experience? Laura, you're unmuted.
Yeah, I fully agree. I'm a little bit careful because I'm not from this region. I have some very close collaborators from the region, so I feel like I can share some of my observations, but of course I don't have a very in-depth knowledge. What I see in a lot of places is, for instance, purchasing equipment or finding grants that would allow you to purchase equipment is probably relatively easy compared to actually building the capacity and making sure you have people in a lab who stay there for a longer period of time and become very knowledgeable of all the projects that are happening in the lab. I had several projects where technicians, there was such a fast turnover rate in technicians that there was almost like a permanent confusion of where the samples were and what had been processed and what still needed to be processed, which of course is a shame because people became trained and then had a great opportunity somewhere else, which of course I fully understand that they take that opportunity then. So I think one is of course making sure that the infrastructure is there in terms of equipment, but I think providing the incentives that people stay there and have good opportunities locally, which motivates them to stay there, is really, really crucial.
Absolutely. The retaining of expertise. Absolutely.
Yes, exactly.
Daisy, you have a hand raised, and then thank God, you're second. So please go ahead, Daisy.
Okay, thank you. From my experience, the critical infrastructure need that we have as scientists is we lack equipped laboratories. So in most cases, you find someone has got the knowledge, but the laboratory is not equipped for particular experiments. And in most cases, for some labs to get some equipment or to buy machines, they depend on grants from like international bodies. So you find if you don't have a grant, you may never be able to get a particular machine in your lab, because when it comes to the government, in most cases, it has a very limited budget to support that for a university, for example. And then also, sometimes after getting the machines and then sustaining it becomes a very big problem, because you'll have the machine, but getting the reagents, another, because the reagents, you have to ship them from abroad. And where would you get the money to ship the reagents to bring them to the lab? There is no money to do that. So you find it really, really difficult. Thank you.
Thanks, Daisy. Thanks for raising that. I see a lot of heads nodding. Thank God. I'm sure you have some opinions about that. Please go ahead. We can
Thank
hear
you,
you.
Mariji. Okay, can you hear me now? I'm just trying to paste something in the chat box. So tell me if you see that. Did you see anything?
Yeah, we
It's an
do.
image. Yeah, so I mean, some of us, my name is Tango Ebenezer, so it's good to see everyone here today. I think there is this idea that if you sequence outside is cheaper, right? And with so many other things. But one thing we kind of, just as Professor Anamugai and Daisy also talked about, and also Laura, is that there is need to look at the opportunity cost essentially across the value chain. And that opportunity cost mainly is, if you ship it out, then you ship out the knowledge, the ability to do those things locally. You ship out the, all of the potential maybe revenues and stuff that could have come in locally. So, but when you add all of those things, you find out that at the end, you've lost the know-how, the technical know-how. You've lost the ability, the knowledge about the species. You've lost the intellect or the science that would have actually driven the activities. And then you've lost so many things. But when you check it, you think that, okay, you're saving $2,000, $1,000 or $3,000. But at the end, you're actually losing more. So to give you an example, when we started AfricaBP, one of the ideas is to sequence, and it still is, to sequence everything locally on the ground. The idea behind that is that when you do that locally, you will unlock so many opportunities on the ground, right? And that does not mean that, within the project, we don't partner with international partners. No, we partner with international partners. We have, for instance, the African BioGenome Project in the US. We have Illumina. We have all of those cooperation. But the idea is just to kind of try to switch things in the opposite direction. How can we actually sequence it? How many papers published, genomic papers published, have African first authors? How many have African last authors? How many do you have? If you have a paper of 20 authors, how many are Africans on an African species? How many African scientists kind of publishing in this genomics journals? So those things are things that really needs to begin to happen. And in the past 2 to 3 years, we've also been able to begin to see the fruits of that. So I pasted a link in the chat box about 2 partners who actually got involved. They are actually AfricaBP partners, and then kind of bought the first Revio machine on the continent. You know, that is very, very impressive. Like that is the kind of thing we're kind of looking for, you know, through public-private partnership, you know, stuff like that. They talked about maintenance. You know, it's very difficult to maintain the systems. And also, unlike in most countries or regions, you don't tend to see maybe a constant flow of the research funding from the government or regional funding agency to the people. A public-private partnership as this provide that ecosystem and that enable environment for that to work. And just partners came together and kind of did this. But before that happened, we had to show the value in the project. The AfricaBP ecosystem had to show the value, whether directly or indirectly, by providing that ecosystem through this workshop, through sequencing on the ground. Because if we were sending the samples outside, they wouldn't have invested in the technology. You know, so I mean, if you Google online, you will know how much it costs. So the question now is, how do we take that and replicate it and scale it? That is the essence of this workshop. That's the direction things need to go. But we need to kind of then change the mindset that, oh, it's cheaper outside. No, you have to do it on the ground. And over time, it just kind of increase. So that is just my contribution to this. And hopefully we'll be able to provoke more action locally, because that is where the science needs to go for there to be an ultimate impact.
Great, thank you, thank God. That is so important. And that links to my following question about funding and collaboration.
Yeah, so just to add one more point, Malaria, is that that link I posted in the chat box, that was a tweet I did many weeks ago, many months ago, that I said, and that connects to the area things around brain drain and stuff like that. And I asked myself, I said, why don't we see brain drain in the entertainment sector in Africa? I mean, if you fly to Europe or US, you see everyone playing African music, like the you know, the Davidos and those in East Africa and those in, you know, Jerusalem in South Africa. You see it. So you see it happening. So why don't you have brain drain? So it means that in that sector, there is something that is happening there that science isn't doing. It's very simple.
Yeah.
There are ways whereby things work in Africa in terms of, you know, to give an example, the telecommunication sector, they were having these issues that the genomic sector and science was having many years ago. You know, where you have in West Africa, in Nigeria, where I'm from, you have the big, you know, you call them the National Telecommunication Agency. They have this big thing and stuff like that, and then it's a bit challenging to get things. But when we have the mobile phone creep in, it unlocks so many things. You know, in Kenya, you have the M-Pesa when it comes to finance and other areas. So what are those sectors doing that the genomic sectors needs to learn from? So I think that is the essence of that, my tweet. How many times do we see brain drain in the entertainment sector? Can we learn from them? Thank you so much.
That parallels make so much sense, yes. There's a hand by Tobias again, and then Zemach. After Tobias, please go ahead. You're muted, Tobias. There you go.
Yeah, you can hear me now?
Yes.
Yeah, I think the angle of sustainability is most times, even though I'm not from the East or Central region, yet we have a common denominator in Africa. Everything about us is almost same, except here and there, partial differences. Otherwise, we are operating under what we call under similar infrastructural ecosystem problem. So I think here, for instance, in Nigeria here, you have some equipments paid for and then locked up because of the technology, the manpower aspect of it. You see suppliers coming to dump some equipment, you make orders without the relevant technology too. So we in Africa should be making an effort to tie our requests or orders we are making should come with some periodic trainings that should come from the suppliers of those equipment to build capacity. Those who are here, technologies are here, not many are, you know, at home with some of these modern equipment. So there is need to bring in, as part of agreements in purchases, training components Again, there is need also maintenance culture. This is part of where we are not getting it right. The first phase of the supply, maybe in the next 6 months to 1 year, it won't be running. After that, the equipment packs. Meanwhile, just a little thing that may be changed here or repaired here for the thing to function, they will pack that one and will keep on crying. There's no this equipment or no that equipment. So there's need for us to have, as part of our ordering agreements, that there must be that technical component in it, not just supplying us with the equipment, but also matching it with the trainings. And then following us up in the next 6 months, the next 1 year, the next 2 years, thereabout, before we finally disengage, and within which period, the local scientists or technology managers will have been made to be conversant and very much at home with the runnings of those equipment in order to supply, in order to add up the maintenance aspect of it. We lack a maintenance culture. Most of the time, these are part of the frustrations we have here. And we did not enter into the store. You see jounts of equipment of different era purchased and dumped because of a one little problem or the other. These aspects have to be considered in order to, in terms of sustainability we are talking about.
Right. Yes. No, you're absolutely right. To link on, Zamar, you had a hand raised. I can't see it at the moment. Did you want to say something? Are you good? Okay, I don't see it. Just unmute yourself.
Yes, please. Are you hearing me?
Okay, yes. Go ahead.
Thank you very much for your nice presentations regarding how to conserve the natural resources in our environment for the future use of for our society. So my point in your point of discussion that is regarding the limitations. One is the infrastructures, of course. My point is if we are using the infrastructure in our environment, in our region, collaboratively, with the help of these collaborations, we can address a lot of students, a lot of expertises. One example here I want to raise is that the examples of using HPC, high computing resource, is one of the limitations. But these limitations can be addressed through a VPN. I do have the experience of using the cloud or the computing environment in Germany, being in Ethiopia. This by using the connections of the VPN. So in Africa, if we have some HPC system somewhere in Nairobi or in Nigeria or in South Africa, and if the students or the expertise is using through the VPN, these resources, so the resource limitation might be resolved in this way. This is one of the experience. If we develop this experience or can bring this experience to Africa, we can solve in this way. The other laboratories also the same. Maybe if we can do some inputs in the laboratories, we can solve the shortages of the laboratories in this way. The other way is exchanging the expertises. We do have a limited expertise here and there, but we are we really using these expertises to their potential? No. So if this network is trying to look what are the existing resources regarding the expertises and as well as the materials, we may solve to some extent. This is what I'm going to say. Thank you very much.
Great, thank you so much. It makes a lot of sense. Ade, you had a hand raised? And then thank God again, sorry.
Yeah, just a quick note on this. I think the question of critical infrastructure can also be answered not only in terms of having or not having the infrastructure, but also whether one is effectively and efficiently using the infrastructure we have. So I'm seeing, at least in my country, Ethiopia, I'm seeing it's a wastage of resources because every institute buys a sequencing machine. buying the machine is one achievement, but not operating it is a wastage. So I think it's important to monitor and also to evaluate whether what we have as a resource in a country or in a region is being operationalized to its fullest. Why do we have to buy, the machines if we are not operating? I'm from the university, so when people come to me to advocate, like, you should have a sequencing machine, okay, you have to buy, this and that. No, I would rather, send my samples to Kenya or to the other institutes within my country instead of my institution buying another machine. So how are we utilizing the resources we have, the infrastructures we have? Are we utilizing it to the fullest, or are we just, owning these infrastructures? That's also the question that has to be addressed. Thank you so much.
Excellent point. Excellent point. That speaks to the collaboration within Africa as well. Thank God. I'm sure you have something to say on that. Please go ahead.
Yeah, thank you, Ade and also Zamek. So again, this speaks to the vision of the African BioGenome Project. So Ade, you kind of captured this well. And when we started the project, we focused on using existing resources. So if you look at so many of the print media and stuff we've written, it's focused on using things that are existing, because the biggest problem Africa faces is not funding. It's not the absence of equipment and infrastructure. It's not limited capacity. The biggest problem is the use of those things. So you just actually took the word out of my mouth, because it's very simple. If someone is in Kenya and the equipment is in Zambia, why not send it there, for instance? Because that also helps. And which is why, for instance, this equipment we bought now is the first on the continent, and we hope that it will support other countries as well. Because the reason for the partners to get it is that it's way cheaper than what is available there in the market, and it gives you much more data. It actually gives you 4 times what you expect. So the biggest issue is, how do you address that usage culture? And to address it, there are several folds. For instance, there's a fold about, okay, let's do it within the continent. It doesn't matter where, right? Let's do it I want to collaborate with international partners, but I want the international partners to maybe be able to send more sequencing machines, because we're not talking about Africa developing and manufacturing its own machines yet. But they should be able to send, for instance, genome technologies. For instance, Africa should buy more technologies, and then rather than having the service done outside, it should be done within the continent, because that helps to create that ecosystem, basically. And I think that also goes in line with what Zamek is using around usage. And to me, that is the biggest problem the continent faces, not funding. Funding is not the issue. And so it's infrastructure. If we say we have limited capacity, we have limited, of course we know, but we know we have limited infrastructure, but the ones that are there, how have they been maximized, right? All the ones that are available, how have they been maximized? In 2021, Nzao et al. Published the paper on the state of the genomics landscape of Africa, and then they have a map of all the sequencing machines in Africa, and over 100 or close to 100 sequencing machines in Africa. But yet, when you go to the global database, the sample sequence in Africa does not match the sequencing machine, right? In terms of like other aspect of things. So, and it's quite possible that many of them kind of stay in a year not doing anything. And which is why a project like AfricaBP now kind of helps with those people, in terms of working with institutions that have sequencers. Like in South Africa, we have the ARC, in Morocco, we have the AGC. And through that, we get samples supplied to them, and they work. And of course, you see the science. Someone mentioned cross-fertilization or pollination. Then that kind of have a two-way thing. And the institution get to think more about it, and then they have people around. But if we continue to send the samples outside, then the optimum level we're trying to achieve will never be reached. Thank you, Ade and Zama. I think that kind of captures the vision of this project as well.
Great. Thank you. Thank God. I saw that someone said in the chat as well that there's a need to map the available resources and expertise in African institutions and find ways of using them optimally. Right now, there's a limited information about these resources, which is a great idea. I think AfricaBP made a good start with regards to connecting everybody in this regard. But I think we should keep this momentum going by getting somewhere a platform that still continuously connect people and update available resources, etc., etc. All right, I just want to check with the workshop organizers with regards to time. I see we've been debating now for an hour, just a little bit over an hour. Do we still have time, or what is the
Well, we
situation?
can wind up the session if you can just do a summary or a
Perfect.
session. I mean, 1 hour of conversation, deep conversations is really, really great.
It is, and it can continue till late in the night, I'm sure, but I'm sure you can continue that for the people who are physically there over dinner. The last thing I wanted to say, I think there is enormous opportunities in Africa, with Africa, for Africa, if we just connect, get to know one another, connect, share resources, collaborate. And I think AfricaBP is the kickstart of something to this effect, which may build momentum going forward, which is great. So I think we can share resources, we can share connections, contact particulars, etc., etc. There's a lot of papers coming out from people working on the AfricaBP projects. It will also give you an indication and tell you who is working on what and where they are, etc., etc. So thank you very much for every single person that contributed to this very rich conversation this afternoon. Thank you so much for that. And thank God, thank you for the opportunity of hosting this workshop and for the hosts physically in the location and the opportunity to do so. Thank you very much. Over to you.
And many thanks to you, Marietta, for steering such an excellent panel. The preparation slides really spoke to the heart and were able to spur the conversations. And Dr. Laura and Professor Adi for your insights. Of course, Dr. Thank God there, a real passionate driver of the African biogenome. The conversations were real. And I think for me, in closing, I can say that the conversation started. Not should start, the conversation has started of what we need to do differently. And I think we who are in the driving seat of not only AfricaBP, but of our African institutions need to make those positions, like Professor Ade says, no, we don't need to buy another sequencer. We need to utilize the ones that are here locally better. Have you thought of shipping samples to this university or to this country, which is just next to me? And we have laws that can facilitate that. Capacity building is a conversation that has started. It needs to continue with a very, very strong momentum. And with that, it's the only way we will be able to drive change so that we can really say that we have for Africa, by Africans, in Africa. And I thank you very much. We've come to the end of the first day of this 2-day conference-style workshop. It really has been an excellent day, excellent presentations. If I can say so myself, I really want to appreciate each and every one of you. It's 5:35 p.m. in Nairobi, and knowing the timing in Kenya, to see all of you here, I'm really, really impressed with your commitment. So tomorrow we start off a little bit earlier. We start at 8:30 East African time. And I hope to see you all for another very exciting day of workshops. So thank you very much. We see you tomorrow. Have a good, restful evening. Rest your brains, because tomorrow will be another day of drinking in African biogenome. And a round of applause as we close our day. Thank you all. See you tomorrow at 8:30 a.m. Thank you. So thank you very much. The conversations were so intense, so interesting, and I'm happy to see you all here. Please grab a cup of tea, visit our exhibition stands, look at some of the goodies that they have for you there, and thank you very much.